are about 45,000 different species of plants, with 2,000 of them being flowering plants, 20,000 of them being flowering plants. Of them, only 5,000 are confirmed to be native species. The other 40,000 either unconfirmed or alien species. By that I mean that they are not indigenous to this area and have been introduced here from a different area, either by natural means or by on purpose. But when these alien species compete with the indigenous plants for food from the soil, water or space, they may start to dominate the area and make it dangerous or difficult for the indigenous plants to live. We call those plants invasive species. According to the Convention for the Biological Diversity in 1982, these invasive plants are the second worst threat to the biological ecosystem. For anyone who's wondering, the first worst would be human encroachment and habitat loss of animals. So, why do we need plants in an ecosystem anywhere? First of all, plants provide oxygen, which is a vital resource for life. And in this same process in photosynthesis, they capture sunlight and carbon dioxide. And we consume these plants and indirectly consume the solar energy captured by the plants. This is how we get our own energy. So this is why we call them the primary food source for all living organisms. They also help regulate the flow of the carbon, nitrogen, oxygen and water cycles. Imagine how disastrous it would be if these vital substances were to remain stagnant in one place for too long. They also help to prevent soil erosion by maintaining the soil integrity, and the larger trees provide homes and shelters for small animals like birds and insects. The objective of this study would be to see the diversity of the flora in the study area, and I want to see which ones are native and which ones are alien. I want to see how the alien ones got here and which are invasive, meaning which provide hazard to the ecosystem, and how did they get here. So the study area will be the Vijayanagar Park in my hometown, which is Vijayanagar. My materials, I will be using a phone camera to take pictures of the plants, a notebook and pencil to write down my observations. The method I use for collecting samples will be convenient sampling, which means I take the easiest, most convenient samples nearest to me. The primary source for identifying the species will be an encyclopedia called Discovering Avenue Trees. That's my first source. My secondary source would be other botanical journals that I found on the internet. I didn't perform any experiments on these plants, so there is no experimental method. Now here's the study area. As you can see in the legend, the green part is the light green is the park border, and the dark green is the plant area in where I collected the specimen. The brown is the walking path. My study area is about 574 square meters in area and 132 in meters in perimeter. So I found about 13 families, 13 species, 11 families, and there are 2 repeat families. All the rest are unique. So as you can see here, this is the photo of each plant, the common name, the species and genus, and its family here. So as you can see, I made a table here of which are the native species and the alien species. The most shocking of the alien species that I thought was the Gulmohar tree, because we see it spread around in India so abundantly near parks and sidewalks, but it turns out it was brought here by the British colonists who wanted to beautify the parks and provide shade to them. Here we have the bamboo palm, which is a small grass-like palm, bamboo tree. It's used for ornamental decorations on windowsills, and it was also brought by the European colonists. Here we also have the great laurel false ashoka, the Indian cork tree, and eucalyptus. The most invasive of these plants would be the eucalyptus, and this is because it absorbs all the nutrients and water from the ground and leaves all the other plants without any water to thrive. So these are the most invasive species according to the Conservation for Biological Diversity in 1992. Not all of these I found on my research, but the most common I do have to say is lantana camera, Parthenium, which is Parthenium is a native to the Americas, especially South America, and this one is most tricky because it digs its roots deep underground and it's difficult to get out. Every single plant in the eucalyptus genus is also invasive, as I told, and also it's because they provide only a third amount of biomass that they use to intake. Here are the reference sections. And lastly, I would like to thank my school, Purna Pramati, all my teachers who helped me on this. And I'm thanking Alva's institution for providing me this opportunity. And thank you all for listening. Thank you.
Questions from students? One question. Any questions? There are no questions. I have a question.
You mentioned many species were invasive and they are dangerous to, they are posing a threat to our indigenous species. So are there any ways that we can solve this issue?
The first and foremost step we have to take to get rid of these invasive species would be to educate the public and most importantly the people who are planting these species, because most of them don't know that they are invasive and they will be planting them aside the indigenous species, so they'll unknowingly be creating a problem within the natural forests. And secondly, we can help to remove these species because, for example, the lantana is very difficult to take out along with the parthenium because they germinate and propagate very easily. They spread their pollen in the air very quickly. So I think the first thing we need to do is to stop planting it in the first place, because research has provided that the removal of these plants would be very difficult.
Okay, if we stop planting those, but you said that it will pollinate itself, no? Like how can you stop that?
It will propagate.
Yeah. Like how can you stop that?
What he means is, first of all, you avoid introducing an alien species. Even accidentally, you see that nothing is introduced. Then comes the next step of pollination. Okay. What he said is, you avoid introducing other than any native species. Don't introduce. That's what he said.
The second thing we should do is to plant more natives or indigenous species so that they can outgrow the other plants and dominate in the area where they're supposed to. So they don't have a soil erosion for the thick growing plants there.
Thank you.
convenient sampling, right? If you do convenient sampling, do you think you will get the real picture of that area?
Actually, I initially I had thought of one idea to collect specimen of the plants in a large area, for example, the sidewalks or multiple trees and parks. But then I realized that some of these invasive species easily propagate on their own, which means they would have grown into the sidewalks without anyone introducing them. So I chose this park on purpose because I know some people who have planted these seeds on purpose. So most of the
My question was different actually. You said you have used convenience sampling. That is, whatever is available near you, you plucked and identified and classified, and you saw whether it is native or it is from outside, it is introduced or whatever. And the source also you have given that. But instead of that, if you would have done the random sampling, you would have got a better picture of that location. That is all. No problem.
Nice presentation, good methodology that you have used. It's close to me because I am from that area, Vijayanagar, Nagar. But there are a couple of parks you could extend to even more random sampling to other parks close to Vijayanagar bus stop near the post office. There is a good habitat there as well. Convenience sampling, as ma'am said, random sampling probably you can choose even more better by giving the area, because easily it will be available in Google Map, right? Google Map, the area can be scanned easily. So picking and choosing 2-3 spots and notifying that would be good. That is one thing. My suggestion. So you're talking about the this particular species, all these listed out. So how did you do the listing out from the Wikipedia and other sources? Any other major, the book, or let's say for example?
yes.
So you got all the information from that?
No, not all the information.
Hmm.
Actually, my father's friend works in a forest department, so I took help.
So you took the help of them. Okay.
For the invasive species,
better to mention them also, because you have to cite reference. Yeah? So nice presentation.
Yeah, a nice presentation. Did you observe a rate of spreading of these species? Because you are close to the park and you have access to that, and whether you how long it will take to spread, whether you studied about that?
I didn't study how long it would spread because most of the plants there were planted intentionally.
Okay.
You do this on your phone. That you give it to him.
Nine.
Holding the controller. Okay.
Time. Yeah, we can start now.
A very good morning to one and all present here. I am Aradhya, a student of grade 9, presenting my part of Lake Symposium 2024. Water bodies, as we all know, are of many uses to us, like they help check floods by absorbing excess rainfall. They form a base of the food web, and they also store water in a large amount, which is later contributed in the agricultural terms. As we all might have been knowing that these water bodies are declining rapidly in count and quality. So I have chosen a topic through which I have stated the condition of a lake, which plays a major role in Bangalore's economy and agriculture. Thus, my topic is limnology of Vattur Lake and its impact. This lake covers an area of 445.8 acres, and it is really surprising to know that 40% of Bangalore sewage is disposed in this lake all alone. This causes eutrophication. This is my study area. The location is as given. Average depth is 1.05 meters, macrophyte is 70 to 80%, and the elevation of the area surrounding it is as given. Now let's get to know a little more about this lake. This lake is located in the south taluk of Bangalore district in Karnataka. This is an artificial tank of an estimated area of 1,478,000 meters square. This was built 1,000 years ago during the Ganga Empire and continues to be still in the process of using. The total land irrigated is 1,537 acres, and crops like paddy, areca nuts, bananas, etc. are grown all using this water. My objectives through this project are to understand the physico-chemical properties of water as they are home to a number of aquatic lives, to know about the different pollutants polluting the water, to spread awareness about giving a better habitat to the migratory birds, and to know the rate of pollution and consequences of those which we already have been facing lately. Frothing of lake. As residents of Bangalore, I'm sure that most of us might have encountered at least once. Frothing of lake, Benandur is a major example of this. And according to research, it is estimated that Vattur Lake will be facing the same condition in few more years if not maintained. This is caused due to eutrophication, which results in depleted oxygen level and lathering. Macrophytes. These are plants that adapt to the aquatic environment. They are mainly of three types: emergent, submergent, and floating. Some of the commonly occurring freshwater macrophytes are Azolla pinnata, Azolla microphylla, Hydrilla, etc. These are the specific macrophytes present in the lake, which are Eichhornia crassipes, Alpha latifolia, Hydrilla verticillata, and Wolffia. Now let's come to the topography. This is the comparative topography of the lake taken through a satellite in the year 2002 and 2024 respectively. We can observe many changes, but the main ones which I observe here are that the lake has encroached the surrounding areas, and urbanization has grown a little more, which has affected the greenery as well. The major inlet is a channel from Bellandur Lake. There are many inlets and outlets, but these are the major ones. And the major outlet is Vattur North. There are two major outlets: Vattur North outlet and Vattur South outlet, which directly drain in the Dakshina Pinakere River later on, flowing in Tamil Nadu. ISQA, Simple Water Quality Index. This represents the water quality of the water body. The desirable limit is 0 to 100, but we can notice that the ISQA of Vattur Lake has deteriorated to 135.5, which is of course a lot. To some extent, water quality is also determined by the air quality, as the particles may fall onto the surface. So I have taken the estimated average AQI of the air surrounding the lake area and the ISQA from 2002 and 2024 and have compared it. We can see that in 2002 it is good, but it has deteriorated to moderate and hazardous in 2024, which shows that it has polluted extremely. This is the comparison using the chart. The blue bar represents 2002 and red represents 2024. These are the microorganisms. There were many found, but E. coli was found in large colonies, and plankton was found in large numbers. This is the picture of corrected microorganisms. I have collected samples from inlet, outlet, and shoreline. We must ensure our safety first by using gloves and masks and carefully collecting the water. Now let's check out the results of the water analysis. So we can see that phosphorus, nitrate and ammonia are not at all present in this water. They are being treated. Recently they are being maintained. And the turbidity has been deteriorated to 25 NTU and the alkalinity is 250 and 260 respectively. Sample 1 and sample 2. In sample 1 and sample 2. These are the species identified. Birds. There are around 150 species of birds which visit this lake annually, and they fly over 13,000 kilometers non-stop. The most spotted apes are Eurasian spoonbill, godwit, northern shoveler, northern pintail, and black-winged stilt. Microorganisms. Eutrophication has given rise to algae, Iconia craspus, which is also called as water hyacinth, bacteria, E. coli, and there are many other viruses and pathogens found in this water. Fish. We earlier had around 30 fish species in this water when the lake was once pristine, but now unfortunately we are only left with 4 to 5 species, which are tilapia, katla, rohu and miggle. This was an incident that took place in Batur Lake on October 17, 2023. So there were 25,000 fish found dead on the surface of water, and after there was some investigations, they got to know that the fish had suffocated due to depleted oxygen level in the water, which was caused due to untreated sewage entry. So this was one of the examples how it is deteriorating the water. If we try, we can also maintain the water in a better level. It has affected environment in various ways, like it has created imbalance in the ecosystem, and the visits of migratory birds are decreasing day by day because the prey is no more available in a good amount. Let's now conclude. We must neutralize and treat the sewage before disposing it in the water body. Neutralizing helps it come near to the pH of 7, so that it neither has acidic or basic properties left in it which affect the marine life. We must avoid agricultural runoff, nitrate disposition and leaching from septic tanks. Phosphate-free detergents must be used by individuals because phosphate helps speed up eutrophication, and lake must be examined in equal intervals of time, as well as lake dredging processes and hydro raking can also take place. I would like to remind us all that it is not only the responsibility of an individual, neither of the government. We all must work together to make the environment better and safer. These are the references. And last but not the least, I would like to thank each and every teacher who has helped me come this way, and I would like to thank each and every IISC official for providing me this opportunity. Thank you.
You said that you were talking about the fish kills which happened at Vathur Lake. So you said that the fish kills occur due to low oxygen. But then due to the pollution, the oxygen must be like low for a very long duration of time. And so for the fish kill happening on that specific day, is there any like special reason which occurred only on that day due to which fish died in such large numbers?
Yeah, so it definitely happens. Like, we definitely keep polluting the water, but at some point of time, and fish also keep on suffocating, but as they die in a less amount, we don't notice it that much. But at a point of time when most of the fish cannot tolerate it anymore, these kind of massive incidents occur.
Any more questions?
That was a nice presentation, but follow-up question of what he said. Is it like suddenly you have a tipping point, or like for the fish to die?
No, sir, it's not actually at one point of time that they all die at once, but the pollution keeps on increasing day by day.
Okay.
So at one time, there's a massive
So
issue.
you have done some conductivity studies and all. If you go back, I saw a table. I'm a physicist, so I will be interested in that. So there are three sampling, right? That is the limit and sample one, sample. Where did you do your conductivity study?
Sorry, sir?
Where did you do your conductivity study, or you got the information?
No, sir, I had got the information from online sites.
What does that convert for you? What do you infer out of this? If you see the conductivity, normally it should be 500 millisieverts per centimeter. So there it is increasing. What you can compare out of it?
Yes, so when there is more
Conductivity.
dissolved salts in it, the conductivity increases. Conductivity is like um, how much can the energy pass through it?
What energy?
Electrical, it means current.
Okay.
Yeah. So it shows that how much can the total dissolved salts. The more amount of dissolved salts, the more amount of conductivity results in it.
Thank you.
Thank you.
Thank you. Thank you.
Thirty-one. Yeah, you can start.
Good morning everyone. Myself Amina Kausa. I am from 9th standard.
Good morning everyone. My name is Saida Manzara. We are from 9th standard. We are from Mangalore Based Girls High School.
And our project is paper waste management in school. We all know that paper is the most essential item in both education and official industries, but we are least concerned about the paper factories which produce toxic gases leading to global warming and acid rain. And as per the Indian Arrow and Recycled Paper Mines, India consumes around 13 million tons of paper and only 25 to 28% of it is recovered by recycling. So we should assume that major share of paper ends up in landfill where microorganisms convert this paper into methane gas, which is again a greenhouse gas. Our study is to see how effectively we can save paper. Our objectives are to continue our previous study about paper waste management in more fruitful way and to spread awareness among our friends about paper waste, to see possibilities of using incinerators at school and using paper ash as fertilizers and weedicides. This is a false slide. It was mistake. Before going for awareness campaign, we did a record to see how much paper waste is produced per class from 1st standard to 10th standard. We observed that higher classes like 9th and 10th was having more paper waste compared to lower classes, and among lower classes only 5th standard was having more paper waste. Compared to previous years, the paper produced this 2 years was very less because our effective ways to save paper. After recording the paper for 1 week, we went for awareness campaign, and these are the pictures of that. We made our friends know that approximately 1/3 of a tree is used to produce standard 200 pages of notebook. If we use this book very judicially and wisely, we could save so many paper. But we also know that torn, worn and spoiled pages cannot be recycled and they end up in landfill. And they say that among in landfill, 26% of 26% of landfill consists of paper waste, and knowingly and unknowingly we are contributing for garbage accumulation also. If we are a responsible student, it's our duty to save paper waste at any measure. We did a comparative studies of number of notebooks and question papers given at school, and this is the chart which shows the comparison. Previous year we used to get 21 books per student, and on an average 861 trees were killed that particular year. And next year our school management reduced the number of notebooks to 15%, 15 notebooks per student, and on an average 246 of trees were saved from being cut. Next comparative study is about question papers and answer sheet. We all know that for our test and exams we are given a separate question paper and a separate answer sheet, and on an average if a student uses 114 sheet for his exam annually, 157.5 kg of paper ends up in landfill. So our school came up with another alternative, that is giving the answer space in question paper itself, where a student was using 114 sheet for his annual exams. Here he saved it. It came down for 48 pages per annum for a student. This comparative study is both economically and eco-friendly, and these are the results of the campaign. Our next study is about paper-based incinerator. When we saw a video of JM3J domestic combustion equipment, which was an incinerator, we discussed with our school management and placed order, and we got the material after 1.5 months. We bought a 2-foot incinerator which was made by galvanization and stainless steel. These are the procedure to use the incinerator. We observed that the incinerator produced very less smoke or no smoke, and the combustion was very quick and easy. The ash was collected in a separate tray which was given with the incinerator. As our IISC took the care of the daily paper-based generation, we did a demo for them, and these are the pictures. Earlier, we used to store the paper waste in the big drum and give it to BBM pick power chemicals once in a week. However, after the incinerator came, the disposal of paper was very easy. On an average, we used to get 272 grams of paper, and from that, it was turned completely into ash by the incinerator. We wanted to check that if we can use this ash as a fertilizer and a weedicide, so we choose 2 plants to see these effects. One we mix the paper ash with water and sprinkle it on the plant for once in 3 days, and for weedicide we directly applied the ash on the roots of the plant and the leaves. So we observed that the plant which we used for seeing the weedicide effect was having 2 or 3 leaves before, but after 2 or 3 attempts, the we saw that the leaves were shed and the base was very weak, and the plant which we chose for seeing the fertilizing effect, it showed very slow growth, but it was steady. The paper was generated from the school can be turned into ashes by the help of incinerator. Also, on average, we can save 59.4 kg of paper from going to landfills. There was no smoke or little smoke during the combustion of paper. Our experiment with paper ash, when we
water in that that indeed shows promote growth. We notice that direct application of ash can destroy the weeds. All the management, school management should take a decision not to impose lots of number of books. It is a national waste. Giving sufficient place for answer in question paper is more economic and it saves lots of paper. Waste donation is on individual schools and strict measures should be implemented to manage the waste by the source itself. Useful inventions like incinerators should be supported and encouraged by the public considering it as long-term benefit to environment. The ash from the incinerators, if used in judicious way, can be turned into fertilizer for gardening. But still, we need to do further studies of chemical effects of ash on insect feeding, nectar feeding insects. These are our references. We sincerely thank Dr. TVRAMACHANDRA and CES IISC for providing us an opportunity to present our project. We thank our school management for buying us an incinerator. We thank our classmates and our teachers for cooperating with us. We thank our school ayahs and security for assisting us to collect the paper for compassion. We also extend our gratitude towards our parents for allowing us to participate in Lake Symposium. Thank you.
Any questions?
Any cost-effective way to dispose these papers?
Disposing this paper like ash is a very good idea for disposing. If we have checked the decomposing of ash also, but it will take so long time and it is not it is not thing to do in school. It will produce more stinks and all. So decomposing of paper ash is not good at school.
No, everyone won't be able to afford incinerator, right? So anything cost effective?
No, buying the incinerator, it's not like that. We can have it.
How did you from where did you buy it and what's the cost of it?
Cost, we really don't know the cost, but we saw the video of, I mentioned that JM3J, domestic conversion. We saw the video and we discussed with our school management.
Oh.
The 59.4 kg, that is of 6 month time or what?
No, it's after excluding our Dasera holidays, summer holidays and all, we just got on an average, we can, it's that number of days. Like we don't come for our holidays for our school and produce paper waste.
Yeah.
So we excluded that days and on an average we got 144 days.
Total overall.
Go back to the conclusion one step before. Yeah. Fourth point, our experiments with paper ash showed that it indeed promotes growth. What is the meaning of this? Can you tell about it when mixed with water? What is the meaning of it? Your interpretation.
Because paper ash, if we use in large quantity, it can destroy the plant. So we mixed it with the water to dilute the paper waste, so that it can promote, it don't harm the plant, but it shows the growth effect.
Growth of plants, you mean?
Yeah, growth of plant.
Incinerator, does it cause any problem to the environment? No.
We said it produced no smoke or less smoke, but smoke was white smoke, which is not that much harmful for environment, and it has lots of space for oxygen to pass through it. So when there is more oxygen, there is less smoke produced.
Compared to the amount of plastic and paper, what do you feel? What is the segregation you feel? I normally feel plastic is more than paper in the class. Chocolate wrappers.
But our study is about paper.
True, but how do you go about? See, naturally, it is not just paper that ends up in the classroom. So, what do you do with that then?
Plastic, yeah, it is more in classrooms, but we are focusing on paper which come from trees.
Don't you feel plastic is the major problem compared to paper?
Paper, I think so, paper, because we cut so many paper trees for producing a paper. So, destroying
At the
trees.
end of the day, paper can be degraded, but the plastic cannot be.
We need to do studies for that.
They have taken up with this part of it,
Yes.
so they don't know beyond the paper related.
But when you see waste, you have to think as a whole rather than just one part of it. Just my suggestion.
Next project you make one plastic survey.
You off it that one. You can off. Then they will leave no that time. Third. 3. On again. Point on. I on.
Somehow it is like, there is no problem.
Then we go to press again. It will on.
Once you press it.
Oxygen break now. He will talk about the insects.
Animals.
Hello.
Hello. Animal.
Over to you all. Thank you everyone. Okay. Let's come.
Yeah. You can start now.
Good morning, one and all present here. Myself, Mahalakshmi, yes, with my teammates Kusuma, Bia and Kavya. We thank one and all present here for giving this opportunity to present our study, Water Quality Analysis of Bilwatadi Lake.
Water quality is a crucial factor in maintaining the health of aquatic ecosystem, including wetlands, which are vital for human well-being. Water is an essential component for the survival of life on Earth, playing a vital role for human beings, plants, and aquatic life. The water from Bilwadali Lake serves both direct and indirect uses, such as providing drinking water, irrigation for agriculture, and supporting local biodiversity. The lake and its surrounding wetlands supports recreational activities, cultural practices, and contribute to the local economy. These ecosystem services are critical for sustaining human well-being.
Our project aims are to analyze physical and chemical biologic parameters of Bilwadali lake water to create awareness to protect the lake. Our study area is Bilwadali Lake, Banagatta Panchayat, Jigni, Hubli, Anekal, Bengaluru South. This is our map. These are geographical coordinates of our study site. Materials used for water quality analysis are TDS meter, EC meter, pH meter, BOD bottle 125 ml, measuring cylinder, conical flask, pipette, burette, and chemical as prescribed in ENVIS Technical Report 77. Various containers, labels, collected water samples from Bilwadali Lake, and microscope, plantanet, smartphone, pen, and notebook.
And methods of water quality analysis are collected water samples from Bilwadali Lake using clean containers, labeled samples using unique identifiers including source, date, and time of collection. Using calibrated TDS meter, we submerged the probe into water sample. We waited for the reading to stabilize and recorded TDS values in milligram per liter. Using calibrated EC meter, we submerged the probe into water sample. We waited for the reading to stabilize and recorded EC values in microsiemens per centimeter. Using calibrated pH meter, we submerged the electrode into water sample. We waited for the reading to stabilize and recorded pH values. For measurement of DO and BOD, we used Winkler's method and recorded all measurements and observations in a notebook including sample identifiers and test results. And this is our physical and chemical parameters at different sampling sites. In inlet 1, pH is 8.7, DO is 7.31 milligram per liter, BOD is 16.25 milligram per liter, temperature is 26.7 degrees Celsius, EC is 0.77 microsiemens per centimeter, TDS is 364 milligram per liter, and so on.
This is our photo gallery. These are identified plantains.
The temperature across the three sites range from 26.7 degrees Celsius to 28 degrees Celsius. These values are typical for surface water bodies in the region and suggest stable thermal conditions. However, slight temperature can affect the solubility of oxygen and metabolic reactions for aquatic organisms. The EC values range from 0.69 microsiemens per centimeter to 0.77 microsiemens per centimeter across the sites. These values suggest that the water has dissolved ions in the water. The TDS values range from 340 mg per liter to 364 mg per liter, which are within the acceptable limits for surface water bodies. These TDS values suggest that the water is not heavily polluted. The pH values range from 7.6 to 8.7, indicating that the water is lightly alkaline. These values are typical for freshwater ecosystems and is conducive for aquatic life.
DO levels range from 4.06 milligram per liter to 7.31 milligram per liter. These levels are crucial for the survival of aquatic organisms. Outlet 1 showed the lower DO level, which is near the lowest limit of which is generally considered as suitable for aquatic life. These lower DO levels could be due to higher organic matter decomposing or lower photosynthetic activities. BOD values range from 12.19 mg per liter to 24.4 mg per liter, with highest at outlet 1. High BOD values suggest higher amount of organic matter in water, which uses oxygen during decomposition. These BOD values indicate potential organic pollution, possibly from agricultural runoff or sewage inputs.
The presence of plankton species such as Stigioclonium, Staronis, Agnathalum and Chattophora were observed. These species were common in freshwater bodies and can provide insights to trophic status of the lake. The presence of Stigioclonium and Chattophora, which are filamentous algae, may indicate the nutrient-rich conditions. This is our awareness gallery.
Bivardhani Lake generally has good water quality with stable physical condition and mostly healthy chemical parameters. However, there is some concern at site 2 where lower oxygen levels and higher BOD levels are observed, such as localized organic pollution. This area should be monitored closely to prevent potential problems. The lake's diverse plankton population indicates that the lake supports balanced ecosystem. Continued monitoring and careful management will help in maintain this balance and ensure that the lake remains healthy for both the environment and the surrounding communities. Regular water quality checks are important to catch any early signs of pollution and take action to protect the lake.
We thank our HM and guide teacher for allowing us to conduct the study. We also thank Sri Vijulal sir and Sri Chaturved sir for guiding us. Our gratitude extends for the scientists, field researchers and our EWRG, CES, IISC friends Bangalore. These are our references. Thank you.
Any questions? You said that the dissolved oxygen was lower at outlet 1, right? So why is the dissolved oxygen lower at only outlet 1? Is there like specific reason why there's something specific happening at outlet 1?
There is one tree near outlet 1 and it doesn't allow the light to pass into the water, and there is no photosynthetic activity in that place. So this could lead to lower DO level in that area.
Okay, thank you.
Any industries in and around this lake?
No, ma'am.
Any effluents flowing into this?
Yeah, there are farms there. So when rain comes, the water goes into the lake.
Compared to Vartur Lake, Ilvardali Lake, is it more polluted or not?
No, sir.
So do you think of comparing the data in future? You want to do that kind of work?
Yeah, we'll try to do.
So why it is not polluted? Can you tell me? Just by observing the habitat surrounding, can you tell me?
There is no houses around there and nobody will go there. Just there are farms and around Bivardhani Lake.
Okay.
So no more people will go there and nothing is going inside the lake. Just runoff from fields are going into the lake.
Human interference is less.
Okay, thank you, thank you.
can you please give that some that hard disk or something to them, because I can keep that entire folder. Huh? Okay, then they can do that like that. Yeah, because it's our also. Okay, I told them to. Sorry, that is poster that they are asking about. Serial number 15. Ananya Reddy and team.
Good morning to one and all present here. I am Ananya Reddy Mule of Standard 9.
And I am TVRAMACHANDRA of Standard 9. We are from Vagdevi Villa School, Whitefield.
And our research is on birds of Bamanahali Lake. Lakes are a dynamic inland aquatic system and they support and provide a balanced community of organisms. Birds are a very essential part of this community as they are essential for factors like pest control, pollination and for being a part of the food chain. We should help preserve these species.
So the objective of our project is to identify and analyze about the birds whose habitat is the Bomanali Lake, as well as to analyze their existing status and to determine the threats affecting the lake.
The methodology used includes the usage of binoculars, a Samsung cell phone camera, a notebook and a pen. The methods of visual and sound-based identification were followed. The identification criteria is the identification of morphological features of the birds. To confirm visual identification, use Merlin Bird ID. As you can see, the study was carried out from 8th July to 26th of September, and the time spent on each visit was 1 to 1.5 hours, and all lake visits were made from 6:30 to 8 am in the morning.
So the study area of our project is the Bomanali Lake, and here is the mapping of the lake. We have also mentioned the size of the lake and many other geographical details regarding the lake.
Here is a logbook of the birds observed. As you can see, on day 1, we observed 9 birds, on day 2, 10 birds, on day 3, 11 birds, and on day 4, 13 birds. It is to be noted that these days are non-consecutive.
So the following here are the names of the birds, the names of the species of birds we have observed, such as the white-throat wagtail, common myna, common coot, along with the scientific name, the family they follow, the family they belong to, their IUCN status, the food diet they follow, as well as their activity during observation. As we can see here, all the birds observed here fall under the least concern of the IUCN red list.
Here are a few photos we captured during our observation. It is to be noted that the Asian quail and redwater lapwing were identified using their sound.
So here's a histogram based on the number of birds we've observed, and such as common coot. As we can see, we've observed around 13 to 14 number of common coots on almost each day of visit.
Here is a classification of birds based on their food diet. As you can see, we've divided them into 5 categories. That is the insectivorous, omnivorous, carnivorous, piscivorous, and frugivorous. As you can see, that the insectivorous and omnivorous are kind of dominant in this region. They stand at 23% each, while we found 4 species in both these categories, and the rest of the categories have 3 species each.
So we've also studied about the nesting habits of the birds, and hence we've classified them as solitary nesters and colony nesters. As we can see, there are 24% of the birds here are colony nesters, whereas 76% of birds here are solitary nesters. And hence we can say that this area is majorly surrounded by territorial birds.
Here is the observation based on plumage, which is divided into vibrant and a dull plumage. As you can see, we have observed 6 out of 17 birds, which is 35% under the category of vibrant plumage. The characteristics of these birds are that these birds, their vibrant colors are used to attract mates as they stand out as breeding displays. They are mostly insectivorous or frugivorous by diet. This is quite in contrast to birds which have a dull plumage. As you can see, they are opportunistic feeders and their muted tones help them to camouflage into their surroundings. The birds with a dull plumage are of 65% dominance in this region. Here is the conclusion over the previous slide. As you can see, number 1 is camouflage. Bomnahali Lake is dominated by birds with a dull plumage, which is 65%, and the reasons are camouflage. Number 1, this proves there are many predators in the surroundings, and number 2, which is metabolic efficiency or energy conservation. The production of colorful feathers is a result of carotenoids. So when they are not producing carotenoids, they are saving energy.
So here we have mentioned the names of the common birds, as in the birds we have observed on all days, and here we mentioned the names of few birds which we have only observed in specific dates along with their dates.
Here are the threats to the lake that directly affect the lake. As you can see, sewage is being discharged into the lake. This is highly risky for the birds as the sewage that is being discharged is contaminating the water body.
So as we can see here, the sewage discharge along with impurities have caused foaming, which also stands as a threat.
Improper waste disposal such as plastic and other contaminants is contaminating the lake and its ecosystem.
So as recommendations, we can say that we should increase the awareness among many schools in order to increase the number of fruit-bearing trees around the lake and to regulate the sewage which is discharged into the lake. And we would also like to suggest that many organizations should take more CSR-oriented initiatives in order to clean the plastic and other dirt inside the lake regularly on a regular basis. And pesticides, the usage of pesticides around the lake should be regulated.
Here are our conclusions. All the birds fall under the least concern of the IUCN Red List. The most observed bird was the common coot. The least observed birds was the grey-headed swampfin, the purple heron, the great cormorant, and the common kingfisher. As you can see, the number of birds has increased as the monsoon has progressed, and birds with a dull plumage are dominant in this region, which is 65%. Insectivorous and omnivorous birds are dominant in this region too. Most of the birds are solitary nesters, which shows that the birds in this region are territorial in nature. Observation shows that birds with a vibrant plumage are predominantly not seen in this area.
So the following are the references we've used for our project.
Thank you.
Any questions?
Any questions? You have proof of letting the sewage directly into the lake.
Yes, ma'am.
Did your school authorities or the students went and informed the local authorities to take any action on that?
Yes, ma'am. Actually, previously, as I've written in the reference, there's a reference bangalore.mirror.indiatimes.com. So previously, there was a corporate social responsibility by Herbalife CSR and was undertaken upon the lake. But then this CSR was dropped midway. So as you can see in this image, I think you can see a few rocks around the sewage. So these rocks have been disposed into the lake to stop the sewage flow and to filter the waste. But the CSR-oriented initiatives were left incomplete. And the organization's name was Herbalife.
Rocks are not a proper way of purifying this. These things doesn't come under any kind of treatment.
The Herbalife had tried to initiate it, but they had dropped the initiative midway.
As just to go back to the number of birds in each day, 8, 9, 10. Yeah. You observed why the number of birds are varying in this 9 and 13, day 4. Any idea?
Yeah,
In your
so these days are non-consecutive and they're from 8 July to 26th of September. So our observation is that as the monsoon has advanced, the number of birds have increased.
Because of that, not because of food habits or anything.
So when the monsoon comes into play, the birds' breeding habits and the food availability also changes. So because of that, number of birds have significantly increased.
Okay, thank you.
Thank you.
You three? Okay, okay. Okay. Go. Huh? Okay, then you can start. You can start. Yes, you can present. Huh? Just a minute. Yeah, yes, I will.
Very good morning to one and all present here. We, the students of Vidyaniketan Public School, being Moha K, Kshitij B.S. and Siddharthi Murthy, are honored to present before you the comparative study on the water quality of Bangalore lakes through chemical analysis. The introduction. Lakes play an important role in maintaining the water cycle, regulating temperatures, and also having ecological balance. However, lakes are also, this was realized by the early planners of Bangalore, and they managed to integrate around 285 lakes by 1970. However, a total of around 100 lakes have just disappeared in just about 40 years, and this is a direct threat to the ecological balance. Next, the drying up of the lakes can adversely impact the ecosystems around the lake. The objectives of a study is to primarily identify the presence of microplastics and assess the impact in the trophic chain. Then to the chemical analysis to assess the water quality index for the human consumption. This is our study areas, one being Ulla Lake situated in Ulla and the other one being Solikere Lake situated in Ramasandra. The perimeter is of the Ulla Lake is approximately 1,569 meter and the area of 0.109 kilometer square against the perimeter of 3,683 meters and area of 0.48 kilometer square. These are the sample collection areas. We have collected 4 samples from each of the lakes. These are the various tests we conducted. The basic parametric test along with the DO, dissolved oxygen test, BOD, biochemical oxygen demand, along with microbe analysis and microplastic analysis. So we're gonna start with the results and discussions. So what we have tried to achieve here is that we have collected, as we have collected 4 samples from each of the lakes, we have averaged all the values to present a suitable value for the overall lake. So these were preserved just after collection to decrease the to manage to preserve it properly. They were cooled immediately. So to have minimal changes between the collection and analysis. I hand it over to Kshitij BS.
It is to be noted that this creation of integrated water sample, this was referred to from a published scientific study, and that is where this idea was taken from.
So
In the slide, we can see a tabulation of all the readings we obtained from the assessment of parameters. The first table is for Ulla Lake. So it displays the values for 4 different samples respective to their mentioned parameters, and it takes a mean average of all the 4 values from all 4 samples, and this will be useful later on. In this slide, we have tried to make a graphical representation of all the values obtained, and as we can see, the average pH for Ulla Lake is 7.4, which is neutral and ideal for the lake, and the average pH for Sulikere Lake is 9.4, which is not ideal. It's slightly basic, and it suggests an unusual amount of alkaline minerals in Sulikere Lake, which can lead to ammonia formation, threatening aquatic life. Then moving on to total hardness. The average total hardness for Ulla Lake is 375, and the average total hardness for Sulikere Lake is 362. Both of these values are above ideal, which suggests that the lake water is a bit harder than what's recommended. And calcium hardness, going to calcium hardness, the average calcium hardness for Ulla Lake is 76.25, and the same for Sulikere Lake is 94. This suggests that it may have calcium rich geology, or that calcium rich substances may have been introduced into the lake somehow. Dissolved oxygen, so average dissolved oxygen for Ulla Lake is 9.3, and average dissolved oxygen for Sulikere Lake is 5.88, which are all ideal according to standards. Biochemical oxygen demand is 3.89 for Ulla Lake and 2.72 for Sulikere Lake. These are in mg per liter, and they are also ideal. Chlorides are salts which are naturally in lakes and rivers, freshwater bodies mainly, and they are essential for the functioning of regular cell function of aquatic organisms. But excessive chloride may lead to salination of a lake, and we can observe here that Ulla Lake has 305 parts per million, and Sulikere Lake has 290 parts per million. Moving on to residual chlorine. Residual chlorine is essential in preventing microbial contamination of lakes. And we can see that Ulla Lake has a 0.3 milligrams per liter of residual chlorine in the water, and Sulikere Lake has about the same, and these are all within limits. Fluoride, moving on to fluoride. Fluoride is essential, as fluoride in drinking water can reduce the risk of fluorosis and maintain dental health. However, excessive fluoride is also adverse, and the absence of fluoride can also be seen as an adverse phenomenon. So, the Ulla Lake has 0 milligrams per liter of fluoride, which is deficient. And okay, moving on to the total dissolved solids, we observe that both lakes have a high TDS value, which is not ideal. I call upon Siddharth to continue.
Moving further, this slide represents the biological microbes we found. So in Ulla Lake, we observed that there were very few to almost no biological organisms found, which also correlates to the lower DO we have found. And Sulikere Lake also, there were lower amount of microbes than ideal. Okay, so let's start off with an analysis of microplastics. So what are microplastics? Microplastics are parts of plastic that are smaller than 5 millimeter in length. So there are multiple ways to identify microplastics in lake water, including Raman spectroscopy and Nile staining. So this technique that we have used here involves a dye called Sudan 3. So we have identified several pockets of microplastics in both lakes, with the higher number of pockets being in Ulla Lake. And the presence of microplastics implicates the release of sewage into both lakes, which is occurring, yes. And it can move up through the trophic chain and adversely affect humans through organ dysfunction, metabolic disorders, etc. Water quality index. So on this slide, we can see the BIS drinking water standards for BIS drinking water standards. These parameters are essential for determining the portability of water and its suitability for consumption. So we'll be comparing all the values from both lakes using these standards. Last one we can wrap up. Okay. Yeah. So as you can see, a few major phenomena we can observe here is fluoride is almost low to none in Ullad Lake, which which is adverse, and pH is very alkaline in Sulikere Lake, which can cause ammonium condensation, which can affect the aquatic life in those lakes. So using these standards, we can classify that Ullad Lake is more polluted than Sulikere Lake because most of the standards have been breached compared to Sulikere Lake. Moving on to issues and recommendations. So finally, we would like to say that the water spread area of Ullad Lake is reducing despite rainfall, and blocked stormwater drains are further increasing this problem. Both lakes have untreated sewage water being dumped into them, which is further increasing the problem of contaminants and effluents flowing into both lakes, and it is also adversely affecting the ecosystem and the wildlife of both lakes. Conclusions. So we would like to urge the local government, the BBMP, to take action on anthropogenic activities taking place in both lakes, and we would like to carry out environmental rallies, and we would like to raise awareness on social media, and we would like to urge the local government to take action, and we would like to propose a system of better filtration using nanoparticle filters and carbon filters. These are the pictures that were taken when we went for collection of samples. These are our references. Acknowledgement. So we are really thankful to the IISC team for giving us this opportunity to present our presentation here, and we are grateful to our teachers and our school for giving us this opportunity. Thank you.
Any questions? Any questions?
Why all the parameters you kept it in one graph? That parameters has different units, no?
Can I answer this?
Yeah.
Ma'am, so the thing is, ma'am, if you go to the slide, please go to the graph. Ma'am, you can see that on the y-axis there is no generic ma'am unit. Ma'am, so it's only like numerical comparison.
But you can see that the pH and all very narrow. Then the fluoride also, that is 0, that's 0.75 now. That we never use like that in the scientific way. Okay.
Okay. Ma'am, so the idea was basically to integrate all the values in one slide, and so that when we go through slides, when we move to the next slide, so we don't waste time much. Ma'am, and then you can notice that we have specific units for each of the values. So,
ma'am, so basically in this slide, there's no generic unit for the y-axis. So you could present all of the values in a single graph without actually reaching any practical fun.
Because of your answer, we can understand that you didn't understand the importance of the water quality parameters. Okay, each and every parameter has their own role. So we have to represent everyone it in the different way. Because even any parameters you can take it, that every parameters has one of the example, that is all oxygen, that has also that they have the permissible limit is different, and this values also is different. You can understand here that all the things. Yeah, pH has no unit. You will put it here, then understood.
Yes, ma'am.
The concern is, see, one parameter
separately, but we understand the time constraint of making 6 slides. It's okay for your level.
No, you should make the pH, that graph separate, and everything you can put it in the one slide. pH graph, that DO graph, that like, because you have two different lakes that you can represent as a bar graph like, but in the different. pH is different, DO that is together you can put that, okay, but not this way that together, okay.
You invited troubles by putting y-axis. If you would have vanished y-axis, there would have been no questions. Y-axis, because of y-axis only. If you don't have y-axis, then this question would have been not there. It's okay. For your convenience, you have put, right? So next time, I think you can separate. It's fine.
She has 10 years of experience.
Yes.
You know her.
Yes, yes. Thank you.
This is control.
Control.
Control F5.
Refresh.
The review. Now it's not coming here now.
This white is not. You cut this PPT again.
Control function. F5.
Otherwise, the PPT mode is not there. No, not this. This is not. What that sign you remember?
Yeah.
What is PPT?
This is PDF.
Achha, this is PDF.
Yeah.
But it can go in PPT mode now.
How? Open.
In Microsoft it is not open. Achha, okay.
Okay, then I can take another.
No, now you take another one. We will see at last.
I can do here. Timer. Then you can take photo only there. Give timer.
Good morning, everyone. I am Bhavna, a grade 9 student from Oromera International School. I'll be presenting a selective case study on Ulster Lake. As you can see, this is the study area of Ulster Lake. We'll be understanding and evaluating 3 specific problems which Ulster Lake is currently facing. That is, the fish kill, oxygen depletion, and conservative methods and efforts used to resolve them. The terms used in this presentation are hyponoxia. Hyponoxia is when the oxygen levels drop below normal, but there is aquatic life which can still survive. Anoxia is when oxygen levels drop below normal, causing most aquatic life to suffocate to death. Eutrophication is the process of nutrients like nitrogen and phosphorus which can harm the water quality
and also increase the growth of algae, which in turn depletes the oxygen level. An introduction to Alsur Lake. Alsur Lake is a beautiful and an important water body in the city of Bangalore, and it is known for its urban greenery and lush beauty. It spreads over an area of 123 acres, and there are several islands which add to its charm. It is one of the oldest lakes in Bangalore, and it is built in the 17th century by Kempegowda II. The water supply of the lake comes from stormwater drains called nalas, and it is channeled from the eastern part of the city. The fish kill incident. Fish kill is a mass death of fish in a short period of time. So factors affecting fish kill are environmental factors like salinity level, pH level, dissolved oxygen level, and excessive algal growth. Pollutants like including pesticides, nitrogen, phosphorus, sewage, and other harmful substances also can cause the fish kill. Hypoxia and anoxia are also the factors affecting fish kill. When oxygen levels drop below normal, there are diseases which fishes are likely to catch. Few of them are helminth diseases, protozoa infections, fungal infections, and etc. On 26 Jan 2005, there had been a massive fish kill in Alsur Lake. These are the factors affecting fish kill in Alsur Lake. First one is sewage impact. During flood events, the inlet in the north region had experienced an overflow in sewage, and it was originating from a nearby nala. This sewage usually adds a lot of nutrients to the lake, but since it was an overflow of sewage, there had been improper waste management. Dissolved oxygen. Oxygen gets into the water through photosynthesis, and a huge amount of chemicals were also added into the lake while there had been a cleanup in the swimming pool. And there had also been use of phosphorus in the lake, which had harmed the water quality. And fishes need at least 5 milligram per liter of oxygen to survive and have a good health. But in Alsur Lake, there had been less than 1 milligram per liter of oxygen, which had made it difficult for the fishes to survive. So, during like a fish kill, most of the people had noticed an unpleasant odor surrounding the Alsur Lake. So, why does an unpleasant odor occur in Alsur Lake? When fishes die due to low oxygen level, a stinky smell can occur like rotten eggs, and this is due to an acid called hydrogen sulfuric acid. It is a very toxic substance, and it can kill many organisms if it comes in contact with the respiratory system. These are the main problems of the fish kill, which resulted in the fish kill in Alsur Lake. And a few of the solutions I believe which can prevent the future fish kill are: regular water analysis should be done, and aeration helps in the prevention of the fish mortality and enhances the level water quantity. And bioremediation also basically is a process which uses useful microorganisms to clean up contaminated soil. And one of the example is grass carp, which is used to it is known for eating unwanted aquatic vegetation. Oxygen depletion. Oxygen depletion is the decrease in the conservation of dissolved oxygen in
in the water body. And when oxygen levels drop below 2 milligram per liter, it can stress the aquatic life. And when it drops below 1 milligram per liter, it can cause a massive fish kill. Factors affecting oxygen depletion are temperature. Temperature, as the temperature increases, the oxygen level, dissolved oxygen level decreases. Algal bloom and decomposition. So warm water causes more bacteria to grow excessively. And metabolism. Since the water is warm, fishes require more oxygen, and due to which their metabolism increases. The factors which I believe which will improve the levels of oxygen are hypoelementic aeration. Basically, it adds oxygen deep into the layers without disturbing the thermal layers and reducing the nutrients coming from the stormwater drains. And also, we can promote the growth of certain fish species. Conserving the lake. Also, lake has been facing several challenges during the years, and a few of the key problems are improper waste management. There's several fish kills due to low dissolved oxygen, and also due to heavy rains, there is excessive overflow of sewage. And in the recent years, we have seen Alsi Lake with a few conservative efforts. And while I compare the recent and the past and the present methods, I found that there is a shift in the technology. And the old methods like desilting the lake, they used to de-water the lake, they used to displace the silt, and then it used to take a long time. But then the recent methods were Kashi Ball. Basically, it was a short-term measure. And after analyzing the problems, few of the solutions I can convey is natural filtration system, and it can basically filter out nutrients like phosphorus and nitrogen. And phytoremediation, planting water purifying species in the edges of the lake. And these are the references I used. And I would like to thank my school teachers, my friends, my family for helping me. Thank you.
Any questions to be asked? Any questions?
How many times you visited the lake and any primary data collection analysis?
I couldn't take primary data collection due to
this is a review, right?
I tried to take it. I also spoke with the workers, care workers there, caretakers, and I got a lot of information from them.
It's a good presentation. Take care of pronunciation. It's not modality. It's not hydrogen sulfide. It's not hydrosulfuric acid. A couple of things you have to take care. Other than that, it's okay.
Water analysis is regularly done, or any idea whether you got?
Water analysis is done every 3 weeks. It's only done recently and after the fish kill, which happened in 2016. And after that, they've been taking care, and it's a specific group which is taking care, and they only come on Sundays.
Thank you.
Shall I ask you one question? You people are very tensed because last few presentation I understood that you all are heavy. You whatever you people knows, you just come and present. This is not for the mark and all. You are doing the wonderful job. You please give respect to yourself actually, because this type conference you can come and you can
give your opinion to public. This is the wonderful choice, na? You should give presentation properly. We all are friendly with you. Our judges, they are not you don't feel like judges, they are your friends. Okay, we are also friends. With your everyone is friends. Yesterday you went, you visited all the place together only, na? Then why tension? Without tension, you please come and give the presentation. Okay.
Good morning everyone. My name is Vishal.
My name is Vaibhav.
We are from Vagdevi Villa School, Vartur. Today we are going to present comparative study of soil. Soil is the cornerstone of agriculture, provides water, minerals and other essentials for the plant. The health and productivity of the soil depends on the soil quality. With a small test, we can find the fertility of it. With these results, we can add the needed fertilizer to increase the productivity in it.
The objective of our project is to conduct NPK test, pH value test, percolation test and moisture test to check the quality of the soil. These are our study areas.
We have dug 4 to 6 inches below the soil surface, then collected the samples in a Ziploc bag and labeled it. We have done this multiple times. pH test. We have used 2 methods. Method 1, using soil testing kit. Reagents are pH reagent 1, pH reagent 2 and decolorizer. We have taken 10 cc of soil in a beaker, then added 25 ml of pH reagent 1 to it and a pinch of decolorizer to it and stirred it well for 5 minutes. Then we have let it obtain its color, then we have filtered it out. Then we have taken the obtained solution and added pH reagent 2 to it and we have let it obtain its color. Then we have compared it with the pH color chart.
Method 2, using pH paper. Take 50 grams of soil in a beaker and add 50 ml of distilled water to it and keep it for 24 hours and dip the pH paper in the solution and compare it with the pH color chart. These are the results obtained.
Study area 1. Farm soil from this area we had got 7.5, which is very slightly base. So add the government recommended dose of pH fertilizer to it. Lake soil from this area we had got 6.5, which is very slightly acidic. So add the government recommended dose of pH fertilizer to it. Garden soil from this area we had got 4.5, which is acidic. Add lime to decrease the acidity level in the soil and add the government recommended dose of fertilizer to it.
In study area 2, farm soil which is 7.5, very slightly base. So use government recommended dose of fertilizer to it. In lake soil which is 5, which is slightly acidic. So reduce the usage of ammonium sulfate and add the recommended dose of the fertilizer to it. In garden soil, which is 8.5, so use gypsum to bring it near to the neutral level and add government recommended dose of fertilizer to it.
Estimation of nitrogen. Reagents are nitrogen reagent 1, nitrogen reagent 2 and decolorizer. We have taken 5 cc of soil in a beaker, then added nitrogen reagent 1 to it and a pinch of decolorizer to it and stirred it well for 5 minutes. Then we have let it obtain its color, then filtered it out. Then we have taken the obtained solution and added nitrogen reagent 2 to it and stirred it well for 5 minutes. We have let it develop its color and we have compared it to nitrogen color chart.
These are the results obtained.
Study area 1. Lake soil from this area we had got M2, which is medium, 80 to 120 kg per acre. Add the government recommended dose of nitrogen fertilizer to it. Farm soil from this area we had got L2, which is 25 to 49 kg per acre. Add 25% more than the government recommended dose of fertilizer to it. Garden soil from this area we had got M1, which is medium, 50 to 80 kg per acre. Add the government recommended dose of nitrogen fertilizer to it.
In study area 2, lake soil, which is L1 low, it contains 25 to 49 kg per acre. So add 25% more than the government recommended dose of nitrogen fertilizer. In farm soil, which is H1 high, it contains 121 to 150 kg per acre. So add 25% less than the government recommended dose of fertilizer to it. In garden soil, which is H2 high, it contains 150 kg per acre. So add 25% less than the government recommended dose of fertilizer to it.
Estimation of phosphorus. Reagents are different but the method is same. Reagents are phosphorus reagent 1, phosphorus reagent 2 and decolorizer.
These are the results obtained.
Study area 1. Lake soil from this area we had got M1, which is medium. Add the government recommended dose of fertilizer to it. Farm soil from this area we had got L2, which is low. Add 25% more than the government recommended dose of fertilizer to it. Garden soil from this area we had got L1, which is low. Add 25% more than the government recommended dose of fertilizer to it.
In study area 2, lake soil, which is L1 low, so add 25% more than the government recommended dose of fertilizer to it. In farm soil, which is H2 high, so add 25% less than the government recommended dose of fertilizer to it. In garden soil, which is H1 high, so add 25% less than the government recommended dose of fertilizer to it.
Estimation of potassium. Reagents are different but the method is same. Reagents are potassium reagent 1, potassium reagent 2 and decolorizer.
These are the results obtained.
Study area 1. Lake soil from this area we have got L2, which is low. Add 25% more than the government recommended dose of fertilizer to it. Farm soil from this area we have got M2, which is medium. Add the government recommended dose of fertilizer to it. Garden soil from this area we have got M1, which is medium. Add the government recommended dose of fertilizer to it.
In study area 2, lake soil, which is M1 medium, so add the government recommended dose of fertilizer to it. In farm soil, which is H2 high, add 25% less than the government recommended dose of fertilizer to it. In garden soil, which is H1 high, so add 25% less than the government recommended dose of fertilizer to it.
Estimate. Take 50 gram of soil and place it in a funnel with filter paper, then pour 50 ml of water to it and let the water percolate and measure how much time it has taken to percolate and how much water has percolated.
These are the results obtained.
Study area 1. Lake soil from this area has the percolation rate is 1.7 ml per minute, whereas in study area 2 it's 1.37 ml per minute. Farm soil from study area 1 it's 3 ml per minute, whereas in study area 2 it's 2.38 ml per minute. Garden soil has taken 1.4 ml per minute, whereas in study area 2 it has taken 1.14 ml per minute.
So the graphical representation of percolation test. Moisture test. Collect the soil sample from all the study areas and wait before dry, and let the soil dry for 24 hours and dry it and check the difference of all the soil samples. These are the results obtained. I conclude that the data obtained from the soil analysis can be used to guide appropriate fertilization and crop selection and soil management practices, and ultimately contributing to more sustainable, efficient land use and providing a foundation for future research.
These are our reference links. I thank IISC for providing us such a good opportunity to present our research.
And I also thank Vagdevi Vilas Institute to give this good opportunity.
I also thank R&D department for their guidance and support. Thank you.
Any questions?
Good morning. As you told the pH test, the nitrogen test, decolorizing. What kind of decolorizing you are using and why it is used? And one more thing, for the sample you have added distilled water. Why not the tap water?
Distilled water gives correct, accurate value.
It is free from salts and minerals. So, then what about the decolorizing agent? What kind of decolorizing?
They didn't give in the soil analysis kit. Just they may mention the reagent.
You have done the practically, right?
Yes.
Then what kind of decolorizing?
They didn't give in that soil analysis kit.
Fine, thank you.
So it was mentioned in the presentation on multiple occasions that government recommended dosage of fertilizer. So could you please state what that government recommended dosage of fertilizer is? And it was also mentioned 25% more or 25% less. So could you please state how you calculated that formula? That finding.
Different type of crops have different amount of fertilizers to be used. We can find the amount of fertilizer to be used in soil health government. In this website we can find out how much fertilizers we should put for the specific crop and how much we have to put extra for it.
For every, I'm not saying crop specific. It will mention government recommended dose of fertilizer. So could you please state that one standard value, if you could? Thank you.
I think he answered your question. For different crops, different.
What he means that there are a lot of crops, lot of things in the chart. So you have to take 25%, and there is already mentioned, so less or more depends on you. So you already done it. But take care, and I think it's a good suggestion from him. Probably you can mention on an average. Give him 2-3 crops, and yeah, that might answer, right? Yes. Thank you. It's a good presentation.
Second one. uh, before starting all their presentation, you should uh, stand the not stand the one people's back, okay? Because we are taking the video, no? Then everyone's face want to see. So you should just spread it and spread. Yeah. Yes, yes. Yeah, like that, okay? Because everyone's face should come, no, that is the thing.
Good morning to one and all present here. Myself Likana. We are from Vidyanjali Primary and High School. I thank one and all present here for giving me an opportunity to present my study, Butterflies of Shiva Temple Hill, a detailed documentation. Butterflies are the insects in the order Leptoptera, which also includes both moths and butterflies. Butterflies are the essential indicators of biodiversity and environmental health. Often they are also used as bioindicators because their presence and diversity reflect the health of the ecosystem they inhabit. Their life cycle and interaction with the environment make them particularly sensitive in the changes like habitat quality, climate, and human activities. Our primary objectives of this project are identify and document the various butterfly species present in study area, analyze the distribution and abundance of these species in different zones of study area, assess the habitat preference of these species.
This is our study area. Shiva Temple Hills, Rivers Colony, Gottigere, Bengaluru South. These are GPS coordinates of our study area. This is our map. The materials that we use in our projects are butterfly identification guide, notebook and pen, smartphone for photograph and GPS, data sheets for recording data. This study was conducted over several weeks. The study area was visited weekly, specifically every Saturday 2 p.m. to 4 p.m. and every Sunday 8 a.m. to 10 a.m. The study site was divided into distinct zones based on the habitat type such as rocky area and garden area. Butterflies were photographed for the documentation and random sampling method were used in the study field. During the study period, the temperature was moderately hot with no humidity. List of butterfly species that we documented. Blue tiger, common crow. Checklist of butterfly species and abundance in different zones.
The study at Shiva Temple documented a total 38 species of butterflies. The documented species span across various families including yellow and white, swallowtail, brush-footed, blues, skippers, indicating healthy and varied ecosystem. In rocky area, we identified total 15 different species. The common grass yellow was the most abundant species. This high abundance can be attributed to the species adaptability of open sunlight area and minimal vegetation. Other notable species like common crow with 19 individuals, pale grass blue with 11 individuals, and 20 coaster and common rose were also seen. In garden area, a total 30 different butterfly species were identified. Common cerulean was the most abundant, and these other notable species like pale grass blue and common grass yellow, both were 15 individuals recorded. The garden area's diverse plant life, including flowering plants, fruiting trees, provides a ample nectar source for various butterfly species. The presence of common gazebo and great orange tip presents the highlights and garden area's role in habitat conservation. The comparison between two zones reveals the distinct pattern in species abundance and distribution. The rocky area is characterized by its open space and minimal vegetation, supports the species that thrive in such conditions like common grass yellow and pale grass blue. The garden area is characterized by its rich vegetation and diverse plant life, provides ample nectar source and host plants for various butterfly species. Next. Each habitat plays a unique role in sustaining butterfly population. Empathizes that the conservation efforts should not only focus on butterflies but also on their host plants.
These are the some features of butterfly that we have documented. Common grass yellow, common gull, dark blue tiger, lemon pansy, common cerulean, common jay. Zebra blue, tailed eggfly, common merman, red pioneer, common crow.
In semi-urban areas like Weaver's Colony, small garden patches like garden, parks and even balconies can be a crucial habitat for butterfly species. While talking to youngsters of Weaver's Colony, we mentioned that pizza ingredients like tomato, basil and relay or butterfly pollination. This shows that how food is connected to our nature. Enhance local agriculture. Butterflies are pollinators of various plants and including vegetables, fruits and herbs. Since they help in local agriculture and home gardening, promotes ecotourism. Butterfly gardens and ecotourism initiatives can lead to the extension of local business like cafes, restaurants and tour operators, education and job opportunities. Rising awareness about butterflies can lead to the educational initiatives, project communities, generating employment and environmental consultancy, organic farming and eco-friendly product sales. The study at Shiva Temple documented a total 38 different species. 15 different species were found in rocky area and 30 different species were found in garden area. The study at Shiva Temple highlights the distribution and abundance of these species in different zones of study area. The Shiva Temple provides a crucial habitat and contributing to urban biodiversity.
We thank our HM and guide teachers for allowing us to conduct this study. We also thank Sri Brijulal sir and Sri Chaturved Shet R for guiding us. Our great gratitude extends to the scientists, field researchers and experts of the EWRG, CES, IISC and Friends Bangalore. This is our reference. Thank you.
Anyone wants to ask question? Okay.
My question is that are butterflies important in the ecosystem? If yes, then what kind of importance do they have?
Yes, of course they are important in ecosystem because they are pollinators of plants. So that's why they are so important in ecosystem.
Thank you.
Suggestion, one correction there. It doesn't pollinate vegetables, fruits. It pollinates fruit yielding plants.
Thank you.
Now you can announce. You can announce tea break.
Students can have 5 minutes break and come. Please come fast.
22. On side that on.
Achha?
Yeah, yes.
Okay. Good morning, judges. Good morning, students. Good morning, audience. My name is Namana Ajay from Presidency School, Bangalore South. My topic is ecotourism and carbon sequestration in the Western Ghats. Now, my area of study would be Kodaikanal in Tamil Nadu, a part of Western Ghats itself. Now, to calculate carbon sequestration, which we'll be doing later on, we'll look at bare shoulder falls. To give a basic understanding of Tamil Nadu, it has the population is 7.21 crores and approximately 50,000 people are in Kodaikanal. Now, our objectives for this project would be to define deforestation because it's a link between carbon sequestration and ecotourism. Then we will look at how to increase the carbon sink in Bengaluru. Now, what is deforestation? Deforestation is the cutting down of trees on a large scale. It's primarily done due to urbanization and the greed of humanity. Now, deforestation has become a big issue in the Western Ghats. Why? Because we've lost 20,000 hectares in the last 17 years. Looking at the graph on the right, we can see in our area of study, there's mostly negative or no change, which is very bad. Now, let's look at the impact of deforestation. So, we've lost 37% of the carbon sequestration ability in the Western Ghats. 12% of that is the part in Tamil Nadu where we are studying. And we have lost $4.5 million in the ecotourism sector, which is 318 million rupees. Next, we have the deforestation rate. We've lost 25%. There's been an increase in 25% of deforestation rate in Western Ghats. That has led to the 3.8 degrees Celsius increase in the annual average temperature. Now, moving to ecotourism. Ecotourism is the sustainable travel which emphasizes on leaving the place how you found it, or even better. Basically, what happens is when tourists come and visit a location, they leave the place just as they saw it. They interact with the locals, they learn their culture, their traditions, the way they do their stuff, the way they do their daily jobs, and this enriches the culture. Next, we move on to carbon sequestration. Carbon sequestration is the ability to hold carbon in the roots of trees, plants, or the above ground biomass. So, the Western Ghats holds 1.23 mgg, which is million gigagrams or GT, in vegetation and soils. And Western Ghats hold $1.4 billion worth of carbon. Now, the process of how carbon sequestration occurs would be in four major parts. It would be first the emission of the carbon, which is done by any activities like burning of fossil fuels, etc. And that will get captured by the nitrifying bacteria or the phytoplankton in the water. Those bacteria will then transfer the carbon dioxide through any waste or the animals itself. And then it's finally stored in those stems or roots of those plants and trees. Next, we will see how we can calculate this carbon sequestration. It's pretty simple. You take above ground biomass, below ground biomass. First, you find the above ground biomass, and you find how much mass is there in the trunk of the tree. You add the 0.5 because you have to add the stems, the leaves, etc. So you get 0.25 into d square into h, diameter into height. So the overall green weight of the biomass is estimated to be 120% of the AGB value. So we know that if 100% is the AGB, and then so BGB will be 20% more of that. So we just add BGB. BGB is equal to 0.2 into AGB. From the above two formulas, we get that the total biomass will be AGB plus BGB, but we would actually need only dry weight. So the dry weight will be 72.5% of the whole tree. So then to find dry weight, we take the total biomass and multiply it by 72.5. Now, what we need is carbon sequestration. So we need to find the amount of carbon in that tree. So we take the total carbon and multiply it by 0.5 because a tree is approximately made up of 0.5. The whole of it is approximately half is 50% of it is approximately carbon. The weight of CO2 it is determined by the ratio of CO2 to C, which is 44 by 12, which is equal to 3.67. Therefore, to determine the weight of the carbon dioxide sequestered in the tree, we multiply the weight of the carbon in the tree by 3.67. Now, I've taken an example for a tree here. Its common name is a Malabar ebony, and the scientific name is Ecotourism malabare. So we have calculated the carbon sequestration in that tree and found that this tree stores 3.725 tons per year. Now we move on to the wild carbon program. The wild carbon program is a solution that is drafted to reduce the carbon dioxide levels in Bangalore. Now it is it's an initiative that aims to increase the carbon sink in Bangalore without trying to disrupt the daily people's life. Now, if you see the graphs, I have marked out possible places where you could plant trees. No, not just any trees. Trees that are high carbon sponges. Trees that are easy to maintain, reliable. They're just simple oaks. Oaks can hold up to 4 tons of carbon in a year. And they're pretty cheap. So you see at the bottom, we have a kusum tree. It's a oak. And it holds like 4.25 tons of carbon a year, and it's costing only 200. Now, where did I get these costs? I called up a bulk supplier called Sealand. Their headquarters is in Pune, and I called them and asked the basic necessities to get those trees to Bengaluru. With the bulk discount and the shipping cost, we can get approximately 7,000 rupees per site. But not all sites can hold trees. Maybe some are infertile, some are used as garbage dumps. So that is a variable in this problem. Next, we can look at how this will be implemented. Any site that is left unused can be used as a carbon sink, which in result will give us less carbon dioxide in the atmosphere of the city, and it will help the people around. Now, moving on to the recommendations if carbon sequestration and ecotourism is applied. We can see that the Western Ghats over the next 100 years will have a rich biodiversity, and it will be an opportunity for people to use their ideas in ecotourism sectors, carbon sequestration sectors, and the carbon levels will be reduced. Now, the weakness, the problem over the next 100 years would be that people never stop hunting. They'll have illegal logging of habitat, there'll be like fragmentation in the wildlife, and there'll be displacement in the whole of it. Now, the opportunity would be the growing ecotourism demand, sustainable development factors, and the threats would be the climate change impacts, land use changes, and policy gaps. Now, what will be a result? The implementation of ecotourism and carbon sequestration will give us nothing but benefits. Now, these benefits can be put into the wild carbon program. It is initiating people to plant more trees. Now, these are references. I thank everyone who gave me an opportunity here today, IISC, the judges, the panel, and the audience. Thank you.
Any questions? Any questions to be asked?
In earlier slides, you mentioned the total carbon stock of the Western Ghats, 1.23 million kilogram. So from where you get this data, or you?
I used IISC research papers for this data. So in the IISC research papers, they have given research regarding how much a tree holds, like how to calculate carbon sequestration, how much it can hold, the whole Western Ghats, how
In
much
that
it will work.
research paper, it is also mentioned how they did, like if there is a forested area, so how you will calculate with carbon, how you will do the sampling?
In a tree, like how much it calculates, like how do you calculate carbon?
Mathematical portions I understood. In tree it is mentioned there which method that was employed there. How you will get the carbon stock, how you will get the biomass.
I think like how do you get those values?
Yeah.
The values. You take the approximation of how a tree is built up. You see that the trunk holds most of the moisture content, with the roots supplying water to it.
I'm just talking about measurements.
Measurement?
Yes.
The height and the diameter?
Yeah, how?
You can just use normal, use measuring tapes.
You said height and?
Height and diameter.
Diameter. At what height you will get diameter? Tree is very long.
We can use the...
One standard thing is there, the value is fixed.
He's asking about carbon. How do you get the value of carbon?
Carbon in the tree. How much carbon it holds in the tree?
How do you do that? Where do you take the measurement?
You take the diameter of the tree.
At what height? Because tree is height is like
yeah. Like
where you
the trunk of the tree. The trunk.
That's so.
Thank you.
Number 42.
Good morning, one and all present here. Myself, Deeti from class 9. These are my team members. Debashish from class 9 and Shalini from class 9 from Sri Sai Vidya Mandira. Today we are going to present a topic on types of soil. So the main objectives of our project is to understand soil composition, identify major soil types, analyze soil properties, to learn soil usage and importance, explore soil formation process, and etc.
Our presentation includes introduction, soil type, their features, experiment carried out by procedures, instruments, recorded data, and reference. Introduction. Coming to the introduction, soil can be thought as loose material that constitute the land in a high proportion. It consists both organic and inorganic matter. It varies with their chemical and physical properties, and they are formed by weathering of rocks and inundation.
So there are majorly 4 types of soil which we have researched in our project. So sandy soil. Sandy soil is very dry in structure, and sandy soil is light, warm, and tends to be acidic and low in nutrients. Sandy soil are often known as light soils due to their high proportionate. When it comes to agriculture, sandy soil cultivate very less amount of vegetables such as herbs like carrot and beetroot. The features of sandy soil are, it contains mainly some particles and very little amount of clay and silt. It lacks humus, very less fertile and no moisture in the soil. So when it comes to silt soil forms by the river water flowing. The structure of silt soil is very slippery when it is wet, and transported by water during floods, the river silt creates a fertile deposit on valley of the floor. The main features of silt soil are: silt is granular material of a size between sand and clay and composed mostly of broken grains. The silt soil is fertile and most common everywhere. So when it comes to loamy soil is in high retentivity. Loamy soil is a type of soil that is a mixture of sand, silt and clay. Loam soil holds nutrients and is texture that retains water long enough for plants and to do it. Loamy soil contains more nutrients in nature. And when it comes to agriculture, loamy soil is best suitable for agriculture. All the types of crops can be grown in loamy soil, such as wheat, jowar, pulses and etc. The features of loamy soil are well draining and fertile, well moisturized, has a crumble structure, supports healthy plant growth. When it comes to clay soil is very sticky in nature and they are found all over the world, including in the most part of US. One of the sure way to recognize the clay soil, if the water sits around a rainwater or sits around after a rain, or if your soil is sticky, you can identify that soil as a clay soil. The main features of clay soil are, it is very sticky and nutrient rich, difficult to work with. And in clay soil, when it comes to agriculture, clay soil we can cultivate mixed farming such as shrubs, herbs, etc.
Coming to the instruments that were used for collecting pH data were filter paper, pH meter, conical flask, funnel, oven, weighing machine, measuring cylinder, beaker, sieve, mortar and pestle. So the procedure that we followed for our experiment, first we collected all the soil sample 4 inches deep from the ground and collected 25 grams of soil sample in 4 individual beakers, then added 50 ml of distilled water, and later by stirring the heterogeneous mixture for 15 seconds, we left the solution undisturbed for 30 minutes and we made sure the suspension broke. Then later, using the pH meter, we recorded the pH data. So the result, loamy soil measured 6.7, clay soil measured 5.2, silt soil measured 6.3 and sand soil measured 5.8.
Our teacher has recommended us that use visual aids to illustrate types of soil and simplify the complex terms, engage the audience with interactive elements. And here I'd like to conclude this. Soil is a rich and natural resource that supports life on earth. We explore the various types of soil, their unique characteristics, and they play a crucial role in agriculture, constructions, and ecosystems. Understanding soil composition, properties, and the factors that influence its formation helps us appreciate its importance in sustainable land management and environmental conservation. By taking steps to conserve managed soil effectively, we can ensure the long-term health of our planet's landscape and ecosystems. Thank you.
Thank you for Alva's institution for giving me this opportunity to present. Thank you. References we took for this project are Chesworth Ward, it's a website, and Encyclopedia of Social Science, it's a book, and Dordrecht, Netherlands Springer, it's also a website. Thank you.
From where did you collect your sample?
Near our school.
You got all the soils?
Yes, 4 types of soil we got.
Okay.
our Karnataka, how many varieties of soil types are there?
Ma'am?
In our Karnataka, how many varieties of soil types are there?
In our Karnataka, majorly 8 types of soils are there, ma'am.
I think the teacher suggested you some recommendations, whether you have incorporated here. The teacher's recommendations, whether you are incorporated in this presentation. Real world examples.
Except 2.
Okay, thank you.
Thank you.
Good morning. I am Malhar Khadilkar from Vagdevi Vilas School, and my presentation is The Birds of Dudnaundi Lake. Introduction to Lake. Dudnaundi Lake is a very large body of water located in the densely populated area of Dudnaundi in the eastern side of Bangalore. It has an area of 98.2 acres and a perimeter of 2.82 kilometers. The lake was in a very bad condition until early 2023, but extensive rejuvenation efforts by the BBMP have drastically improved the water quality of the lake. There are 2 islands present in the lake. They were built artificially in 2014, and they provide excellent nesting habitat for large birds such as herons and pelicans. Objectives. I have 3 main objectives in this presentation. The first, to observe the birds present at Dudnaundi Lake. Second, to analyze and state the factors that make Dudnaundi Lake ideal for the habitation of the observed water birds. And third, to recommend measures to improve the lake ecosystem and make it a better place for birds to live. Materials and methods. The data shown in this report has been collected by visiting the lake 12 times over a period of 6 weeks. I used an app called Merlin Bird ID to identify the birds, as the app can identify them based on sound, images, or even the general information of the bird. Every visit that I made was made early in the morning between the time range of 5:45 AM and 8 AM. I also took a camera with me, and therefore I have images of every bird species that I have documented. For a water analysis that I will be showing later in the report, I collected water from 5 different sites and used the Jaltara water testing kit to test the samples. This is a satellite view of Dudnaundi Lake. As you can see, the lake outline has been outlined in red, while the 2 islands have been outlined in yellow. These are the images of all the bird species that I documented. You can see that the Dudnaundi Lake is host to some relatively rare birds such as the spot-billed pelican, painted stork, and the oriental darter. This is a table of all the bird species that I have documented. I have categorized the birds based on their common name, local name in Kannada, their scientific name, the number of birds that I have seen, the activity as in what they were doing at the time, the conservation status given by the IUCN, the time of observation, and whether the birds were migratory or not. All the birds in this presentation are non-migratory. This is a pie chart of all the bird species that I have observed. As you can see, the most common birds are the Asheprinia at 30 specimens, the black-tailed at 25, and the rose-ringed parakeet at 20. The least common birds are the oriental darter at 1, the pale-billed flowerpecker at 2, and the glossy ibis at 3. This is a table of the water quality of Dodnandi Lake. Why is Dodnandi Lake ideal for the observed water bus? Dodnandi Lake is ideal for the observed water bus because of the following reasons. First, it has cleaner water than nearby lakes of similar size. There's only 3 nearby, that is Belandu Lake, Vatur Lake and Yelamalapa Shetty Lake. And using reliable research paper, I can conclude that Dodnandi Lake has cleaner water than all 3 of them. Second, it has 2 islands with dense vegetation. The dense vegetation is required so that the birds can protect their offspring from natural predators. Third, it has a large size so that the birds can nest away from human contact. Fourth, it has large amounts of emergent vegetation. Some of the bird species that I have documented require emergent vegetation for nesting, therefore this is advantageous for them. Next, it has a great amount of large trees on the island and around the perimeter, again for the birds to nest. It is also host to a large variety of small insects and animals for the bird to feed on. And the lake also has large amounts of taifa. Again, some of the bird species that I have documented require taifa for nesting. These are some of the photos of the lake. First, you can see the entrance to the lake. As you can see, this is newly built. Next, you have the overview of the lake with the 2 islands visible here and here. This is a diverted drainage to the lake. There used to be 7 drains entering the lake, but in 2023, all of these were diverted by the BBMP. Therefore, no sewage enters the lake at the moment. This is the taifa at the lake. These are the tall trees around the lake perimeter. And these are 2 rose-ringed parakeets nesting in a coconut tree. Here is a stone wall that blocks any sewage if it were to unfortunately enter, and this dry patch is a buffer zone for the sewage. This is the southern outlet of the lake that drains into B. Naranapura Lake. Measures to improve the lake. While Dodnandi Lake is ideal for the habitation of the observed water birds, there are still some measures that could be taken to improve the lake and make it a haven for all birds. First, to increase awareness about the importance of lakes for urban ecosystems and the need to conserve them. All conservation activity will only be taken if the people are aware of the importance of lakes. Therefore, we need to raise awareness among them. Regular checks on encroachment, water quality, biodiversity and pollutants entering the lake. We can only know where to improve the lake on if we know where it lacks. Therefore, regular checks are necessary. Third, periodic maintenance of the lake in a way which will not harm the biodiversity. During the 2023 rejuvenation, methods used by the BBMP were found to be very damaging to the biodiversity. Therefore, I recommend using methods that are not so damaging. Fourth, introduction of specific fish species in the lake in balanced numbers. If we want to attract a certain species of bird to the lake, it might help to introduce fish species that the bird is known to eat. These species have to be native so that they don't cause any damage to the biodiversity. Lastly, ensuring that no sewage enters the lake. This is currently being done but also must be continued in the future. In conclusion, Dodnandi Lake is host to a very wide array of birds. While I observed 32 species of birds, there are reliable reports that suggest that over 150 species can be found over the year. The lake is home to relatively rare birds such as the oriental darter and the painted stork, and even the spot-billed pelican. Dodnandi Lake is also the largest lake in the area. It is necessary to conserve Dodnandi Lake since it is a vital part of the urban ecosystem, and it is also necessary to improve the lake so that it becomes a better place for all birds to live. These are the references that I have used. I have used multiple scientific reports in various journals. Thank you.
Any questions? Any questions?
Fifth slide, can you show me?
Huh?
Fifth slide. Yeah, what is this red line? Red boundary?
The red line is the outer perimeter of the lake.
You digitized this?
Yes, on Google Earth.
How you know this is the lake boundary?
The walking path of the lake, you demarcates the lake boundary.
But this is not the correct way. Cadastral maps are there. It will give you what is the exact historical your lake boundary. Then you can compare what is the present water spread area. Then you can calculate encroachment. This is digitized by you. It's an idea only.
Okay.
One doubt I have. Why you have chosen that Melbourne app, bird app? Why what made you to use that app? Another thing is like what is the percentage that accurate information you'll get? But because you can use even Google Lens. Google Lens has a accurate picture. Just you need to click the picture of the bird, you'll get to know. So what made you to use that Merlin bird app?
Merlin Bird ID, I mean, firstly, is created by a university called the Cornell Lab of Ornithology. They have a much wider database than Google Lens, and one more major reason for using Merlin Bird ID is that it has a feature called Sound ID. So I can just turn on Sound ID, and I mean, if the app detects the sound of a bird, it will immediately recognize it. But in Google Lens, as you said, I have to click a picture of the bird.
But Google also has, just by sound also they can get to know what type of bird it is.
Merlin Bird ID also, like it said in the app itself, that Merlin Bird ID is a very reliable app. The reliability is always around 90%.
How can you get to know that accurate information you will get to know, like proper detailed information of that bird is you will get to know from that app?
Because like once the app identifies a bird, you can click on the whatever bird, it gives you a detailed data set of the bird.
I think he said that it is from ornithology because it is dedicated app for birds only. So Google can be generic, but that is dedicated only for ornithology, right?
Yes, yes.
So it is from a university.
Cornell Lab of Ornithology.
Cornell. Thank you.
You mentioned that 32 species to 150 species recorded. You have done literature survey, right? Through that you said up to 150 species has been recorded. Is there any other lake which has recorded more than 150 bird species?
All the reports that I read put the number at around 150.
Serial number 40. 40.
Good morning, one and all present here. Today we are here from Vidyaniketan Public School to present about sustainable agriculture and organic farming. I am Aditi S. Kumar. I am Falguni Pragnavi. I am Kushi J. Gowda. So our topic for today's presentation is sustainable agriculture and organic farming. Sustainable agriculture is a type of farming that keeps in mind the needs of the future generation, environmental health, social equity, and economic profitability. Organic farming is a type of sustainable agriculture that uses eco-friendly pest controls and manure that is mainly based on plant and animal wastes.
The Green Revolution in India introduced many innovative farming techniques such as the The Green Revolution in India introduced many innovative farming techniques such as the usage of HYV seeds, chemical fertilizers and pesticides. However, in order to meet the increasing demands for food, over time, this has led to the degradation of soil and loss of soil fertility. Hence, organic farming, which follows every principle of sustainable development, can help to restore the ecological balance, keeping in mind the future needs of the people.
One of the biggest advantages of chemical farming is that it's a solution for the growing population of our country. It involves a large use of chemical fertilizers and use of harmful antibiotics in animals. Despite its advantage, it has a big concern, which is the impact it causes on our environment, like soil degradation and water pollution.
Our objective for this research is to find out the various differences between comparative study on organic farming and farming using chemical fertilizers. In this, we will be studying about various aspects associated with organic farming and the need for the same.
We have collected soil samples from 2 regions to test it out. First, we have collected soil from Bidgi, Vajrahalli, which this is a farm which uses chemical fertilizers in its soil. We have also collected the sample of the lake nearby to check whether the chemical fertilizers have been leached or not. Next, we have collected a soil sample from Akshaya farm in Anekal. This farm does not use any chemical fertilizers and it's an organic farm.
We collected the soil from different regions across the farmland by digging out a V shape into the land to a depth of 15 to 30 centimeters. We mixed these soil samples in order to determine the average nutrient content of the soil. So these are the experimental parameters used for soil testing, and these are the experimental parameters used for water testing.
We observed a few things during our farm visit. First, we observed that the organic soil was darker in color, it had less content of rocks, more humus content and more moist and soft. In the chemically fertilized soil, we saw that the soil was lighter in color, it had many rocks and pebbles, it had less moisture and had a rough texture compared to the organic soil.
These are the results that we have acquired doing the following tests for both our soil samples. And we have also tested NPK, and these are the results that we have acquired.
From the results of the soil analysis, we can infer that the water holding capacity and the soil permeability of organic soil is more, and the organic soil is more denser.
We have also checked the water. We have also tested the water that we have collected near the chemically farming using chemical fertilizers, and these are the results that we have acquired with the following tests. We have also interviewed 2 different farmers who use 2 different types of farming, and one farmer uses organic method and the other uses chemical fertilizers. The summary of the interview of organic farmer is that a natural fertilizer called Jeevanamrita or Dhanamrita is used, which is mainly made up of cow dung, organic jaggery, dicot seed powder, handful of soil and some water. And also natural pesticides like sour buttermilk, neem oil, burnt ash and other natural fertilizers are used. Organic farmers also use multi-layered crop rotation and mixed agriculture methods, and glyceria trees are planted in the borders of the land to prevent the seepage of fertilizers, chemical fertilizers from the neighboring land, and the breed of the cow also influences plant growth as it is used in manure. The summary of the interview from Bidgi farm is that chemical fertilizers such as DAP and urea are used.
Chemical farming have certain effects on human health. Chemical farming uses huge amounts of chemical fertilizers and pesticides. This pesticide or fertilizer contains a substance called carcinogenes. This causes cancer in humans. Almost 45 different types of cancers can be seen in rural workers who use chemical farming. About 385 million people have been affected by this, including 11,000 deaths in India per year. We can also say that it causes DNA damage and leads to a weak immune system.
By understanding these negative impacts of chemical farming on human health as well as the ecosystem, there is a need to practice organic farming. Organic farming follows and promotes complete utilization of natural resources. Techniques such as crop residue management, wherein The excess plant material left on the fields can be used for mulching, which retains the water moisture and the favorable temperature for plant growth. These residues can also be used to feed, used as a fodder for the livestock, which in turn produces dung and other organic material which can be used as manures. Initially, organic farming results in much lesser yield compared to the farming using chemical fertilizers. However, over prolonged duration of use, the soil with chemical fertilizers may no longer sustain plant growth, whereas the organic soil can still support the plant growth. Many techniques such as multi-layered farming, crop rotation, multi-cropping systems can be used to grow more crops in a small area. Techniques such as terrace farming, which is commonly known as rooftop farming, can be used in urban areas due to constraints of space, wherein people can grow vegetables and fruits on their rooftops according to their needs.
From our research, we were able to conclude that both organic farming and farming using chemical fertilizers have their own pros and cons. However, keeping in mind the needs of the future generation and its sustainability rate, we were able to conclude that organic farming is a much better practice for us to follow. Keeping in mind the texture of soil, growth rate of plants, durability, irrigation, and other aspects. These are the references that we have taken to acquire our research.
We would like to thank the IISC team for providing us with this opportunity to express our thoughts.
We are very thankful to the teachers of our school for giving us this opportunity. We would also like to thank the farmers who we have interviewed and our fellow friends and all the teachers and all the people who have supported us through this process. Thank you.
Good morning. You mentioned green revolution. Could you explain?
Green revolution was introduced as there was a scarcity of crops in the 1960s. So, in order to increase the yield of crops, many techniques like HYV seeds, which are high-yielding variety seeds, were introduced, following with chemical fertilizers and pesticides and other modern techniques. Sorry.
Just a suggestion. More than one sample from more than one farming area, it is better. You have taken one sample from chemical fertilizer using place and another one is from organic. If you have more samples, it's better.
Yes, ma'am.
I will. Okay, thank you. I will attend, but the PPT only you just check the timing. Okay, I will provide. Sound issue. Yeah, I will come. I will. You come. Timer. Okay. This one. So that's right. Here restart. Okay.
So good morning everyone. I am Ankur Gokhale from grade 9 Vagdev Vilas School, Anandpura, Varanasi. So today I am going to present a detailed study on Cordon Lake. So let me start with a brief introduction of the lake.
Kaudanhalli Lake is a small lake located in the eastern part of Bengaluru, Karnataka. It's an important green space in the city, offering a peaceful environment. The lake helps recharge groundwater and is home to several species of birds and other wildlife. The lake is also an important part of the local community as it offers a nice space for people to enjoy and connect with nature. The main objectives through this study is conducting a water analysis of the lake, conducting a study on the flora and fauna found in the lake, and finding out ways to improve the lake's condition. So this is an image of the Kaudanhalli Lake that I have taken. The Kaudanhalli Lake is a small lake located in the eastern part of Bengaluru in the Kaudanhalli area near the neighborhoods of Ramamurthy Nagar and Krishnarajapura. The lake has a total area of about 66,000 square meters or 55 acres and has a perimeter of about 1,400 meters. These are the latitudes and longitudes of the lake. The Kaudanhalli Lake is also home to several species of birds, including the great egret, common kingfisher, and the Eurasian coot. The Lake Trust. The Kaudanhalli Lake Trust, under the leadership of C. Narayanappa, has played a pivotal role in the development of the lake. It has done so by mobilizing funds and engaging the community in restoration and cleanup efforts. The lake now features a walking path, LED street lights, a kids play area, drinking water facilities, benches and other facilities. However, ongoing challenges remain like the presence of stray dogs and littering. Lake after renovation. The Kaudanhalli Lake was renovated in the year 2017 and 2018, during which they received rupees 2.5 crores from BBMP. Here, they planted trees, built drinking water facilities and public toilets, and also installed other facilities like benches and fences. The Lake Trust also received rupees 1 crore from the MLA of their constituency, using which they built sit-outs, a kids play area, a yoga area and an outdoor gym. The lake was also recently renovated in the summer of 2024, during which bunds were created. The water inlet area was renovated to avoid soil erosion, and also a filtration unit was also added. Collection of water samples. So I collected water samples from 3 different parts of the lake, giving a time gap of 15 days between each collection. Conducting water tests. So TDS and pH were the only tests which were conducted on spot. All the other tests were conducted in the lab. So in total we conducted 13 tests. The results were found to be exceeding the requirement desirable limit in 9 of these tests. The lake water also has high levels of nitrate and phosphate, which contributes to eutrophication. From the results of these tests, we can conclude that the lake has a very highly polluted water. Moving on to the major features of the lake. So what is a bund? A bund is like an embankment built around the periphery of a lake to avoid water runoff. Bunds and trenches help prevent soil erosion and also increase groundwater levels by increasing filtration. Bunds also decrease the water scarcity during rainfall, scarcity of rainfall. A waterway. A waterway is built to avoid flooding in park areas and walkways. The Kaudanhalli Lake is fully fenced for the safety of the walkers and to stop them from littering. The Kaudanhalli Lake also features boats to educate the public. The lake also has a very good drainage system with 2 paths for the inlet of water and 1 for the outlet. However, water from both the inlets is mainly sewage water from the sewages of ITI colony and Ramamurthy Nagar respectively. The Kaudanhalli Lake also faces several major problems, including encroachment, poaching, inlet of drainage water and garbage dumping. So what can we do to improve the lake's condition? Without government fundings and policies, we cannot majorly improve the lake's condition. However, we can help improve the lake by taking part in community efforts to clean the lake, informing people about the defacement caused by littering, and spreading awareness about the importance of the lake. In conclusion, the study of Kaudanhalli Lake tells us that it is an important place for both the natural and the local community. The lake features many plants and animals. However, the lake now faces several major problems, especially pollution and defacement, which threatens the lake's sustainability. By spreading awareness about the lake's importance, we can work together to sustain the lake for future generations. I would like to thank our teachers, especially the R&D department, for guiding us through this opportunity, through this project, and also I would like to thank IISC department for giving us this unique opportunity. These are the reference links that I have used for the study. Thank you.
In your methods, you told that you gave 15 days gap between sample collections. Why did you give that gap?
What happens is, as I mentioned, that there is the inlet of water is from the sewage, right? So you never know, there might be a difference in the inlet of water, like the water coming in. So we might get different results, and then there's not a great difference if you do tests in consecutive days. So I left a gap of 15 days between each tests.
What is the importance of pH in our water?
So, ma'am, pH tells us about the nature of the water, like if it's basic or acidic, and about its alkalinity. Ma'am, sorry?
What is the importance? Basic and acidic, that basic water, then acidic water, then what is the problem?
What is the problem, ma'am?
Yeah.
So, for water to be considered potable, it should be neutral. It should neither be acidic nor basic. The water
This
here
is
is
your
like.
lake water, no?
Sorry, ma'am?
This is your lake water.
Yes, ma'am.
Yeah. Then the lake water, what is the pH value?
It is
the standard value. What is the standard value of pH?
Ma'am, it is around 6.5 to 8.5.
Why that?
Ma'am, so that between 6.5 and 8.5, the water is around neutral. So when it exceeds these levels, it's either acidic or basic. But the water should not be either acidic or basic. Why it should not be either acidic or basic, ma'am?
No. It is more acidic, it is more alkaline condition. What happened to that water, that the organisms and all?
It affects the ecosystem, ma'am.
Okay.
One suggestion.
Yeah.
One suggestion. You should include DO for the on-site parameter. DO, you included only pH and second, what?
Sir.
On-site parameter. You talked about on-site.
Yes, sir. And
TDS
and pH.
So please include DO also.
Okay, sir.
That is dissolved oxygen. After some time, it will be go.
Okay, sir.
No, that
Yes,
on site.
Okay.
Yes, like this is on site.
Sir, only TDS and pH were on-site. I did DO off-site.
Understood. It's suggestion.
Okay, sir.
Thank you. 36.
Good afternoon, everyone. My name is Anya Poya. I'm Siti Markar. We are grade 9 students from Auramira International School. Our topic is solar energy. Our methodology. We took an online survey using the platform Google Form and spread it through the helps of our friends and family. 201 people took our survey where we asked many different questions about their awareness on solar energy. We have drawn multiple conclusions from our survey. I think most of us know what solar energy is, and that was the first question we asked on our survey. Out of 201 people, 2 people weren't aware of what it is, and for those who don't know what solar energy is, it is the energy derived from the light and heat from the sun. These are the applications. It's not good. Government data on solar energy. As of 31st July 2024, India is the third largest global producer of solar energy and stands at 87.21 gigawatts. The breakdown includes ground-mounted solar plant, grid-connected solar rooftop, hybrid projects, and off-grid solar. India receives significant solar radiation with an estimated 5,000 trillion kilowatts of solar energy instance. The daily average solar power plant is generation capacity is 0.30 kilowatts. Government initiatives and support. The Indian government has allocated 10 billion rupees for Clean Energy Fund and National Solar Mission. Subsidies have been provided to remove dependence from fossil fuels and adopt solar energy. And schemes, which I will talk a little bit more about later, have been implemented by the government to boost solar energy production. The Ministry of New and Renewable Energy provides regular data on the culminative physical process of solar panel installations and state-wise detailed data. The pros and cons of solar energy. The pros of solar energy are it makes us energy independent from the grid power. It also makes us independent from non-renewable resources like fossil fuels. It also provides us with long-term savings and is very low maintenance. However, there are cons to solar energy as well, as there are space constraints and the initial installation cost of solar energy is pretty high. It is also pretty sunlight dependent, so during the night, solar energy will be difficult to get. We asked people if they were aware of the benefits, and 15 people were not. The likelihood of people considering solar energy in the next 5 years. We asked people how likely they were to consider solar energy in the next 5 years, with 1 being highly unlikely and 5 being very likely. So 103 people said that they were highly likely to opt for solar energy in the next 5 years. This shows that people are indeed very keen about installing solar panels at home but have different reasons, like the cons I mentioned before, stopping them from doing so. Why are people not switching to solar energy? We asked for feedback on our Google form, and so people are quite concerned about the high costs, which is also one of the cons I mentioned, and they're also concerned about the environmental impact of manufacturing the batteries required for solar power, and they also feel that there should be more awareness regarding solar energy. So as I mentioned before, here are some of the schemes implemented by the government. The first one being National Solar Mission. The aim is to basically make India as the largest global producer of solar energy. PM Kusum is a scheme that has been provided by the government that aims to give financial support and technical support for farmers to install solar panels. The last one is Solar Rooftop Scheme. This is like how I mentioned before. They want to basically encourage people to adopt solar power, solar energy consumption. So here are some of the facets of solar energy sector: challenges, opportunities, policies, and the future prospects. Is solar power cheaper than grid power? So although the initial cost of installing solar power plant is quite high, in comparison, if we take like a 10 to 12 year comparison of grid power versus solar power, with subsidies coming in using solar power, it is much cheaper and it's much cleaner.
So this is a rooftop solar power plant installed at my house. The plant has a capacity of 8.6 kilowatts and it occupies space of 550 square feet.
This is the grid power plant in my house, solar. It has a capacity of 6 kilowatts and it is 120 square feet.
This is a deep dive into the solar power house, which is my house. So I interviewed the owner of the house. He originally considered solar to reduce electricity bills. He finds that it is very low maintenance and only requires cleaning once a month. He says that it will take 10 to 12 years to recover the initial cost, but he is confident that it will give him benefits in the long run. Solar power plant is an investment. The owner of the solar power house, as I mentioned before, the initial cost was 6,62,970 rupees. So the owner says that had he put this money into a bank, he would have received the same amount as he has saved on electricity bills. So this is the pie chart on. We asked them how many people have installed solar panels at home. So 122 people did not have solar panels installed at home.
So these are some of the conclusions we drew from our survey. So people are most likely to consider solar energy in the next 5 years. Many people do want to install solar panels, but due to cost and space constraints, they can't. People are much more keen to learn about solar energy. People are aware of the government schemes and have researched. And the final thing is that most people do, worry about our environment's health, but are also worried about the materials in the batteries and the solar power plants. Over here, we asked people as the ending question whether they wanted to learn more about solar energy, and only 27 people did not. Thank you so much. We appreciate your valuable time and attention.
Can we totally depend on solar energy?
Maybe in the long run, obviously not right now, because we also have multiple EV cars and all of that. So not only solar energy, but probably battery and electricity, but yeah, not completely solar energy. Maybe in the long run though.
How is electricity generated in solar cell? Can you explain? Solar cell.
So basically, like the solar panels collect the heat and light, and then that is the
Heat
machine.
and light.
Sorry?
Heat and light. I'm only talking about solar cells. Electricity. Solar electricity.
Yes.
How is it generated? Process.
Like the generation, I'm not completely sure.
Light converted to electricity.
Light, yeah.
How?
With using the solar panels. So.
How working? Inside there should be some material, no?
What is the mechanism?
What is the mechanism?
I'm not so sure.
Any idea why the people are not using or not popular?
So I have mentioned that in the previous slide why people are considering it. They have space constraints and it is quite expensive. Like for some places, if it's a small house, the amount of time it would take to earn the money back is a long time. Like for my house, it's only 10 to 12 years. But for other houses, it will be more. And there's also space. Like if you live in an apartment, there isn't enough space to install a solar panel.
Just come back to that. Your conventional power, conventional electricity. How do you get? What means? Is it renewable or non renewable? Because you are explaining about solar power, you should be knowing about it, right? Conventional power, for example, domestic power. How do you get it?
There are multiple ways like hydro energy and all of that, but
Hydrothermal, okay. Okay, hydrothermal,
But the reason-
hydroelectricity, yeah, okay.
The reason we chose this topic is basically because a lot of fossil fuels are used, like for petrol and diesel, and we wanted to highlight this
But
because.
you should be knowing about how electricity is produced in solar cells to compare the efficiency. Yeah?
Thank you.
I think we are getting some noise from your side, so we will, because we are not asking questions, that's why they are making the noise. Okay, next presentation onwards we will ask the questions. No, and what he told not that like that I just open it, press it, but it is not showing here, no. It is PPT only.
Good morning, everyone. With the rapid loss of forest cover in the recent years due to urbanization and deforestation, our Mother Earth is getting destroyed in a disastrous way. The very few trees left on Earth today, it becomes not only important to understand different species of trees, but it is equally important to understand the significance of trees in maintaining ecological and atmospheric balance. A warm welcome to all of you. I am Anish.
I am Chakshu.
And today, we would like to present on our project, Estimation of Carbon Sequestration of Trees in our Campus. Nowadays, one of the toughest and the hardest challenges which we all are facing today is climate change. It becomes very important for us to explore and implement strategies which will help us to mitigate its impact. One such strategy is carbon sequestration. Here, we find out the amount of carbon sequestered by the flora of a particular area. Then we find out if that particular flora is enough to make the place carbon neutral. Our campus, with a diverse array of around 467 trees, provides us a vital, unique and special opportunity to study and enhance carbon sequestration. By conducting a study on the carbon sequestration of a campus, we aim to understand how our local environment contributes to the global effort of reducing carbon dioxide levels.
These are some glimpses of our campus. This is the study area. We have used Google Earth Pro to represent this.
Our objectives are to measure and quantify the amount of carbon sequestered by the trees of a campus, to estimate the total amount of trees present in our campus. Finding the number is not only enough, so we also aim to find out their common name and botanical name. We also aim to raise awareness among students, staff and local community about the importance of carbon sequestration and the role of sustainable practices. Lastly, we aim to implement strategies which will help to increase sustainability of our school. This may include planting more trees, better waste management, etc. The methodology used here is, we measure 4.5 feet from the ground using a measuring tape. We mark this point on a tree using a piece of chalk. Then, we found the circumference of the tree in centimeter by wrapping the measuring tape around the point marked on a tree. We tried to take circumference for each tree at least 3 times, then took the average value to be more specific. Since it's not efficient to measure 4.5 feet each time, we took our chest level as standard height for measuring the circumference. Then, we refer to the dry weight conversion table from naturalresources.base.com to convert the dry weight of a tree, to convert circumference of a tree in centimeters to its dry weight in kilograms. So this is the dry weight conversion table. As you can see, the dry weight of different trees in kilograms with circumference ranging from 1.5 centimeter to 200 centimeter is clearly given here. So let's take an example. Suppose circumference of a tree is 150 centimeter. By looking at the above table, we get to know that its dry weight is around 1964 kilograms. To find out the amount of carbon stored in kilograms, we need to divide this value by 2. So the amount of carbon stored in that tree is 982 kilograms. To summarize, circumference converted to dry weight divided by 2 gives us the amount of carbon stored.
The materials used. We have used identification books and apps to identify the different species of trees. We have used measuring tape to measure the different circumference of the trees. We have used dry weight conversion table to calculate the dry weight of the trees. Result and discussion. This is the tabular column of trees in our campus.
So here we have given the common name, botanical name, followed by the number of species of the particular tree, the dry weight of the tree in kilograms, the amount of carbon stored by the particular tree, and at last we have given the total amount of carbon sequestered by all the species of trees. So, in this slide, we have aimed to find out the amount of trees required to make a place carbon neutral by the year 2030. So, to find out the amount of carbon dioxide exhaled by average human in a day, we searched in Google. So, it came 1.043 kilograms. So, to find out the amount of carbon dioxide exhaled by that human in a working year, we multiplied 1.043 into 250 because there are around 250 working days in a school. We have around 900 students. To find out the amount of carbon exhaled by them in a working year, we multiplied 260.75 kilograms into 900. So, our school was opened in the year 2008. Due to 2 years of pandemic, we had online class. So, by the year 2030, our school will have completed a span of 20 years. So, the amount of carbon exhaled by those people in 20 years, to find this out, we multiplied 2,34,675 into 20. Through our studies, we found out that the amount of carbon sequestered by the trees of a campus is around 1,33,065 kilograms. So, to find out the amount of carbon not sequestered by the trees, we subtracted 46,93,500 kilograms by 1,33,065. So, we got that our campus trees still have to sequester approximately 45,60,435 kg. We searched for an example tree for us to grow. We searched for many options. We took neem as an example since there are a lot of medicinal values as well as carbon sequestration capacity is also high. So, in a school, the average circumference is around 100 centimeters. So, the amount of carbon sequestered by it is around 334 kilograms. To find out the amount of trees required, neem trees required to make a place carbon neutral, we divided 45,60,435 by 334. With this, we got to know that we have to grow more than 14,000 trees to make a place carbon neutral. This shows that we have a long way to go to make a campus carbon neutral. Our study need not stop here. We still can do a lot of things like finding out the amount of carbon released by even the vehicles of a school, and also to make the number of trees required to make a place carbon neutral more specific, we have to convert the carbon into carbon dioxide. There's a formula for that which we didn't use. So we plan to do it in our further studies.
The present study has estimated the carbon sequestration of 461 trees in our campus. The maximum carbon sequestration capacity of trees in the campus is found in mainly banyan trees, followed by Indian beech tree, peepal tree, and camel foot. A total of 40 different species of trees are there in campus. The most dominant species of them are the silver oak, which is native to Australia, with a total of 65 trees. Through our calculation, we have found that the trees on our campus sequester approximately 133.065 tons of carbon.
Recommendations. We can organize tree planting campaigns, etc. Among all the trees in India, teak trees have the highest carbon sequestration capacity. We have around only 2 teak trees in our campus. So it can also be another option for us to go in place of neems. We can also grow trees like tamarind, eucalyptus, gulmohar, etc. By planting a mix of trees, we are not only increasing the carbon sequestration capacity, but we also, it has other benefits like providing shade, reducing heat, etc. Here we have focused on trees for carbon sequestration. We can also grow other trees which help in biodiversity, like providing maximum oxygen, providing shelter for trees, shelter for birds, and other trees which store water in their roots. Conclusion. Urban green spaces provide a major role in reducing the carbon sequestration, in reducing the carbon footprint of a place. Our campus trees not only provide aesthetic value to our campus but also help in reducing the overall carbon footprint of a campus. Our study underscores on the importance of maintaining and expanding urban green spaces within areas. In conclusion, the next step, we urge other tech parks and societies to plant trees in similar manner and join hands to protect biodiversity. Our government can also plant trees along with moving towards urbanization. These are the reference links which we used. We like to thank IISC for providing us such a great platform for showcasing our studies.
We thank Chairman Sri K. Harishwar and Vagdev Villa School for giving us this opportunity.
Special thanks to all the R&D teachers for their constant all-time support. Let's nurture the nature for a better future, else we'll be in danger. Thank you.
Any questions to be asked?
Eucalyptus trees are, first of all, they are non-native trees and they deplete the water systems of the area. So why would you plant them in your campus?
In some areas, eucalyptus trees absorb all the harmful water which is present in a particular area. So suppose we dump any wastewater, eucalyptus also absorbs them. So that's why they are planted in many places along the roadside so that they absorb many of the wastewater. So we thought just as an option for us to grow.
Thank you.
Why you wanted to study about the carbon sequestration?
Ma'am, we wanted to study about the carbon sequestration because we first thought that our school campus is very good because it has lot of trees. Then we thought, what if it's not enough? We need to grow more trees to make the atmosphere very safe. So we thought to take up the study so that we get to know more about how much far we need to grow to make our place carbon neutral, ma'am.
One suggestion, please don't promote invasive species. In many literatures we saw native species you can also plant like acacia, eucalyptus, that what silver oak, all are not native. That girl is right actually. And second thing, can you go to the 8th slide?
8th, okay.
Why you divided this circumference converted to triangle by 2?
Sir?
Why you divided circumference converted into triangle by 2?
Sir, because the amount of carbon stored in a tree is half of its dry weight, sir. So I divided it by 2.
50%. And you have GBH values, circumference you have at 1.3 meter. So why you didn't go for allometric equations? Mathematical equations are there. You can compare with that equation that is popular actually. Because this destructive method we are not doing. We are using for non-destructive method allometric equations. Mathematics is giving us all values. So you read some literature now. All things are mentioned.
Yes, sir. We look into
okay, it's another suggestion. You use your values with those mathematical equations and compare.
Yes, sir. Okay, sir.
That eucalyptus is not acceptable. In conclusion, what you mentioned, those all are non-native.
Okay, sir. We remove.
Okay. Even also native species has more carbon potentials in. They are stronger than even in yesterday's presentation. I don't know if you see or not, that evergreen forests all are native. They have more potential in mangroves, which are native to the our region.
Okay. So that's why we first thought of neem trees, sir.
Yeah, yes.
Yes, sir. We'll remove that eucalyptus and. Yes. Okay.
Okay, thank you.
Thank you, everyone.
After their presentation, we can take the break. Okay.
Namaste everyone, I'm Surabhi Shastri and I'm Anuradha Devi. We study in 9th grade in Samvida. Today we're going to present to you the bird of Kapanpa. Just imagine a world without the soothing melodies of a songbird at dawn, or the sight of an eagle soaring high in the sky, or just the simple joy in watching a hummingbird dance from bird to bird, through flower to flower. Every bird, every tree, every flower reminds me what a blessing and a privilege it is just to be alive. This is a famous quote by an author, Marty Rubin. So what will exactly happen if birds disappear? Well, if birds disappear, the plants they help to pollinate would struggle to survive, and plants would no longer grow in certain places because birds are not there to spread their seeds. Forests would become silent and slowly wither, leaving behind a barren land where life once thrived. Without birds, the world would become much quieter, lonelier, and broken. The loss of birds would mark the beginning of a much larger environmental collapse, one that would be reversible. These are the importance of birds in our ecosystem. Firstly, birds are our main pollinators and seed dispensers, as in they help reproduce crops which are very essential for humans. Without birds, plant biodiversity would decline majorly. Birds are also our pest controllers. They help regulate rodent population. Without them, it would lead to an outbreak um, it would lead to pest outbreak. Ecological balance. Birds maintain food chains and habitat stability. Without them, our ecosystem would face collapse. The objective of this project was to observe, identify, and document the birds of Kabanpa. This is a map of our study area on the left, and on the right is the path or the polygon we covered. The total area of Kaban Park is 300 acres, and the area we explored is 0.83 kilometers in perimeter and 0.02 kilometers square in area. Why we chose Kaban Park as our study area? Firstly, Kaban Park is home to diverse flora, as in it has a diverse variety of trees and plants, which is home for a range of birds, both native and migratory. Variety of birds. It is home to over 100 species of birds, including common birds like urban birds as mynas and crows, and also special birds like dogos, sunbirds, etc. Kaban Park has very quiet mornings, as in there is very less human activity, which is that silence provides an ideal area for bird watching. Here are the materials and methods we used. The method we used was transect observation. So what it is that
You walk along a set path and then you take random intervals and then you note down or document the birds you've noticed so far. And the materials we used were an iPhone 13 camera and binoculars. Here are the results of the birds we found.
Analysis. Out of the 33 birds we found, 31 were listed as least concern on the IUCN Red List. One was listed as threatened and the other one as vulnerable. The Ordidae family was the most common one we found, and followed by Corvidae and Phalacrocordae. The most common species were cormorants, egrets, herons, and mynas.
Here is the data analysis of the families. The most common one was Ordidae. So why did we find less of one family and more of the other one? So we mostly hung out at water bodies expecting birds to be there. So the Ordidae family stays mostly near water bodies because of the food source. They mainly eat fishes and their beaks are shaped in a way that they're meant to catch fishes. And then their hunting strategies, nesting and breeding, shelter and safety.
Another concept we were introduced to was parallel evolution. What parallel evolution states is that two birds belonging to different families, they often develop same features, as in their beaks are often sharp or they have longer wings. A common example is the Indian cormorant and the black darter. They both are water birds and they exhibit parallel evolution, as in their hunting strategies are kind of similar, and sometimes birds also mimic their calls in order to attract prey or other birds.
So with the observations and conclusions we've made so far, we found mynas and crows and pigeons mostly. And then birds belonging to the Ordidae family were found the most because we were usually hanging out near the water bodies. And we also found more birds in Kaban Park because it supports various habitats.
Here is a list of the references we used. Acknowledgements. We would like to thank the Lake 2024 organizing committee for this amazing opportunity, and our school Samvida for letting us explore this, and also our parents for cooperating with us.
We'd also like to thank Rijulal sir for giving us this experience. So here are some pictures we took, and we apologize for the bad camera quality because when we went to Kaban Park, to our surprise, we weren't allowed to take any cameras, so we managed with what we could. Thank you.
I have one question. In the Kaban Park, lot of the human disturbance is there, na? That will affect the birds population or something.
We actually once visited in the evening. When we went in the evening, there were a lot of people there, so we couldn't really observe any birds. We only found about 2 to 3 species on that day. But on the other days, we went early in the morning, like 6 o'clock to 8 or 9. During that time, we could find birds.
Then what about the pigeons?
Pigeons.
They're more commonly found in urbanized cities and areas because they're more.
What about your park area?
They were also found there.
But you were not mentioned about that, na?
We have.
No. In the commonly found species. No. Yeah, here. You are given only 4 species.
Yeah.
Any other?
are the main pollinators, birds or bees?
Bees.
Then why did you mention birds are the main pollinators?
Yeah, we never said about the main pollinator.
In slide 10, you said that out of the 33 birds that you observed, 31 were least concerned, one was near threatened, and one was vulnerable. Which birds were near threatened and vulnerable?
The that one. The Scudra cormorant is vulnerable, and Oriental ibis. The Oriental ibis is nearly threatened.
Okay, thank you.
Okay, thank you all. Now we can move to break. Okay, we can have good food and come back. Not the one. Now 1:05. No, 1:35. No, 1:35. We will tell. Then they will come.
So guys, please
1:45. 1:45.
1:45 assemble back here only. And you guys know right where to go? Netravati mess. Yes.
ready? What is? Hello, hello. Okay. You did. You finished your lunch now? That means you have a lot of energy now. You had paisa. Huh? Okay. Sambar? Ah. Huh? Ah. Okay, now we can start. Full of energy? Okay. Then? Vaibhav, we can close that.
Good morning everyone. My name is LK Vaibhav and I am a class 9 student from Vagdevi School, Vattu. Today I am going to present my project on the impacts of urbanization in Bengaluru, which is a part of the late 2024 Symposium focused on wetlands for human well-being. In this, the lucid BLR stands for Land Use Change Investigation and Demarcation in Bengaluru. This name was chosen as it reflects the project's focus on analyzing and mapping the significant changes in the land use patterns in Bengaluru. Also, the term lucid emphasizes the clarity and understanding the project aims to provide regarding these transformations. Today, I will guide you through the following sections: the introduction, the methodology, the study area details, results, discussion, and finally the conclusion. So let's begin with the introduction. Bengaluru, often referred to as the Silicon Valley of India, has undergone remarkable development over the past few years. Starting from its humble beginning as a garden city, it is now often referred to as a concrete jungle. The problem arises with the increase in population, which was about 7.96 lakh in the year 2009. It has now currently increased to 1.4 crore in the year 2024. Therefore, this project addresses the rising concerns of urbanization and its impacts on the environment. The primary objective of my project is to perform a comparative study of land use based on mapping using Google Earth Pro. Specifically, I aim to assess the land usage, quantify urban expansion, and monitor the status of the natural resource. For this, I utilized Google Earth Pro and QGIS software to create a boundary polygon, and then the areas were categorized as agricultural land, water bodies, built-up areas, and the forest areas and the barren land. My study area includes an area which has a perimeter of about 39.8 kilometers and it has an area of about 93.5 square kilometers with the coordinates as shown. My study area has about 12% of Bangalore's total area, which is 741 square kilometers. Some of the key locations in this area include Kadgudi, Whitefield, and parts of Kerpuram and Hoskote. So based on the categorization, I mapped a total of 1,213 individual places for both the years of 2009 and 2024. This extensive mapping provides a comprehensive overview on how urbanization has impacted us over the years. So let's start with the agricultural land. The agricultural land, which was about 43.67 square kilometers in the year 2009, it has now currently decreased to about 28.02 square kilometers. The decrease in the agricultural land is a direct consequence of urban expansion. And next comes the built-up areas. In the contrast with the agricultural land, the built-up areas has seen a significant increase. The built-up areas, which was about 25.72 square kilometers, it has now currently increased to 46.88 square kilometers, and it shows an increase of about 82%. And now comes the water bodies. The water bodies, which was about 4.16 square kilometers, it has now currently decreased to about 3.64 square kilometers. The loss of water bodies plays a major role as it affects the city's water needs and its biodiversity. Now the forest areas and the barren land. Here the green areas represent the forest areas and the barren land. In forest areas and barren land, which was about 90.96 square kilometers in the year 2009, it has now currently decreased to 14.02 square kilometers, representing a decline of about 29%. So this is the map for both the year for 2009 and 2024. In summary, the results indicate that the built-up areas continue to increase while the agricultural land, forest areas, and the water bodies go on a decline. Later, the data which was obtained from Google Earth Pro was transferred to QGIS software. This allowed for the creation of specific detailed maps that enhanced our understanding on how urbanization has impacted us. So this is the map which was done for the year 2009, with the differentiation marked as agricultural land, water bodies, built-up areas, and the barren lands. So this is the map for the year 2024. So this pie chart provides a visual comparison between the 2 years with a gap of about 15 years. We can understand how the changes have occurred. The impacts of urbanization in Bengaluru is multifaceted. We are witnessing large-scale rise in buildings, while there is also decrease in the agricultural land and the water bodies. Therefore, this leads to many environmental problems, including the decreased groundwater table, increase in the pollution, poor air quality index, and also increased temperature because of the urban heat island effect. This is the temperature for both the years 2009 and 2024. In 2009, the temperature, which was about 21.4 degrees Celsius, because of the urban heat island effect, it has now increased to 24.1 degrees Celsius. And future indications show that the temperature might still increase more if urbanization continues at this rate. It is important that we take some necessary steps to mitigate the impacts of urbanization in Bengaluru, such as promoting localized climate adaptation measures, establishing green roofs, vertical gardening, and permeable pavements which allow for the percolation of rainwater. Also, establishing growth boundaries and investing in green spaces is also very essential. In conclusion, balancing urban expansion with conservation efforts is very essential if we want to mitigate the impacts of urbanization. Block the sprawl, let nature thrive. In green spaces, our spirits revive. Cherish the roots, keep cities in check. For a balanced world, let's build and connect. So these were the references which I used for this project. These were very invaluable in shaping the outcome of the project. Finally, I would like to thank IISC for providing me with the proper opportunity to participate in the Lake 2024 Symposium. Also, I would like to thank Vagdevilas Institution as well as the R&D department for their continuous guidance and support throughout the project. Urbanize with care, keep nature's flair, concrete jungles rise but green must share. Thank you everyone. I look forward to your questions and discussions on this important topic. Thank you once again.
Any questions to be asked? Any questions?
You digitized each class built up agriculture in Google Earth in both year 2009 and latest. Then where you calculated the area?
Sir, just I mapped the area and then I added it manually.
Where you calculated in Google Earth Pro only or?
Yes, sir.
Yes, that I'm asking. Then in QGIS, some analysis you did?
Yes, sir.
What?
No, sir. Last time they just asked for to create maps just to show direction. That's it.
Okay, thematic maps you prepared there.
Yes, sir.
It's a lot of hard work because you digitize these data. Nowadays we are using machine learning algorithms, deep learning to do this. Because it's a good thing because according to your class, you did the right job. But in future you can look also artificial intelligence you can use.
Sure, sir.
Within some time you can do this work. But according to your class, it's a nice job. So you digitize each classes.
Yes, sir.
So what is the prominent thing you observe?
Sir, I found out that the built-up areas have increased considerably, and while the other areas have declined, have gone on a decline.
Concretization.
Yes, sir.
Okay. Thank you.
Did you identify the reasons for marketization?
It was mostly because of the increase in the population. In 2009, the population was 7.96 lakh, but in 2024, because many people also come from other states, because Bangalore is not only about only people from Karnataka, it's many people come from other places because of its IT hubs, and as it's also known as the Silicon Valley of India. In 2024, the population is now currently 1.4 crores. It's mostly urbanization is because of it's driven by the population growth.
So you went to the for ground sampling also, like for the verification?
Yes, sir.
Simply on Google Earth?
Yes, sir.
Okay. Okay.
Thank you very much.
Yeah. Yes, you can start. Here, correct, huh?
It is correct. Yes.
Namaste everyone, I am Pooja.
And I'm Niharika from class 9A. We are the students of BGS National Public School. So here we are going to be presenting about effects of butterflies on invasive plants.
Objective of our project is to find what are butterflies and invasive plants, and how are these invasive plants different from the normal plants, and the habit of butterflies and the invasive plants again. Role of butterflies and invasive plants in the sustainable environment. How does these invasive plants affect butterflies and human life?
So this is the study area where we did. It was in our school and another one in the nearby parks of our area. So let's start off with the introduction of butterflies. So but as we know, butterflies are winged insects, and they undergo 4 stages to complete the entire life cycle. Stage 1 eggs, stage 2 larva, stage 3 pupas, then we get stage 4 butterfly. And yeah, butterflies are usually found active during the summer afternoon times. So few examples of butterflies are common glass yellow and common crow. So the common glass yellow was found flying close to the ground near the open grass area, and the climatic condition to which it adapts is the sunny season. It's found during the morning time. And next, the common crow. It's often seen in the herbs place. Its weather is um, a warm and humid climate. Okay, now let us know about the invasive plants. These plants do not occur naturally. These are either spread by animals like birds, and these plants have a very aggressive root system so that they can kill the other plants which they grow nearby, and they densely populate near other native species, and they do not need any fertile soil to grow. They produce a large quantity of seeds. Okay, these are not the differentiation between normal plants and invasive plants are normal plants are originated and developed in their own surroundings. Like if you take a example of apple, it's only produced in the Kashmir and it needs the cold climate. It can't be grown in Bangalore. So invasive plants, these are very harmful to the environment and they can be like very harmful to the native species also. And they naturally grow near the lakes, forests, rivers, agriculture, and etc. And the native species do not create any harm to the human life, whereas the invasive alien plants create human harmful to human life. And the normal plants do not cause any harm until and unless you have an allergy. But invasive alien plants also be uh, like implicated in the spread of viruses such as flu and HIV. The role of butterflies in a sustainable environment. So basically, the butterflies they attract to colorful and bright plants. They suck nectar. The body collects pollens which are used for the other plants. So now let's see how does these invasive plants affect the human lives and the butterflies. So mainly to talk about the human lives, these invasive plants cause diseases like wounds, stings, bees, and allergies. Also, they affect the food chain. Also, they affect the food chain, which also it's also bad. Also to look at the butterflies. If the butterflies are attracted to the invasive plants, then it causes excess pollination. If the population of the invasive species increases, then there would be a loss of native species. If there is loss of native species, then the butterflies cannot reproduce successfully. Now let's take an example of an invasive species which is milkweed. And this milkweed is potentially poisonous to human beings as well as animals like cattle, horse, etc. This milkweed plant contains a broad leaf which contains the two poisonous called as cardiac glycosides and cardiolides. First, let us know about cardioglycosides. These have some medical properties as well as some poisoning agents. This cardioglycosis is being used for rashes, respiratory issues, etc. And this cardiac glycoside disrupts the molecular pump that allows the flow of in and out of the ions of a cell. And this I've mentioned about the molecular pump. Molecular pump means the transmembrane protein which allows the movement of in and out of solubles and ions through a biological membrane against the concentration gradient. Now, I've mentioned one more toxin called as cardiolides. This is being consumed in the larva stage of the butterfly. This is actually present in the leaf. So, caterpillar consume the leaf and they get this poison in their body. And this mutation happens due to their consumption of this in the earlier stage. And this has made this two poisons have made the butterflies toxin resistance. So they have a better tasting if any predators come and try to attack them. And they can tolerate this food source because of a particular protein called as sodium potassium pump in their body. And this cardioglycosis, it helps.
Yeah, it is helpful for the butterflies to protect themselves from any predators too. So this was the field work which we did near our park, and this was our area. This was covered with invasive plants, and after removing, you could see a very clear area. So from this, we can say that the invasive plants do not need any fertile soil to grow. So this was the field work which was done in our school, and yeah, this was done near my home, and yeah, the field work. Conclusion, I would like to conclude that we should plant more native species instead of more invasive plants, so that the rate of butterfly species can increase. Unless and until we know the plant, we should not smell, eat, or touch them. Before buying a plant, we should know whether they can attract butterflies or not. We should keep our surroundings clean from any invasive or host plants. Report any invasive plants are there to your local Interior Bureau Field Office. Thank you.
What is Interior Bureau Field Office?
It's like the offices near, like forest department or whatever like that.
So it's available in cities as well?
Yeah.
Okay, thank you.
About milkweeds, did you say they're invasive species?
Yeah.
I've owned a milkweed plant. I'm not sure if they're invasive or not, but I found that they need intensive water care and they need fertile soil to grow. So
Yeah, like not every invasive plant require very fertile or non-fertile. It's according to their classification, like whether they need a very fertile soil or a non-fertile soil.
The example in the figure is that what we was getting. And then they showed the soil after removal of.
Yeah, that's not the milkweed plant which was only there in that case. It was Langankana and Paramecium.
While naming, there is something called normal plant, abnormal plant. Okay, you have said normal plant. That's not the correct naming of it. Okay. So you put it as native species.
Okay, sorry.
also you mentioned that a lot of invasive species of plants have negative effects to the indigenous plants and animals. What would be the negative impact on butterflies from the milkweeds, or like any animals?
Okay, if you take the case of butterfly, apart from the monarch butterflies, they have a resistance to the toxin.
Isn't that the plant's defense mechanism to protect its leaves from not being eaten?
Yeah, that's why the butterflies die. So like only the monarch butterflies are resistant to that butterfly, like apart from the other butterflies. That's why no other animal is like consuming it in a quantity, like other plants do.
I'm not convinced with that, that's what. We should keep our surroundings clean from any invasive or host plants. Two are different aspects, right? Some need host plants. It may not be harmful also. Okay, thank you.
Good morning, everyone. Good morning, everyone. I am Manvi.
I'm Maitri.
We are from Vagdevi Vilas School, Biddi. Here we had to present a presentation on analysis of Vrushbhavati river water.
So the widespread problem of the water problems are affecting our health. So the unsafe water kills more people. The unsafe water kills more people each year than the other war and the violence combined. So less than 1% of the fresh water is actually accessible to us. So if we will not take up the challenges, then we can face any problems in the future. So water is uniquely vulnerable to the pollution. So the toxic substance from the farms such as fertilizers, pesticides, and from the towns such as sewage disposal, and from the factories, the chemicals from the factories get readily dissolved and mixed with it, causing water pollution. So the sewage is if the sewage is discharged untreated, then its phosphorus and nitrogen content can lead to the pollution of lakes via process called eutrophication. So as the population is rapidly growing, they may pose the problems such as contamination of the surface waters and sewage disposal. And our main objective is, we wanted to know whether the river is being polluted, and if yes, we wanted to know the water pollutant, the amount of water pollutants present, and we wanted to monitor the soil quality because this water is being used in the surrounding agricultural lands, and we wanted to investigate some ecological impacts of it, and we wanted to identify some remediation strategies for this problem. And we wanted to raise awareness among the people and promote education. And this is our mapping of study area. And this is our sample collection method of both soil as well as water. And so the on-site parameters like pH, temperature, electrical conductivity and TDS were also checked. And methods of soil analysis. So estimation of pH was done using pH reagent 1, reagent 2 and the decolorizer, and finally we compared the results with the pH color chart, and in the same way for the nitrogen, potassium and phosphorus we used their respective reagents and decolorizer, and we compared the result with their respective color charts. And these are some images where we are doing the analysis of soil, and here we are comparing the results. And materials and methods for water analysis. So for estimating dissolved oxygen is very important for the respiration of aquatic organisms. And for estimation of dissolved oxygen was performed using manganese sulfate, alkaline potassium iodide, and potassium and phosphoric acid. So these were the reagents, and starch was used as an indicator. And this sample solution was titrated against sodium thiosulfate. Turbidity. So to check turbidity, we had two vertical tubes that were given in the soil water testing kit. So at the base of one tube there was a cross mark visible. So we joined those two tubes and we started pouring the water until the cross mark became invisible, and at that point of time we noted down the nearest NTU value. The total hardness was estimated using adding ammonia buffer and eriochrome black tea powder, and was titrated against EDTA till the color changed from wine red to the blue, and the amount of EDTA consumed was noted to calculate the total hardness. The residual chlorine. So first the pH of the sample water was tested by using the pH paper, and after that 8 to 10 drops of the orthotoluidine reagent was added, and the color developed was compared with the standard color chart.
Here are our results of our on-site parameters which we performed, that are pH, temperature, TDS and electrical conductivity. They are all within the limits. The standard permissible value which we took from the Bureau of Indian Standard. And dissolved oxygen, residual chlorine, phosphorus, iron and hardness were also checked. So we can say that like mostly they are all within the permissible limits, but iron is almost reaching the standard permissible value. And alkalinity, turbidity, chlorides and ammonia were checked, where turbidity is very high, like 100 NTU, but the permissible limits are 1 to 5 NTU, and even ammonia content is high in the water, and chloride content is quite less. And here are results of soil analysis. So the observed pH was 7.5. So we can say it is slightly alkaline. So the alkaline soil must be treated with gypsum, which is a base. So it neutralizes the soil. And nitrogen. So the nitrogen level what we got was low. So according to some web pages and soil health card and all what we referred, so we got that if it is low, it will be lesser than 240 kg per hectare. So nitrogen was 240 kg per hectare.
And the phosphorus is low, so we can say that we can add 25% more, and the potassium is medium, so that it is manageable, so no need to add more or less. So we can conclude that the river is getting polluted as the time is passing through. So in the present investigation, we can see that many parameters found to be increased, and these kind of changes would affect the aquatic environment, which leads to the process of eutrophication, and we have to take up some of the purification methods before introducing any foreign material to the body.
Some recommendations are we have to totally ban the activities which cause the water pollution, such as discharge of untreated sewage, industrial effluents and immerse of idols, etc. So we have to follow proper bioremediation techniques, so which will be used to improve the water quality. And these were our references. We use water quality analysis testing kit, Jaltara users manual and nice soil testing kit. Thank you.
What is the unit of EC, electrical conductivity?
Ma'am, we didn't know the unit, and we just wrote down the value.
Then how you measured this one? EC, electrical conductivity.
Microsiemens, ma'am, yeah, microsiemens per centimeter, I think. Microsiemens. I think. Ma'am, I got confused with that. But tell your microsiemens. Ma'am, microsiemens per centimeter.
Okay, thank you.
Thank you, ma'am.
What is soil health card and why it is used?
Actually, soil health card is given by the government. So there we'll get to know like it is mainly about the soil. So like nitrogen content, phosphorus, and many other metals which will be dissolved in the soil. So we'll get information about that.
Okay.
Thank you, everyone.
Good afternoon everyone. Myself Hitashriam. I am studying in 9th grade. I am representing Bangalore Blaze Girls High School from Bangalore. My topic is soil study. Introduction. Soil is the loose surface material that covers the most land, consisting of inorganic particles and organic matter. We wanted to know, even if the same vegetation was found in different areas, whether there will be a change in soil quality. We decided to take samples from 6 different areas. Actually, when we took, it was 5, then we added 6 with the almost similar vegetation to study in detail of porosity, acidity, alkalinity and nutrient contents. Objectives: to analyze the characteristics of soil in a given area, to study how the soil quality affects water quality and availability, to evaluate the fertility and nutrition content so that the suitable plantation could be suggested, to determine any salinity, acidity or alkalinity problems, to understand the compressibility strength of different soil, to analyze the effect of various chemicals on the soil. Materials required: soil samples, soil testing kit, pH stripes, measuring jar, distilled water, TDS meter, weighing machine, logbook and pen. Test for minerals. We dried a small portion of soil sample, heated it in a sun to dehydrate, taken the sample in a bottle, added 10 milliliter of distilled water and shaken well, left it for 10 minutes to settle, collected the filtrate in a test tube, divided the filtrate of each into 4 portions to test pH, nitrogen, phosphorus and potassium. So this is the comparing the result with phosphorus color chart, and this is with potassium color chart. Testing minerals. We use a soil testing kit in our chemistry lab to check nitrogen, phosphorus and potassium. So our samples were from Nagarbavi, Nayandali, Malatali, Atiguppe, Chandralayot and Kengeri. So the nitrogen content of Atiguppe was medium and other were low. The phosphorus content of Malatali sample and Atiguppe sample were high, and the sample from Kengeri showed medium and other were low. The potassium content of Nayandali, Malatali and Atiguppe were medium, other were low. Actually, the high levels of phosphorus can cause the plants to grow poorly or even the plants may die. Testing porosity. We weighed the 10 gram of dry soil, taken the soil in a test tube, then we added a distilled water from a measuring jar using fillers. So this was our observation. Our sample from Nagarbavi showed medium porosity and sample from Nayandali showed low porosity, other were high porosity. Since the soil is compact, 30 to 40% porosity is considered as high. pH testing. Taken the sample soil in a beaker and added distilled water, dip the pH stripes to find the color change, check the strips against particular color gradient. So this was our observation. Here the sample from Nayandali and Kengeri showed mildly alkaline and other were highly alkaline. Generally a pH between 6.0 to 7.5 is suitable for most of the plants. We read that the hard water with high mineral content can contribute to higher pH level. We also checked that even the same kind of plants were found commonly in this area. We found that the guava plants were common in this area because the guava plants may grow in alkaline soil, so this can be the reason. Testing total dissolved solid. Filtered the soil sample after adding the distilled water. Check the filtrate with TDS meter, recorded our observation. So this was our observation. TDS in the soil that is less than 250 milligram may indicate that the soil lacks healthy amounts of minerals like magnesium and calcium. For agriculture, a total dissolved solid level less than 700 milligrams per liter is considered safe. Testing of soil texture. Put the soil sample in a measuring jar, poured a small amount of water and shaken well, left it for settling for 10 minutes, recorded the layers and depth of each layer in millimeters. So this was our observation. We found that our sample from Nagarbavi was silt loam, other were sandy loam. In general, soil with 2 to 4% of humus is considered healthy. In that aspect, sample 1 and sample 6 can be healthy, but sample 1 could be more healthier because it is silt loam. Silt loam is that soil material that contains 50 to 80% silt and less than 12% clay. Sandy loam is that 7% clay, less than 50% silt and between 43 to 52% of sand. Testing soil samples for plant growth. Taken the soil samples in milk cover, sowed Menthia fenugreek seeds. For better observation, we shifted the plants saplings from milk covers to paper cups, and this was our observation. Here we found our sample from Nagarbabi was growing fast and Nayandahalli was low, Malathalli was very slow and other were average. Since all the six samples were receiving equal amount of water, sunlight, we assume that this is definitely the nutrient in sample 1 made the samplings grow faster. So this is the comparative study of all the samples. Link between soil quality and water availability. We have read that higher the humus content, greater the chance of water retention by the soil. It is also said that higher the percent of sand, weaker the soil. Our sample from Nayandahalli showed more percent of sand. Of all the 6 samples, the soil from Nagarbhavi and Kengeri area showed more humus content, so we assume that the water availability can be more here. However, the further conclusion can be drawn only after studying the depth at which borewells are dug. So we are concluding that soil study gave us an insight into various factors that affect plant growth. We found that though the soil samples showed light variations in pH, mineral contents and texture, they supported similar vegetation. For example, we found that the guava plant was common in these areas. Since the soil samples were analyzed to be alkaline, native plants which grow in alkaline soil could be grown widely here. Recommendation: soil study can be made a part of curriculum in school. It is essential to share the soil type so that a suitable vegetation could be suggested. There could be a portal which is easily accessible and through which authentic soil study of every area could be publicized. Our search about organization that conducted soil studies showed that they do so at a cost and provide the person in need takes the sample to their premises. So far, soil health cards are issued free only to the farmers as a part of RKVY by the Government of India since 2014 to 15. Though there are several websites related to where to do the soil study, we could find only general information, not the specific. Soil study, if encouraged in school and college, can lead to create awareness among the public about the need to conserve such precious gift of nature. Since the percent of sand decides the strength of soil, we feel the authorities could take more care about the constructional activities in such areas. We thank Dr. TVRAMACHANDRA sir and his team of CES IISC Bangalore to provide us an opportunity to present our project. We also thank our school management, office coordinator Ms. Pushpa, and teachers and classmates for accompanying us in our project directly or indirectly. We even thank our parents for supporting and their encouragement. Our sincere thanks for Mr. Vrijilal sir for his motivational classes during the Schools of Prakriti Koplam. And these are our references. Thank you.
Any questions to be asked? Any questions to be asked?
How we can write the pH one? pH.
We check the pH.
Yeah. How you will write in your notebook?
We will first check that in a gradient.
pH. How you will?
Small p, big letter H.
But in some cases, go back.
No, here.
There. Slowly.
It's
here. PK.
Last.
Yeah, here the P has become mixed.
Go to conclusion. One more page is there.
questions?
What do you mean by highly alkaline
Like
condition?
alkaline will be like one minute. Like 7 to 8, it may be like mildly alkaline, but not conformly alkaline, but mildly alkaline. Like 8 to 9, it may be alkaline, full alkaline, so it's called highly alkaline. Like we didn't got the perfect result, either it is 7 or 8, so we written mildly alkaline, other we got 8 to 9, so it was.
Then alkaline water we will use, what happened to our health?
No?
Alkaline water we will use, what happened to our health?
If we use alkaline water, what will happen to our health? That's what she's asking.
So, but it is about soil, I just
saying.
It may.
Anyway, coming to the 3 and 4 sample area, Mallathulli and Atiguppe seems to be very, I think pH also is more. You can go back to other observation. Can you go back? Yes, here also it's high now. What might be the reason?
Actually, in this area, we found that the soil was little hard and mixed with little cement.
Why? Surrounding, did you look at the surrounding?
Yeah, like construction was going on in these areas.
Only because of that?
Even the cement was mixed with the soil.
Not only porosity, go back to the other data.
I took one food.
Yes, before. Go back to before.
Like everything.
The data, no, it's
Yeah.
everything. So you showed some other data where Mallathulli and Atiguppe had high content of something. This show. No. Okay, fine. Thank you. One question.
The girl just now presented, please come here. The girl just now presented. There's one question to ask.
You mentioned many study areas. So, like where exactly in those places did you do your research?
Took it in a site. Nagarbavi was in ground, other were in site. Like properties. Yeah, but there were so many other plants also grown there.
Nagarbavi, there was no like.
But it was near our school where we found a ground, so we took it there. But other were constructional areas.
All were similar.
Yeah, all were similar, leaving Nagarbavi.
Another question, like why do you have to add only distilled water by doing the soil porosity?
Normal water content, it will be containing minerals of, but the distilled water is pure. If we put normal water, it can even show its mineral content for pH. So we use distilled water. Standard.
Super.
22, 37. 37.
Shall I start?
37.
Greetings everyone. I am Mohit, this is Navneet and this is Status. We are from Vidyaniketan Public School, Bangalore, Ullal, and we are presenting our project on the topic Study of Contamination of Nayanandi Lake. Why is it that everyone is emphasizing so much about lakes? This is because lakes are important freshwater sources which support daily human activities and they provide indispensable services to humans. The quality of water sources and lakes are a key indicator of a country's sustainable development. Despite advancements in technology, water quality across the globe is deteriorating. In fact, 80% of diseases in developing countries is caused due to the poor water quality sources. So now let's talk about a lake in our neighborhood, the Nayandahalli Lake. Nayandahalli Lake, located in RR Nagar, Bangalore, spans over an area of 15 acres and has a capacity of around 120 million liters. The samples that we have collected are from the eastern side of the lake, and the sample 2 and 3 are from the southern side of the lake. The special thing about Nayandahalli Lake is that among the 200 lakes in Bangalore, it is filled with purified water. This is the mapping respectively, and it's a mapping of the samples. As we can see in the picture, the outlet is close to the inlet and the presence of sewage next to the lake, and the outlet is connected to the sewage. The objective of our study is to analyze the lake water quality by measuring the physical, biological and chemical parameters, and to check the presence of contaminants like microplastics after the treatment. The materials that we have utilized are water samples, pH kit, total hardness kits, residual chlorine and fluoride and chloride kits, microscopes, BOD and DO materials, and calcium kit.
So now let's move on to the physical parameters of the lake. All the samples were light green, 28 degrees Celsius for sample 1 and 29 degrees Celsius for sample 2 and 3. All the samples were transparent, odorless with some suspended particles. So now let's move on to the comparison of chemical parameters. The standard range of pH should be 6.5 to 8.5, which has been satisfied only by sample 1. The total hardness should range between 200 to 600 mg per liter, which is satisfied by none of the samples. The calcium should range between 75 to 200 mg per liter, which has been satisfied by sample 3. Magnesium should range between 30 to 100 mg per liter, satisfied by all the samples. The lake is free of fluoride. The total dissolved salts, it should range between 500 to 200 mg per liter. The residue, which has been satisfied by none of the samples. The residual chlorine should be 0.2. However, the lake has no residual chlorine. The dissolved oxygen should be more than 6 mg per liter, which has been satisfied by sample 2 and 3. The BOD should be 2 mg per liter or less for 5 days at 20 degrees Celsius, satisfied by sample 2 and 3. So this is the biological parameter or the microbials that are found. Firstly, we found spirulina. Spirulina are basically green algae that are significant in lake water. They are important to remove nutrients such as nitrogen and phosphorus, and also enrich the contents of potassium, antioxidants, carotenoids, etc. Microcystis, it is usually found in very large blooms. It is a cyanophilian member, hence also known as cyanobacteria. It also helps to remove the toxin content, which makes it to resistance for gazing population. Spirovera are basically free floating green algae which are present in freshwater habitats. They increase the level of oxygen in the habitat they are present. Microplastics are basically large lumps of plastic which have been degraded into small pieces of plastic which range up to 5 millimeters in length. They are very harmful both for aquatic bodies and aquatic life. They alter many situations of aquatic species, such as their relation between the prey and the predator, and their water moving patterns, etc. It is also found that the microplastic contaminated water causes cancer. So now the conclusion of the biological parameter, the presence of spirulina is basically due to high pH value. The spirulina which is found abundant in the lake is very good protein substituent. Microplastics are found in the lake even though the lake had went for purification in the mechanical plant, meaning that it is not satisfactory. Though the lake has undergone purification in the water, it contains some more harmful substances in it, meaning the government should take more care on this situation.
So this is the picture gallery of me and my team performing experiments in the lab. This is the picture of Nayandali Lake. So now let's move on to the results and discussions. The Nayandali Lake has higher pH than the ideal one. Due to the purification of the lake is free of chloride and fluorine. The lake is less in calcium content, and also the physical parameters and the biological parameters are not in the way they should have been according to what the lake advertises. Now let's move on to the recommendation. The purified water should be utilized for practical purposes rather than being left stagnant or used as a path. Even the government resources should be directed towards more beneficial and impactful initiatives, such as we should educate the community, should also organize cleanup events. We should make use of specific bacteria which can degrade microplastics, and we should use specific plants which can trap microplastics. So these are the pictures of some plants and bacteria which can trap microplastics and break them down. So the first one is Legionella saccharis, second one is Pseudomonas pyrrhida, third one is Pseudomonas aeruginosa, the fourth one is Phragmites australis, and the fifth one is Staphylococcus. So these are the links and websites that we use for our presentation. We would like to thank the Biennial Lake Symposium team and TVRAMACHANDRA sir for this wonderful opportunity and an amazing platform to work on. We would like to thank our teachers and our mentors and lab assistants for their support. We are indebted to our school, Vidyaniketan Public School, for their support, and also we would like to thank our parents for their support. Thank you.
Any questions to be asked?
Yeah, as mentioned in the presentation, is a good substitute. It is a good substitute for protein and nutrients. So in that way, it can be effectively used.
So for what purposes it can be used?
Yes, as protein substitutes for as medicines and etc.
Spirulina under green algae or blue-green algae?
Green algae. Green algae.
Now, spirulina under? No, some scientific words, no, we cannot skip, okay?
So you said that the government advertises something about the lake. So you said that it is not as per the advertisement. So what all should be there actually?
As we could see in the chemical parameters, many of them didn't meet up with the BIS standards. According to what the BBMP reports advertise, it is completely purified. I mean dual water purification, and we cannot see the results over here.
Okay, thanks.
You guys know what is BIS full form, right? Yes, okay, fine, good. Thank you.
Forward, backward, long press.
Sorry?
This is a forward, backward, and this is long press pointer.
Oh, so the line.
This half.
Good afternoon everyone. I'm Krishpreet Kaur and I study in grade 9 in Automira International School, and today I'll be speaking to you about the birds of Sena Vihar. Sena Vihar is a noble residential complex situated in Kalyan Nagar, which is situated in the north of Bangalore. Its green spaces attract a lot of bird species, and today I'll be talking to you about the observations I made about these species. My methodology. First, I studied the role of birds in the environment. Then, I found out the importance of bird watching. Then, I identified how urbanization affects birds. Then, I also identified the various threats to birds in urbanized areas. Then, I referred to literature from experts, made observations during these bird walks, conducted an online survey, identified the genus and species of the various birds observed. I then concluded my research. So, role of birds in the environment. We all know that birds play a very vital role in the environment. We know that they fertilize blooms and pollinate plants. They also eat seeds and then get it back, give it out again, and then which come back to the ground and come up as new plants again. So, importance of bird watching. There are three main things that come up when we speak about importance of bird watching. First is appreciation of nature. It helps us connect with nature. It also helps us initiate conservation efforts, and it also helps us. Bird watching can also become an educational tool. Characteristics of birds. Birds can be observed on the basis of their beak shape, plumage, and behavior. These can help us. These can tell us a lot about the breeding pattern, nesting pattern, and behaviors. While bird watching, we must observe how these birds observe their food. Mainly, most of the birds obtain their food by foraging, hunting, and scavenging. And we must also observe the nesting and breeding patterns and habitat preferences. Urbanization in birds. So, urbanized birds show a difference in their behavioral traits, showing more song frequency and bolder behavior than birds in the rural areas. Threats to birds. Few threats to birds that I have identified so far is global warming, pesticides, kite flying, and electricity wires. As humans, we all have perceptions on birds, and I conducted an online survey with the residents of Sena Vihar, asking them their perceptions of birds. So, it was noted that most of the residents of Sena Vihar agree that birds have become an integral part of their lives in Sena Vihar. They also find birds annoying or problematic because they ruin their outdoor furniture, cars, etc. They value birds in the environment because they are a part of the ecosystem. My fieldwork was conducted on 22nd June 2024, where I was accompanied by Pooja Agrani, who helped me identify the various birds and species. I was also accompanied by, on 24th August, I was also accompanied by Anisha Jayadevan. Oh, I'm sorry, I messed it up. 22nd June, I was accompanied by Anisha Jayadevan, who helped me identify the various bird species. 7th July, I was alone, where I identified the bird species on my own. And on 24th August, I was accompanied by Pooja Agrani, who also helped me identify the bird species. The tools I used were the Merlin Bird app, binoculars, reference chart, notebook, and pen. This was the reference chart that I used. So I observed many birds, one of them being the black kite. It has a forked tail and is brown in color. I also observed the blue rock pigeon, which has a blue-grey body and a very guttural call. I have also observed the oriental magpie robin. I observed the male one. It is black and white in color, and it is a very small bird. I also observed the red-whiskered bulbul, whose plumage is red or orange and has a very melodious call. I also observed the male quail. which has a glossy black plumage and ruby red eyes, I observed the bill flower pecker, which is a small pale bird and is really hard to identify. I observed the purple sunbird. I did not know this is the male sunbird, so I haven't identified the female sunbird yet. But the male sunbird is purple and blue in color and is quite a small bird. I also observed house crows. Like people say, where there are humans, there are house crows. So, yeah, it is a black bird with a grey neck. Then I also observed the white-cheeked barbet. This is actually quite a loud bird, and it's shy. You can't see it often, but you can hear it quite often. Then I saw the myna, which is dark in color again with a yellow beak. Then I saw the egret, which was quite surprising because it's a water bird. But if you notice, the bird has an S-shaped neck. This bird was, as I put it, was found in Sena Vihar because of the Heba lake nearby. I also observed the common tailor bird, which was the highlight of the walk for me. It's a small bird and cannot be identified that easily. So this is just the tabular of the bird spotted, and this is the places of the bird spotting. So we may conclude that birds play a very important role in the environment, and while bird watching, we must observe their migratory patterns, behavior, etc. And bird watching helps us initiate conservation efforts, connect with nature, and in educational opportunities. Sena Vihar has many trees and plants, approximately 1500 to 1700 trees and plants. Trees such as the banyan tree, the Indian cork tree, the gulmohar tree, etc. exist in Sena Vihar. Many of these birds reside in Sena Vihar, while many rest here during their flights. Residents have their own perception on birds, and most of the residents value birds because they are part of the ecosystem, while many find them annoying or problematic because they make a mess of their outdoor furniture, cars, etc. Urbanization has led to the decline of generalist species, the decline of specialist species and the dominance of generalist species. House crows and rock pigeons are quite common in Sena Vihar. The male and female koels show different behavioral traits. Many male and female birds, though they are of the same kind, show different features. Example, the male koel and the female koel, and the purple sunbird and the female purple sunbird. Most of the shy birds have very loud calls. Example, the white-cheeked barbet and the quail. Sena Vihar's green spaces contribute a lot to the presence of bird in this area. These were the apps I used, and I would really like to credit Anisha Jayadevan and Pooja Agrani for helping me identify the birds. Thank you.
Any questions to be asked?
Just curious, how did you conclude that 18% of them found it annoying and the 10% found it pleasant?
I conducted an online survey, so I automatically took my data and transferred it into an Excel, which made it into a pie chart for me. But the percentage was already given after the survey that I did.
Okay, what about the rest of them?
The rest of them, well, there were many responses, but I just took the main one out. So,
okay, thank you.
So according to you, what age category people, like now students are encouraged for bird watching programs and all. So according to you, what age category people can get more encouraged into bird? Like you can just mention primary students or high school students and all.
I would say high school students, but it is high school students, but most of us aren't interested in this. But I would say that if you can, it's a really interesting topic to go forward with.
One more question, like what was your survey method? Like you went to them and asked directly to a questionnaire, or you formed a Google Forms and did?
It was a Google form.
Okay.
Were there any migratory birds?
No, there weren't any migratory birds so far.
What is your view on cutting down trees? In your Sena Vihar, for example, imagine BBMP comes down and says, fine, we have to cut down a tree. It's very immediate. Has it happened before? Because
yes.
this thing will happen, right?
it has
Very
happened.
close to you then.
So about a decade ago, BBMP officials had made a ruckus in Senavihar saying that they wanted to cut down the trees. But since it's a military campus and retired officials stay there, they came together and fought with them, and they weren't allowed to cut any tree. Yes, they do trim trees, but there's no big fallings of trees.
Thank you.
Welcome.
Good afternoon to one and all present here. I am Deepika.
And I am Vaishnavi.
We both are from Standard 9 from Vagdev Villa School, Marathalli.
And for this year's Lake Conference 2024, we are doing the topic of water analysis of Panattur Lake. Lakes are an important resource for our ecosystem. In the present years, accumulation of greenhouse gases such as carbon dioxide and methane and other adverse human activities have affected the structure and stability of lakes. This pollution of water bodies has also led to extinction of species and a lot of ecological damage.
Especially in Bangalore, lakes play a very important role in serving agricultural and domestic needs. However, with the increasing population of the city have come many unplanned developmental activities which have led to the drastic reduction in the number of water bodies in Bangalore. The existing water bodies present in the city are contaminated by various industrial, agricultural, commercial waste and effluents. In order to restore the ecosystem, it is necessary to assess the level of pollution.
Therefore, today we will present a case study focusing on the assessment of water quality of Panattur Lake.
Our main objectives include analyzing the water quality of the lake and coming up with a report of the same.
We also want to come up with sustainable and cost-effective solutions to improve the water quality.
To identify the most immediate threats that are going to affect the lake.
We also want to create awareness about the importance of lakes on our ecosystem.
Now we will talk about our study area. Our study area is Panattur Lake, which is located in Kadubisnahalli, Panattur, Bengaluru 560103. Latitude and longitude are mentioned in the slide. We have measured the perimeter and area using Google Earth and its various features. The perimeter, as you can see, is about 1 kilometer and the area is about 41,000 square meter.
Also, we have collected some interesting facts about the Panattur Lake. The Panattur Lake used to have an island called the Panattur Island. This island was also used by Tipu Sultan in the 18th century during his battles against the British. This lake is also a home to a variety of amazing birds. Now we will in detail discuss the various materials and methods we have used. The basic materials we have used include test tubes, beakers, droppers, etc. To collect the sample, we have also used ladles, gloves, masks, etc.
Our methods include collecting various samples periodically from the month of June to the month of September, and we have taken the average of all the samples as 4 samples, and we have also used some of these samples for microbe identification, and a species identification method we have used microscopy.
To assess the water quality of Panattur Lake, we have run different chemical tests on it. These tests include on-site tests and off-site tests. Now, on-site tests include TDS, temperature, oil analysis, turbidity, DO, etc. The off-site tests include chloride, residual chlorine, ammonia, etc.
These are some pictures we have taken during our lab testing in our school R&D lab with the help of our teachers.
Also, we have observed the ecosystem nearby and have taken pictures of the various flora and fauna we have observed. The various flora we have observed include pink water lilies, jamun, badam, almond, etc. We have also observed birds such as the Indian spotted eagle, cormorant, etc.
We have listed all the species we have observed along with their kingdom, order, family, genus, species and their local names. Some of them are pink water lilies whose scientific name is Nymphia pubescens and whose local name is Kannai dile. We have also observed bougainvillea whose scientific name is Bougainvillea glabra and whose local name is Karubadani.
We have also observed the Indian spotted eagle, which is scientifically known as the Tlanga hastata, and its local name is Chukke giduga. We have also seen the Indian pond heron, which scientific name is Adiola grey, and its local name is Kolada baka.
In this slide, we have listed the various results for our on-site tests such as pH, TDS, temperature, odor, and we have also compared this with the permissible limits prescribed by WHO. And in this slide, we have listed the various results for our off-site tests such as ammonia, phosphorus, nitrate, etc. And here too we have compared the results with the permissible limits. And from this we concluded that all the samples have given results within the permissible limits for most of the tests except for ammonia and phosphorus, which were high and have been represented in red as you can see. Turbidity was also found to be high. The DO and the fluoride levels were found to be low and have been represented in yellow.
Also, we have represented the data we have got in form of two graphs. The first graph compares the permissible limit with the samples. From this graph, we can see that the variation between the four samples is almost negligible. The second graph compares the permissible limit with the sample average. From this graph also, we can see that the ammonia and phosphorus levels were high.
We have listed the various microbial species we have observed along with their names. These include Chlorophyceum such as Keratosmus corticauda and Spirogyra. We have also observed microorganisms called rotifers moving in the lake water. From this, we have concluded that a variety of microorganisms are present in the lake along with a large number of algal species.
Also, we have observed the live rotifers moving in the lake water and have taken videos of the same. Also during Ganesh Chaturthi, we had observed that there was a separate area for idol immersion. The people of the area also knew where to immerse the idols and did not pollute the lake. But we had also seen some ignorant people who had thrown the puja items and idols into the lake.
From our project, we have concluded that all the samples have shown the results within the permissible limits for most of the tests. Also, a comparison between all the samples showed that there was not much of a change in the lake over the week. And the DO levels are low and there has to be some action taken towards this. The presence of rotifers in the lake indicates controlled bacterial growth.
The lake is also home to a variety of amazing birds and insects. Many trees are also found in the periphery of the lake. Many fish are also inhabitants of the lake. All these are indicators of the good water quality of Panattur Lake, and therefore we have concluded that the lake has been well maintained. Also, strict action should be taken during festivals. Since we have observed that phosphorus and ammonia levels were high, there might be increased algal growth. Therefore, we have listed some methods to reduce the algal growth. These methods include addition of algae-eating fish such as Plecostomus and grass carp.
Another method is biological augmentation, in which microbial strains are introduced into the lake water that either directly or indirectly control the amount of algae in the water. And since we have observed that the DO levels are low, a method of increasing oxygen levels and reducing algae levels is aeration, in which the lake water is sent through pumps and sprayers periodically.
During our research, we had found out that there were about 9 fish kill incidents in the year of 2022, but situations have improved from then.
We have also collected a picture of the Panattur Lake from some years ago from a news magazine called Deccan Herald. You can see how polluted and degraded the condition of the lake was, but BBMP has taken appropriate action towards the lake, and you can see how clean and vibrant the ecosystem is now.
We have also listed the various references.
We thank IISC for providing us with such a wonderful opportunity. We would also like to thank our school, Vagdev Vilas Institutions.
We would also like to convey our heartfelt gratitude to our R&D department who have provided us with such an amazing learning platform.
Thank you.
Thank you.
Any questions to be asked?
Yeah, so I just need to clarify, how did you collect the water samples?
with the ladle, you mentioned that you use ladles.
Yeah.
So won't that affect your DO readings because the oxygen may escape?
No, for the DO we had like collected
Separate samples?
Yeah, separate samples.
Okay, thank you. Also, why low if the BBMP has put an effort to conserve?
Sorry, can you repeat?
Why are the readings low if BBMP has like taken measures to conserve the lake?
The BBMP has not taken measures to clean the lake, but you can see how the condition was. Now they put a fence around it, walking path is there, and we are not talking about the chemical cleaning, but the physical part of it.
Okay, thank you.
You mentioned few microbes found in the lake. Can you explain what does it signify except the rotifiers, and please justify?
Okay, so rotifers are microorganisms and come under zooplankton. So the reason their name is rotifers is because they have a crown of cilia around their head, and when they move in a wheel-like manner. And in Latin, the word rotifer means wheel bearer, and these help to control bacteria by eating the bacteria present, and also help to keep the lake clean by eating organic matter, and therefore recycle the organic matter also.
Okay, I have one doubt. Now, during Ganesh Visarjan time and all, people used to immerse those idols into the water bodies. So, what do you think? What other methods can be done? Like it should be balanced. That procession also should be done, but it should not harm to the water bodies also. What, according to you, think those can be done?
Yeah, and separate area can be maintained. Also, the idols, they are mainly sometimes made up of paints. They're painted, and the paints are usually not very eco-friendly, or else the normal clay is usually healthy for the lake's ecosystem, but the paints which are used are not very eco-friendly. Therefore, there can either be a separate area or paints should not be used.
What type of paints can be used according to you?
Paints are usually very not so good in nature, we can say. Chemical paints. So natural paints can be used, but I am not sure about that.
Thank you. Number 18.
Shall we start? Have you ever wondered what the prowlers on your walls are? Yes, they are geckos. They are a type of lizard. And today we are presenting to you about lizards. Namaste, I am Bhavya.
Myself, Mohit.
And we study in grade 9 Purna Pramati. The adaptability, appearance and the stealth of the lizard caught our attention in the vast diversity of animals found in Chitrashala Ashrama.
Next slide.
What happened that?
Chitrashala Ashrama, our study area, is in the eastern Ghats containing many variety of fauna and flora. So, introduction. Lizards. Lizards are animals that belong to the classification of reptiles. They are cold-blooded, scaly, and are also a close relative of snakes. Their appearance is simple. They have 4 legs, a long tail, a short neck, and a small head. They are found all over the world, but not in Antarctica, as it's hard to regulate its body temperature there. Its classification is this. It belongs to the kingdom Animalia, phylum Chordata, class Reptilia, order Squamata, and superorder Lepidosauria. It plays a huge role in maintaining the ecological balance as they are preys and predators to other animals. They control the insect and pest population. They are also pollinators, help in seed dispersal, and are also biological indicators. So this work is an attempt to present the discovered diversity, habits, and the role lizards play in the ecosystem.
Now, importantly, let us know about our study area, Chitrashila Ashrama, located in Chulagiri of Krishnagiri Taluk in Tamil Nadu state. So, as you can see here, the green area which you see, it is our range of study, while the yellow color represents the agriculture area, and especially the pink and red color areas, they are areas of resident and rocky terrain respectively, in which we found most of the lizards. And this is the address. And the Chitrashila Ashrama is a mix of dry deciduous forests and thorny scrubs spread across an area of 20 acres of land and elevated between 680 to 715 meters.
The method we use for observation is random sampling. So the total observation was done for 3 weeks from 1 to 1.5 hours daily. So the total observation time is more than 20 hours. So for collecting the data, we took photos and videos of lizards. For that, we use these devices. So these are the websites we used in order to identify the species.
Now, just to give you a glimpse of what of our findings in Chitrashila, we found 9 species of lizards belonging to 3 families, namely Geckonidea, Agamidae, and Skinkidae. So 3 in each family, as you can see in the chart. Now, let us begin with the family of agamids. So we have very similar looking Blandford's rock agama and South Indian rock agama. Especially in this, the South Indian rock agama is endemic to the peninsular India. And below them, you can see respectively the female species. And all the species which you are going to see in the upcoming slides are all least concerned in the IUCN Red List. And in every text box you can see the common name in English and below the vernacular name in Sanskrita. So we have a video.
Wait. Don't. Wait.
Yeah, there is audio, but it is not playing.
This is online video or saved video?
No, it is from the Google Drive. Okay, we'll continue, ma'am. No problem.
Okay, you can continue. Later we can.
Okay, sure.
It is not moving.
Another member of the Agamidae family is the Oriental garden lizard. It is very popular as you might have seen it everywhere. And moving on to the Geckonidae family, we have termite hill gecko, tropical house gecko, and the flat-tailed house gecko. You can observe the nocturnal eyes of them. And in the Skinkidae family, we have 3 members: the keeled Indian mabuya, the bronze mabuya, and the very unique common snake skink. And as we ventured through our study area, we observed many things about the lizards. And beginning with our observations, so as you saw, the peninsular rock agamas or the Blandford's rock agamas, mainly the male species were more vibrant. They tend to show off more, while the female species of the same were more shy. They were dull in color and camouflaged. And the lizards, we found the lizards and skinks during the day, while the geckos were more active during the night. And even though the natural habitat of the lizards was forest areas and rocky terrains, the geckos especially were adapted more to the urbanization than any other lizards, and only with the vocal sounds. And many lizards could change color at their will, and the skinks and agamas were the only terrestrial lizards, while the others were arboreal.
So we have classified the features based on physical and habitual. Physical based on their appearance and habitual based on their behavior. So as you can see, these are the basic physical features of a gecko. In habitual features, we have mainly focused on their defense mechanism and basking. So in defense mechanism, we can see the female, you can see the female rock agama tends to blend in with its surrounding camouflaging. We can also see autotomy, which means the lizard detaches its tail at its own will in order to escape from the predator. We can also see lizards making sound and appear aggressive in order to scare the predators. So basking. Basking is a comfort habit of lizards. It is also called as sunbathing. It is usually done during the mornings and the evenings, as the temperature is neither too high nor too low. While after basking, we can also sometimes see them do push-ups. They do an upward and downward movement called the lizard's push-up. This lizard push-up is usually done to communicate with other species and attract the mates. So to recommend, what I would suggest is, due to habitat loss and human intervention, slowly the population of lizards are decreasing. So I would suggest that we would not interfere with the lizards and not be afraid of them, as most of them are harmless. I would also recommend to do a longer study, as the time was short. We could not do a deep study, and we should do a long and extensive research.
Now to conclude, we found 9 species of lizards belonging to 5 genera of 3 different families in the area of Chitrashila Ashrama. From this kind of diversity, we can know that this geographically important area has many types of prey and also habitats. And also, from this healthy environment, we can still say that there is more potential to find a lot of lizard diversity.
So these are the websites we used for reference. We would like to thank all the parents and guardians who helped us out. We would especially thank Dr. Sridhar Subarna for giving us this opportunity, and Sudhir Anna for lending us his camera. I would also thank my friends of Bala Avagamana 2024 and also Pune Pramati School for encouraging us. I would also thank Prajula Lanna and team for guiding us. Thank you.
Sorry? Yeah. Like if there are lizards, so we also said that it plays an important role in the ecological balance. So if you see more lizards or lizards in a suitable in a particular habitat, it shows that the ecosystem is balanced. So it is also a biological indicator. Like if you see lizards, we can say that it is a healthy environment. So that's what we mean by biological indicator.
Geckos are a type of a lizard, and also you can also see when they're active. As you can see, most lizards, for example skinks and oriental garden lizards, are active during the day and they're diurnal, whereas geckos are nocturnal. They're active during the night.
Geckos are very similar to lizards, or they have some differences?
So as you said, geckos are a type of lizards. It comes under the Geckonidae family. So why we consider they consider the geckos as separate, because it has very unique features which we commonly don't see in the other lizards. So if you see the 1 minute. So if you see in this picture, this lizard has claws. Can you observe that? Yeah. But if you go to the geckos, you can see it has flat tails. So they are adapted to the urbanization, as you said. So they are considered differently.
And also, geckos are nocturnal, so they have nocturnal eyes.
Yeah, one more question. How can lizards be pollinators?
See, for example, oriental garden lizards and all, they live on trees and other plants. They're found commonly on trees and other plants. So therefore, when they climb on flowers, the pollens get stuck and they can also be pollinators.
The lizards have various types of diets. Like not only insects they eat, but also kind of flowers and leaves they eat. So when they are in their diet, so you can see that the pollens of the flowers stick on their feet, and as they move around, it will get pollinated.
Usually they will excuse me, the tree and root of the house, no? How can they pollinate?
Like not all lizards we are talking about. Like the area in which we conducted our lizards are eastern Ghats. So in these places you might not find, but some lizards are also like pollinators. Like not only they are in the rocks or on top of your roofs, they are also terrestrial. So as we said about rock agamas, they are terrestrial lizards. Hope I answered.
Most of the lizards are harmful to humans, right? That makes some of them harmful to us. How are they harmful?
They are not harmful. Like
We did say that it is harmful. They said in one slide.
when we say most, we are referring what we see around. Generally, they are geckos. Thank you.
You have dealt with lizards. Generally.
Thank you.
Now we have to 20 presentations are there.
20 more are there. Only one question.
Yeah.
Especially from students. Relevant questions.
Seven.
Good afternoon to one and all present here. I am Anvita.
I am Sanvi. We're from Bhagdevi Villa School, Marathalli, and our topic for Lake 2024 is plants and trees.
Plants and trees are essential in our environment as they provide us oxygen. They help in conservation of water and soil. They provide shelter to many animals and birds. They also make our environment beautiful. But these are only the basic properties of plants and trees. Plants and trees have many more properties like neem tree, which is used for antibacterial and antifungal properties, and also jamun tree, which is used for treating diabetes. But most of us do not know these properties, and that is why we took up this project to expand our knowledge about plants and trees.
The main objective of our project is to find and share enough information to find possible outcomes for tree conservation. So we decided to start small, and first what we did is we printed out numbers and then we laminated them and numbered all the trees in our school campus. Then we identified all the trees. While identifying, we found the properties, so they would help us differentiate between different species under the same genus. Then we made QR codes for the trees. Once a QR code is scanned, it will produce an image with all the information related to that particular tree.
The study area of our project is our school, Bhagdevi Villa School, Marathalli, which is 560037, and the area of our school is around 8 acres and the perimeter is 900 meters. In this area, we have approximately around 450 trees and 77 different species.
This was us numbering trees. So for trees with thicker gourds, we use bigger nails. For trees with smaller gourds, we use thinner nails. And for trees which are more like vines, like bougainvillea, we use tags as to not hurt the tree. These are the examples of the QR code we made. Once they're scanned, they lead to images like this. For example, here we have Curupita gianensis, which is known as cannonball tree or Nagalinga pushpa. It is called so because it has very hard fruits. This tree produces very fragrant flowers, unlike the extremely bitter smelling fruits, which is why they have never been used for human consumption but have been fed to livestock. Some parts of this tree have medicinal properties such as healing, toothache, malaria, etc.
We use several methods to identify the species of the plant. First, we use Google Lens, where we had to take a picture of the plant and it gave the information about the plant. We also cross-check this with different websites like identifythatplant.com, etc. We also face a problem differentiating between water apple tree and mango tree because the leaves of water apple tree are very similar to those of mango tree, but we differentiated it as water apple tree because it did not have the significant smell of the mango tree, and also the branches were arranged lower in the trunk, unlike mango tree where the branches are usually arranged higher. So this way we use our senses and check the shape of the leaves, how the leaves were arranged in the branch, how the branches were arranged in the tree, the trunk of the tree, different flowers and fruits blooming on the tree, etc. This is the tabulation form of our results. So out of approximately 450 trees, the maximum number of trees in one species is of coconut tree, which is 56 trees. Next is Indian cork tree, which is 49 trees, and the third one is ficus tree, which is 34 trees. We also found 3 rush species in our campus, which is Rudraksha tree, Queensland curry, and cannonball tree.
This is the graphical form of all the families of trees we found in our school campus. So the dominating family in our school campus is the family Bignoniaceae, under which we found 86 trees of this species: Melantonia hortensis, Tecoma stans, Jacaranda mimosafolias, Spathodia campanulata, and Callistemon viminalis, along with 3 species under the genus Tababia, Tababia heterophylla, Tababia argentea, and Tababia rosea. The second dominating is the family Ericaceae, under which we have 65 trees of this species: Cocos lucifera, Erica catechu, and Licuala grandis. The third dominating is Moraceae. Under Moraceae, we have 51 trees of the species Ficus bengalensis, Ficus religiosa, Ficus carica, Ficus elastica, and Ficus benjamina, and also Artocarpus heterophyllus.
We also found families under which only one tree was present, so we classified it as others. So in others, we have nine families, which is Iliocarpaceae, under which we have the species Rudraksha, Ananaceae, under which we have the species Ramphal, Bixaceae, under which we have the species Lipstick tree, Lamiaceae, under which we have the species Sindhuka tree, Proteaceae, under which we have the species Silver oak tree, Lauriaceae, under which we have the species Avocado, Magoliaceae, under which we have the species Champak tree, and Cimarovaceae, under which we have the species Biodiesel tea, and Sapindaceae, under which we have the species Lychee.
study at our school, we classified the trees into 5 different categories, them being fruit-bearing trees like coconut, mango, bear, jamun, etc. Rare trees, we found cannonball tree, rudraksha and Queensland curry. Ornamental trees, we found pink tababia, cherry blossoms, bougainvillea, chenille plant, etc. Medicinal trees, we found neem, gooseberry, amla, bear, etc. And timber and shade trees, we found mahogany and kadamba, etc.
These are a few of the pictures of the trees found in our school.
The tree identification project at Bharatiya Villa School has deepened our understanding and increased our knowledge about trees. By creating QR codes and finding detailed information, we have taken steps towards the conservation of trees. This initiative will also encourage active participation in the care and maintenance of the health of our trees, as well as promote a sense of responsibility for taking care of the nature for future.
Form, and it can be submitted in form of text and image, and once it's submitted it will go to assigned ID for action, and this will help in preservation of trees. Other suggestions are educational workshops, tree adoption program, nature walk for students, and guided tours for parents. Like during PTMs, our school can conduct guided tours for parents by students, so to tell them about the benefits of trees.
These are the references we used. We would like to thank IISC for providing such a wonderful opportunity, and as well as thank everybody who helped in between with their suggestions.
And we would also like to extend our gratitude to the Research and Development Department of our school, Bhakti Villa School, Marthali, for helping us and guiding us all the way here. Thank you.
Thank you.
Any questions? Any questions to be asked?
How did you create the QR code?
We use an app called MiniQR, so that we had to just tell them what kind of QR code we wanted. We pasted the image that we created and the QR code was ready.
Thank you. Where do you find cherry blossoms in Bangalore?
Stop.
We find cherry blossoms in Bangalore. This is Tabelia rosea. So like it's cherry blossoms, and we confirmed it by using different methods. In our school campus.
The study is done in the campus.
The campus study.
Yeah, nested. Why this is coming like that? Why this is coming like that?
Good afternoon to one and all present here. Myself Ritushri. My name is Anmita ji.
And my name is Janviar.
Today we are going to present our research on Ulla Lake.
A lake is a large, still body of water surrounded by land on all three sides. It is vital because it provides fresh water, supports diverse wildlife, and helps to maintain local climate. Bioindicators are organisms that signal changes in environmental conditions.
So we have two main objectives for this study. The first is to analyze various parameters related to water in Ulla Lake, and the second is to investigate the relationship between total hardness and microbial life in Ulla Lake. So Ulla Lake is situated in Ullal village, a southwestern part of Bangalore. It falls in the Vrishabhavati valley. It covers area about 11.03 hectares and perimeter approximately 1870 meters. This is the comparative study of Ulla Lake from 2022 and 24. We have collected the samples from two sites, and the area, perimeter and location are given below respectively. So in order to check chemical parameters, we have used this testing kit provided by our schools, which include total hardness, alkalinity, dissolved oxygen, pH, calcium, residual chlorine, chloride and fluoride testing kits. So we have collected the samples near the inlet of Ulla Lake. We have tested various physical parameters and chemical parameters. We have also checked, we have also identified specific microbes using a compound microscope. Moving on to total hardness, it refers to the concentration of magnesium and calcium salts in the water. It maintains the stability of aquatic environment.
We have done several tests using the testing kit provided. So we have done the total hardness test, the calcium test, the magnesium test, the fluoride test, the chloride test, the residual chlorine test, the dissolved oxygen test. So here are the results. So the test 1 was done on 27th of July 2024 and test 2 was on 1st of September 2024. So here are the results. Now here is a comparison between test 2022 and test 2024. So we have done these tests based upon the chemical parameters, which includes the pH, which is 9 and 8, total hardness 150 mg per liter and 140 mg per liter, alkalinity is 253 mg per liter and 205 mg per liter, and dissolved oxygen is 7 mg per liter and 6.63 mg per liter. So when we observe test 2024, we can see that it is reduced when compared to test 2022. There are several factors which includes pollution, which may be runoff of sewage, water or industrial waste into the lake, or natural process like seasonal changes or shifts in weather patterns. So then even the increase in nutrient levels can lower the pH and cause more pollution. Now let us move on to microbes. So microbes are vital to the lake ecosystem. They are tiny organisms which influence the water quality. They are very important because they help in nutrient cycling and they maintain the oxygen levels. So understanding their role is very important for the protection and management of the lake. We have found out several microbes which include paramecium. So they are generally found in environments with adequate oxygen. They feed on microorganisms such as bacteria, algae and yeast. They come under the class Oligohymenophobia and they come under the kingdom Protista. Moving on, we found out Euplautus. They are typically found in freshwater and marine environments, often found in sediments or attached surfaces. They feed on bacteria, small algae and other microorganisms. They come under the phylum Coleophora. We also found out Tetrahymena, which are commonly found in freshwater environments such as ponds, and they feed on bacteria, algae and other organic matter, and they come under the class Coleophora. We found out Chlorococcum, which are generally found in freshwater and soil, and they contribute in oxygen production and they help to maintain the nutrient cycling. So they come under the family Chlorococcaceae, phylum Chlorophyta and class Chlorophyceae.
Next, we found out nematodes. They are found in diverse environments including freshwater, marine and terrestrial ecosystems. They feed on bacteria, fungi and other microorganisms. They play a crucial role in nutrient cycling. They also help in controlling pest populations. They come under the phylum Nematoda and class Caesalentiae. Rotifers, they are found in lakes, ponds and freshwater environments. They feed on dead and decomposing matter and as well as unicellular algae. Classification, phylum Rotifera. Next, we found out Oscillatoria. They are found in shallow waters of lakes and slow flowing rivers.
They synthesize their own food by photosynthesis. And classification, cyanobacteria. Anacystis are found on the surfaces of algae and plants. They rely on photosynthesis for its food. They provide insights for nutrient dynamics and classification, phylum Cyanobacteria. These are the microorganisms which we found in our sample. The paramecium found in plants. By this we can say that nematodes prefer hard water with high calcium levels. Rotifers and euplotus, they are found in the marine and freshwater environments, and they prefer areas rich in organic matter and microorganisms. Paramecium tolerates various hardness but might be stressed by extremes. They grow well, Oscillatoria grows well in hard water and high calcium and magnesium levels. Anacystis prefer harder water with high calcium and magnesium levels, influencing its growth. Tetrahymena tolerates various water hardness levels but prefers moderate conditions. Conclusion: microorganisms such as nematode and other algae serve as bioindicators for the pollution of lake. Their presence in high numbers can signal imbalance or contamination, which is often linked to ecological issues such as eutrophication. According to the BIS standard, the water cannot be used for drinking and domestic purpose. These are the references from where we found our information.
We would like to thank the Lake Symposium team and Dr. TVRAMACHANDRA sir for this wonderful opportunity. We would also like to thank our school, Vidyaniketan Public School, for giving us such a great opportunity and continuous support.
We would also like to thank our mentors and lab assistants for their encouragement and support. We would also like to thank our parents for your support in this project. Thank you.
Any questions? Only one relevant question.
How did you correct the year 22's water?
This was from the IISC site, so we found this was the previous Lake 2022 Lake Conference PPT.
This is forward, backward, downward, forward.
Good afternoon, one and all present here. Myself Munisha with my team members Mitra Shri and Navya Shri from Vidyanchali Primary and High School. I thank one and all present here to giving me an opportunity to present my study, The Insect World of Shiva Temple Hill Weavers Colony. The study of insect diversity and distribution is essential for understanding the ecological balance within any habitat. Insects play a crucial role in pollination, decomposition and serve as a food source for other wildlife. The semi-setting of urban weavers colony around the Shiva temple emphasizes the human activity and natural habitat which may impact the local insects population. The temple area is an ideal location for exploring insect diversity and distribution. Our objective is to identify and document the different insect species at Shiva temple, to analyze the distribution and abundance of insect species in different parts of temple ground, to conduct awareness about the importance of insects. Our study area is Shiva Temple Hill Weavers Colony, Gottigere, Bangalore South. This is our map and this is our GPS coordinates. The temple grounds includes open space, flowering plants, small garden patches, rocks and grasslands.
These green spaces, though modest in size, attracts a varieties of insect species. The area also hosts other wildlife including snakes, birds, mongoose, garden lizards, squirrels, etc. Materials we use it to observe the documentation of insect species are insects identification, referring, iNaturalist, notebook and pen, smartphone for photograph, data sheet for recording observation. Weekly visits were conducted on Sundays from 8 am to 10 am. The study area was divided into two zones based on their habitat types. Insects were identified and data was recorded in field notebook and photos were taken through smartphones.
These are the checklist of insect species identified at Shiva temple in June, July, August and September. One of the common name is Faren. Scientific name is Delas sonelopsis. Family name Formicidae. Dwarf honeybee Apis floreapidae. These are the gallery of insects found at Shiva Temple Hill. Tiger mosquito, long-legged greenfly, dwarf honeybee, leaf hopper and ladybug.
Totally 56 insect species were observed around 2 habitat zones. Ants were among the most prevalent insect species observed. Species such as fire ant, soldier ant, weaver ant and giant ant show higher abundance in zone 2 rocky area. Various flies and beetles were well represented. Notable species including jewel beetle, dung beetle and darkling beetles. Flies such as blue butterfly and black horse fly were represented in both the zones, possibly due to the availability of decomposing organic matter in the area. Order Lepidoptera species like common castor butterfly and yellow peach moth were recorded in both the zones with varying abundance. These species are crucial for pollination, indicating the habitat health of the ecological health. The presence of specialized species like carpenter bee and garden bumblebee highlights their role in the diversity. Though low in number, however, the data suggests the need of conservation efforts of this vital species. Garden area is characterized by flowering plants and small garden patches, supported a wide range of insect species. The higher abundance of species such as ladybug and long-legged greenfly indicating that this area provides favorable condition of the area. The presence of various ants and beetles suggests well-balanced ecosystem. Rocky area showed a higher concentration of ants and flies. Species like red soldier ant and giant ant were abundant, indicating pollinators emphasizes the importance of preserving such space of ecosystem health. Some insects like carpenter bee and garden bumblebee play specialized roles in pollination. They through a fewer in number. It is very important to protect their habitat to maintain the overall health ecosystem. Ants, particularly in the rocky areas, are abundant and play an important role in breaking down of organic matter and spreading seeds, which help in nutrition cycle and maintain soil health.
People who have visited temple, we created awareness about the importance of insects.
The study documented a diverse range of 56 insect species at Shiva Temple Hill. This highlights its ecological role in the importance of urban green space. Conservation efforts are needed to protect in both the garden area and rocky area. The presence of many pollinators supports rich plant diversity in the garden area. The rocky area provides a unique habitat for a various ants and flies. We thank our HM and guide teacher for allowing us to conduct the study, and we thank Sri Rijulal sir and Sri Chaturved for guiding us. Our gratitude extends to the scientists, field researchers and experts of Energy Wetland Research Group Center.
29 अभी this one you just check, okay? This here
This is forward, backward, and this is pointer.
Good evening, my name is Shrina Shetty from grade 9 of Vagdevila School, Mathahalli, and today I am going to be presenting a study and assessment of the Kundanhalli Lake. The Kundanhalli Lake is a lake located in the northeastern suburb of Kundanhalli in Bengaluru city. Historically, it has been used as a dump yard for untreated sewage. It is spread across 10.48 hectares and of recent times, wastewater treatment technology has helped the lake regenerate. Objectives. There were 7 objectives that were pursued throughout the project. These were to study and assess the water quality, to compare the water quality with desired standard qualities, to compare these water qualities with qualities in the past, to observe macrophytes, trees and birds in the immediate surrounding of the lake, to assess soil quality of the surrounding areas of the lake, to infer the soil qualities and compare it to the past qualities, and finally to find ways to purify lake water. Study area. This is the satellite view image of the Kundanhalli Lake. The study area is between 2 points within a 100-meter distance in the lake. Materials and methods. Lake water sample analyzation. Excluding pH, there were 6 main chemical parameters that were tested. These were fluoride, residual chlorine, phosphorus, ammonia, nitrate, and finally fluorine. These were the observational images and these will be further discussed. Soil analyzation. The 4 main tests that were taken for soil analyzation, these were pH, nitrogen, phosphorus and potassium through the respective reagents 1 and 2 and decolorizer or decolorizing agent. These were the observational images. Again, these will be further discussed. Moving on to the species identification of trees, we can find a variety of different trees in the Kundanhalli Lake. We are going to discuss the most commonly found and the least commonly found type of trees here. The most commonly found tree was the mountain ebony in a count of 23. Next is the elephant ear tree in a count of 18. And finally the mango tree and the gulmohar tree, both in a count of 13. The least commonly found trees were as follows. The Indian laburnum was present in a count of 5. The cudleaf tree was also present in a count of 5. And finally the least commonly found tree was present in a count of 2, which was the Indian elm. Moving on to the species identification for macrophytes. Macrophytes are aquatic plants that are found in and around the perimeter of the lake. These macrophytes have several biological importances and are of 3 types, that is, of submergent, emergent and floating macrophytes. The 3 predominant macrophytes that were present were the water lettuce, bourgoon and the jointed flax edge. The water lettuce's roots and leaves have been found to absorb harsh chemicals and heavy metals such as zinc, cadmium and chromium and are used for declining in eutrophic conditions. Bourgoon is important for the reclamation of alkaline and sodic soils or those soils which have higher sodium content in it, and it is also important for fish diversity as it provides good breeding and feeding habitats for the fish. Lastly is the jointed flax edge, and the jointed flax edge stabilizes the roots, stabilizes the soil along the banks due to its long roots, and it also provides streamside habitats for invertebrates. Species identification for birds. The 4 most commonly found birds were the Asian quail, the pigeon, the Indian tree pie, and the white-throated or the white-breasted kingfisher. Moving on to the results and discussion, we are going to discuss the water analysis. 2 samples were taken for this and they are going to be compared to the desired standard limit. All of sample 2's chemical parameters fell within the desired standard limit. However, we have observed that in sample 1, phosphorus and ammonia exceeded the desired standard limits. To compare it with 2015 as per a reliable case study, we can show that the qualities as follows. With a pH of 7.4, fluoride of 0.7 ppm, residual chlorine of 0.7 ppm, phosphorus and ammonia of 1 mg per liter, chlorides of 264 mg per liter, and finally nitrate of 0.54 mg per liter. Graphical representation of resultant values. We can see that the blue graph is represented by the 2024 values and the red graph represents the 2015 values. We can also infer from this that 2024 values are better than the 2015 values. And in the first graph, it is supposed to be y-axis. Inference through government interference, especially BBMP activities, we can see that the lake has been restored, but there are still imbalances in the qualities of the water, and more macrophytic and vegetative cover can be seen in and around the lake due to the lake water being better suited for vegetation. Results of soil analysis. The results of soil analysis came out as follows, with a pH of 7.5 resulting in slightly alkaline soil. Slightly alkaline soil is suitable for most crops and plants. Nitrogen, phosphorus and potassium, also known as NPK, which are the main macronutrients in the soil, came out as follows with the results of medium, low and high respectively. To compare, we are going to use a soil analysis in 2016 with the pH of 8.5 resulting in strongly alkaline soil. Strongly alkaline soil is not suitable for a lot of crops and plants. Nitrogen with low, phosphorus with low and potassium with high. We can infer from this that the levels of fertility have improved from the past. However, there are still nutrient imbalances in the quality of the soil, and there can be additional use of fertilizers to make the soil suitable for all types of crops. Recommendations. There are a few recommendations for the crops, for the lake. That is the regulation of wastewater that goes into the lake. Even though there has been measures to make sure only pure wastewater goes into the lake, the slums and factories that surround the lake still send wastewater into the lake. Letting only treated sewage into the lake, factories still send wastewater into the lake, and also there is immersion of Ganesha idols, especially during puja time. Planting more native macrophytic species to regulate the health of the lake, and it's native, so it does not disrupt the ecological balance. And planting more trees as to attract migratory bird species. And finally, bioremediation, which is a biotechnical process in which strains of bacteria and algae are introduced, which naturally help restore the lake. Conclusion. Lake water gets contaminated due to various human activities such as dumping of hazardous waste. To maintain the lake's health and of the biodiversity surrounding it, human activities must be monitored. Restoration of this lake can be done through both natural and mechanical processes as seen before. From the past, the lake's health has improved. However, there is still dumping of hazardous waste and there are still problems which require solutions. There are a variety of both native and exotic plants and migratory birds present during the migration season. These are the major references that are used in the project, which include IISC, agro-scientific groups and government websites. Thank you.
Can you give the one example of the floating macrophyte?
Floating macrophytes in this lake were not extremely prevalent. So these macrophytes were present here. Water lettuce can be called submergent, can be called emergent because the roots are still present under this water, but there are no floating macrophytes here. They are present around the lake surface.
Okay, thank you. Nice presentation.
Thank you.
Okay.
This is forward, backward and long press focus.
Okay.
Good morning everybody, my name is Ishan, I'm studying grade 9 in Automata International School, and today I'm going to be talking about the bird diversity in Ulcer Lake. Ulcer Lake is one of the largest lakes in central Bangalore. It spans over an area of 121 acres and is the home to a variety of birds, insects, and reptiles. It also plays a vital role in reducing the urban heat island effect and also providing feeding and nesting sites for different bird species. Birds also depend on the food sources surrounding the lake for their survival. I've lived my entire life overlooking the lake and have coupled my personal observations with the below. I've used the data collected by a citizens participatory project which has identified all the bird species spotted around Ulcer Lake. Furthermore, I've gone to Ulcer Lake and taken pictures of a couple of bird species. I've analyzed the different categories of birds and how the lake ecosystem affects their population. Birds are an integral part of the ecosystem. They play a vital role in pest control and seed dispersal. They also influence the dynamics of the ecosystem with their various interactions with other organisms. Birds are often known as indicator species. By monitoring the bird diversity of an area, we can find out the state of the ecosystem. A change in the migratory patterns can indicate a change in the climate or change in the ecosystem. The adaptability and response of birds to a change in the climate or habitat conditions can influence the overall ecosystem health. Birds can be classified into 6 types. Aquatic birds are those birds which feed on mainly fish, like the cormorant. Insectivores are those which feed on insects, like the swallows. Frugivores feed mainly on fruits and nuts, like the starlings. Birds which feed on smaller animals are known as raptors, like the kites. Waders live in the shallower parts of water bodies and feed on aquatic plants and animals, such as the egret. Generalists are not picky on what they eat or where they live, like the myna. Birds can also be classified into resident or visitory birds. Resident birds are those birds which reside in a given area. Visitor birds are those birds which migrate to a different area for feeding or nesting purposes. These are some of the bird species spotted in Ulcer Lake. The Asian coral and the parakeet are frugivores, whereas the Brahminy kite and the black kite are raptors. The greater curcle and the common myna are generalists, and the double-crested cormorant are aquatic, whereas the black-headed ibis, white-breasted waterhen, and Indian pond heron are all waders. Over 111 distinct species have been spotted around Ulcer Lake, and this is a graphical representation of these species. As you can see, that the percentage of waders is the highest. This is because many insects such as dragonflies, mosquitoes, caterpillars, and grasshoppers all reside around the lake. Also, a part of the lake has not been paved, facilitating the survival of these insects. Waders are the next highest. Waders live in the shallow parts of water bodies and mainly depend on aquatic plants. Now, the percentage of nitrate in organic matter has increased over the past few years due to the stormwater drain which flows right next to Ulcer Lake. This has resulted in the increase of the population of algae, which has also resulted in the increase of the population of waders since algae is their main food source. The presence of frugivores is positively linked to the availability of fruits and nuts. Trees like false ashoka, African tulip tree, and copper pod are all present around the Ulcer Lake, providing a sufficient food source for these frugivores. Aquatic birds play a key role in the lake ecosystem as predators, herbivores, and distributors of fruits and nutrients. The low percentage of aquatic birds may be due to the close proximity to the city or because of the military using the lake for their water-based activities. I do have noticed that the percentage of cormorants have been increasing. This may be because recent efforts have been taken up to produce more habitats, or because cormorants are piscivores. They eat fish. So the increase in their population would mean an increase in the population of fish in Ulcer Lake. Generalists are the most tolerant urban birds, and they are not picky on where they live. Now, I've noticed that the population of pigeons is lower in the Ulcer Lake than in urban cities. This is because even though pigeons are highly adaptable, they prefer a built-up, sheltered environment. The lack of these areas and the predator cover in the lake makes the lake less suitable for these pigeons. Next, the raptors. The percentage of raptors show favorable habitat edge and also easy access to prey. Also, during the night or in the evenings, you can see about 20 to 30 raptors just flying around in the middle island. migratory birds. Now, around 11% of the total bird species are migratory birds. They prefer a diverse environment, less water pollution, and least human interference. Now, the high percent of migratory birds may be because of the diverse habitats which the lake provides. Recent efforts have also been taken up to provide more ecosystems or habitats. The water level in Also Lake has also receded, exposing mud flaps which are rich in nutrients. And also Also Lake provides many more food sources for these birds. I do believe that the population of migratory birds has reduced over the past few years. This may be because in the pandemic I saw a group black-headed ibis nest in one of the 4 inner islands. Now they stayed there for about 4 to 5 months and then left. Since then, I've not seen them stay in the island for such a long period of time. So even though the pandemic was such a hard time for us, it was actually a safe haven for the birds. Now, from this, I've concluded that even though Also Lake is in the middle of Bangalore, it still provides a vibrant and diverse ecosystem for the birds. However, the reduction in the population of migratory birds is still a huge concern. Some of the efforts which can be taken up to maintain or at least improve the habitats in Also Lake can be sewage management, control of invasive species, creating more nesting sites, adding more floating vegetation, people awareness and involvement. These are the different sites which I have referenced. And I've also made a list of all the bird species spotted in Also Lake. Thank you.
Hi, nice presentation. Can you please tell me the presence of cormorant? Bird is there. Then what we can indicate?
No, because the high percentage, because government officials have created more habitats for these cormorant birds on the banks of Also Lake.
The presence of fish or something?
Yeah, no, also that is one of the reasons, but the other reason also because the population of fish also may be increasing because cormorants' main food source are fishes.
Okay, thank you.
Thank you.
Now onwards, who all are coming here for the presentation, we are not asking any question. We will give mark for them because you all are making the noise. Now how many people wants to do the presentation? You are become silent, then only we will ask the question. You are making the noise, then we will give these people will get the mark.
5 minutes, keep it.
5 minutes.
Those who are using mobile will pick the mobile and will not return it. If you are creating any noise here, your presentation and your certificate, your marks will be withheld. Seriously, I am telling, because you should allow others to present it. If your turn is over, it doesn't mean that you should make noise there and make others inconvenient.
Okay, students, you can have a tea break here only, where morning you had.
PDF.
Where is that volunteer? Disappears. He's supposed to bring the kids back now.
It's okay. Whatever I know that. We can start.
Close the door, front door.
Now you can take video.
Forward, sorry, forward, backward. This is.
Yes, forward is
This is forward.
Tea. That is better milk.
That's why I wanted.
It's okay. We can start.
You should tell if you make noise, we will give 10 to all the presenters and we will cancel all the previous presenters' marks. Not that we will not ask questions, then they will make more noise so that they don't want any questions.
Good evening, respected jury. I am Nayantara Menon of Grade 9, Presidency School, Bangalore South. Today, I will present my research and project, which has spanned about a year and a half on two interrelated topics. The inability of urban children to recognize local flora, especially if they have been geographically mobile, and non-native species often disrupt ecosystems by outcompeting native species, thereby altering habitats and spreading diseases. In my opinion, these two topics are closely intertwined. If today's children are to become effective change makers, they must be made aware of what is present in their natural environment and how they affect the surroundings. My own project started with my inability to recognize basic trees even when I was about 8 or 9 years old. A complete lack of interest in nature was changed overnight when our school Eco Club organized a 3-day study tour to the Western Ghats last year. Being immersed in nature day and night was truly a life-changing experience. India is a country rich in diverse flora, and learning to understand and recognize this diversity can lead us to healthier, simpler lives as our ancestors led. So as I returned from the trip, I wondered whether other children face the same problem. I wanted to integrate technology with ecology and make identifying trees easier. So I began a survey to understand just how common this problem was. I discussed my idea with my school Eco Club mentor and teacher, and they supported my idea of the survey, which revealed fascinating results. The objective of my study was to conduct a survey to analyze flora literacy among different age groups. It was to foster curiosity about the environment, nurturing eco-conscious explorers and conservationists by creating an app to identify flora by making use of prevalent technology, and to understand, using derivatives of the app, the impact of non-native species on an ecosystem. The study area of my choice was the campus of my school, Presidency School, Bangalore South. And through spatial mapping, we can see here, the red region denotes the complete area of our school, and the purple region denotes the concrete cover. So the remaining red area is the green cover of our school. My campus is pretty diverse in terms of the number and variety of trees present. So to create a pilot app, I decided to take photos of 4 random trees from all angles and train my AI model to recognize those trees. The species of choice were African tulip, mahogany, Hong Kong orchid, and flame of the forest. My sample collection method included photographing trees on campus, conducting a survey, tabulating the number, species, and other basic data on trees, as well as going through secondary and tertiary research data. My species identification method included creating an AI app to recognize species with the pictures taken by me from my school campus and at home, and the help of my teacher was also used. The experimental method used for my project included coding to create an AI-based app using MIT App Inventor and Google's Teachable Machine, which uses TensorFlow.js to run models and transfer learning to analyze models. We used pen and tape to measure height, hand method to measure girth, and use of the Clinometer camera app for the height and distance calculator was also used. Going back to my survey, the population consisted of 2 class groups, grades 1 to 4 and grades 5 to 12. Along with the questionnaire, there included pictures of 5 common trees, neem, guava, peepal, teak and gulmohar, along with the pictures of their leaves. The same questionnaire was circulated separately to the 2 class groups, and this was done to understand how students learn from the environment as they grow. The survey revealed that almost half of the students, that's almost 100 out of the 200 responses received, could not identify the trees correctly. While neem was the most recognized, the identification accuracy of the remaining trees continued to decrease. This showed that that age does not play a decisive role, such a decisive role in the identification ability of students. Surprisingly, some students thought that guava was an apple tree and peepal was just a puja tree. Now, I don't deny that the peepal has certain religious connotations in our Hindu culture, but that shouldn't be the identifying factor of the tree alone. This survey highlighted a gap in our knowledge, which became the foundation of my app EcoLens, which aims to help students identify trees on our campus. I used my knowledge of coding and AI to create this app. So let me now take you through it. So this is the splash screen of my app EcoLens, and with the logo and the progress bar to indicate the loading of the app. It opens up into the main menu page, which currently has 5 major features: EcoLens, EcoLog, EcoRec, EcoLog and EcoGallery. EcoLens is the main feature of my app. You must first take a picture of the plant or tree that you want to recognize and click on the Classify Tree button. The AI then scans your picture and displays the name of the tree which it thinks is closest or is the exact name of the picture you have taken. As you can see, this model is able to recognize the African tulip tree with 96% accuracy. Next to the name of the tree, there is a small I button present, and when you click on it, it opens up into a tree identity page, which gives you basic information about the tree, such as the scientific name, medicinal properties, flowering, fruiting, and whether it's a native or non-native tree. Here is a sample training data set that was used for the African tulip tree. As you can see, the data set has got a diverse variety of pictures and important pictures such as the flowering, fruiting, radiation, etc. And this was due to paucity for space. 40 pictures were used per plant and 10 pictures were used for the training. The next feature is EcoLog, which allows you to enter names of the trees that you may see on campus or while on a nature expedition. This can be used by teachers to give marks at the end of the year, and is also a great way for students. It acts as incentive for students to be among nature and for schools to integrate it into existing curricula. The next feature is EcoGallery, which acts as proof that students have indeed seen so many trees. You can take pictures of the trees that you see and save it directly to the app without taking any storage from the phone's gallery. This is the EcoRec, which allows students to take information from ChatGPT and Google regarding any basic doubts they may have, say regarding the scientific name of the tree. But since ChatGPT can make mistakes, it's imperative that information be verified. Finally, we have EcoLog, which allows users to add new databases other than the school to continue expanding database of identification. As you can see here, I have added all the schools of Presidency group of institutions by clicking on the plus new add new area at the bottom of the screen. Now, while exploring, I came across the idea of non-native species when I found out that our campus hosts a mix of both native and non-native trees. Through my research, I found out that non-native trees have adverse effect on the ecosystem as they have higher reproductive abilities, can change forest structure, and they have deeper roots that can destroy the water level and soil fertility of an area. So I wanted to see whether my own school had fallen into this trap, because in India it's very common to plant ornamental or non-native trees in the name of tree planting drives, and while it's very well intentioned, it often ends up degrading the ecosystem even more, because these trees often lack the hydrology, topology and native predators that can keep them in balance. So while we have technology like Google Lens in our pockets, we need technology that can guide us towards planting of native species. So when I conducted a survey of the trees of my school, I found that out of the 77 trees present, only 26 trees are non-native. Or in other, and this is the tabulation of all the trees. Or in other words, out of 66% of trees are native and only 34% are non-native. So based on this, I recommend that schools and communities should focus on planting of native species, and knowing what's around us and making sure they exist there can lead to environmental conservation. And I would also like to point out this is a completely hyper-local app. And in conclusion, I would like to say that the problem of students being unable to identify local flora highlighted a larger issue of lack of environmental literacy, but my app aims to bridge that gap. By knowing what's around us and ensuring that we protect our environment one tree at a time, we can ensure that our world progresses to a better place. These are the references I used in terms of my secondary and tertiary research, including various scientific papers and websites. I would like to thank the organizers of this event for providing students such a large-scale opportunity to present our learnings. Thank you very much.
Don't you think that books are more human-friendly than your app?
So see, there are many species of trees, right? So if you want to be able to identify trees, like when you're going around, like such a huge campus is there. We went to Shobhavana also yesterday. If let's say those name tags were not there on those trees and you wanted to recognize it, would it be easier for you to have a phone or to have a thick manual with all the pictures that you would have to continuously flip through? This will take you exactly 5 seconds to get the answer, whereas plus with all the extra information, whereas your book may take you a lot of time to first look through and find the name of the tree, and then you would have to further look for information.
But I think so that's an interesting work for me to find out the plants. Instead of getting the information there itself.
This is just and it's easier, honestly speaking, to have a mobile app, because first of all, you would have to carry such thick volumes, and it's not really feasible when you are on nature expeditions, because it's always advised to pack light. Secondly, this will provide you more information. It's an offline app, so you don't require any network connection also in the Western Ghats. And it is targeted specially for locations that are biological, like hotspots basically. And certain plants of the Western Ghats are endemic species. You may not find them in common encyclopedias. So you would have to carry a variety of books to find out exact tree names.
Then in the forest area and all?
It's an offline app, so you can use it completely.
Last question, how you trained your app? How it is recognizing the things?
Okay, so initially I had wanted to put 200 pictures, but because there's a lack of space, because I'm making it on MIT App and Mentor, I took 40 pictures per tree of the flowers, fruits, vegetation and leaves, and I uploaded into the Google machine, the teachable machine, the training model. Then that uses transfer learning, which is, they already have the pre-trained model of how image classification works, and based on that they train all the other models. It's basically a simple classification model, and it is supervised learning. And what happens is that they use TensorFlow.js.
It's a supervised learning.
Yeah, it's a supervised learning.
So can you explain any classifier inside that which is classifying your data? Because I think it is limited. You gave some samples. Because enormous lot of trees in Western Ghats, 1,60,000 km square is the area. So it means it is limited to some species, because that girl shared she is interested in books because there is lot of documentation.
Yes.
So in our app there is some restrictions, it means your app.
So my app is just currently a pilot app. I cannot build such a large thing in such a small amount of time. It requires funding and it requires a lot of time also because so many pictures would be required.
Yes.
And there are various species also. So certain plants may look similar, so that requires little time to build it. And regarding the, so what was your first question before that?
Okay, I understood. It means you will train this model
For
again
that,
and
yeah.
again.
And I've also put, so let's say there are two plants that look similar. I have, out of these 4 species, let us say I have taken one plant that looks similar to these species and I have trained it. When I train it, I check whether it is identifying that as this species or it is not at all taking it. So that is how I have done supervised learning.
Thank you.
Thank you.
This is forward, backward, and long press you can focus on.
So good afternoon everyone, I am Kavanashri. I'm Charishma.
I'm Risika.
So our topic is about the macrophytes of Venkatadri Lake. We are from Lubar Public School of 9th standard.
Introduction. Our topic is about the study of macrophytes located near Central Jail of Bangalore South, Parapanagara in Hosaru near Electronic City. Venkatadri Lake is 1 kilometer away from our school, and our aim is to study different types of macrophytes and their role in the lake ecosystem. Venkatadri Lake has a footpath of 3 to 4 meter wide across its boundary, which allows us to view these macrophytes. And perimeter of the lake is like approximately 1.27 kilometers, and the area of the lake is about 13.96 acres.
And next comes the mapping of Venkatadri Lake. And next objectives. To study the objective, we have studied different types of macrophytes and their role in lake ecosystem, and to also study the economical importance of macrophytes, and to analyze the changes taking place due to the presence of these macrophytes, and to analyze the quality of lake water. So materials we used for this project was a phone to capture the images and the macrophytes of Venkatadri Lake, and a computer to search all the information, and methods that we used to visit daily around 4 p.m. to 6 p.m. around, and we have observed different types of macrophytes, and the main thing we observed there was a bad smell taking place, and then we got to know that there was a direct underground connection. Because of this, the lake was polluted and we did not see much macrophytes here. So we raised a complaint to BBMP and requested them to take an action regarding this project.
Macrophytes. Macro means big, seen with naked eye. Phyte means in Greek phyton, the plant species that normally found growing in wetlands either in or on the water is known as macrophytes. Macrophytes are large enough to seen by naked eye. Macrophytes are distributed in lake based on 2 constraints. The first one, water must be shallow enough for light to reach to the bottom, and the physical stability sufficient to allow plants to grow to the bottom, along with pH, temperature, inorganic matter, transparency, gas, saturation, conductivity, salinity, nutrition, concentration of water also affect in the distribution of macrophytes.
Functions of macrophytes. Macrophytes act as a base of food chain, and it also acts as a biological indicator of water quality. They link the inorganic environment with the biotic one through photosynthesis. They influence water chemistry, acting as nutrient sink through uptake and as nutrient pump moving compounds from sediment to water column. They assimilate nutrients directly into their tissue. Some macrophytes have medicinal values and they are useful for wastewater treatment through phytoremediation.
So next comes the type of macrophytes. This image shows the differentiation of the macrophytes. So there are 4 types of macrophytes. First comes the emergent. That means the plants rooted in lake but leaves and stems extend out of the water. Example typha and floating lake. That means the plants rooted in lake but leaves and stem float on the water surface. Example Ibomia aquatica. And next comes the submerged. That means the whole plant is under the water surface. Example Hydrilla verticillata. And last comes the free floating. That means the plant suspended on the water surface and roots are not attached to the bottom. Example Ulfia.
Floating macrophytes, Iconiac recipes, family Potideraceae, genus Iconia, common name is water hyacinth. Water hyacinth is a free-floating perennial aquatic plant with broad, glossy ovate and spongy petioles. And the flowers are lavender, which are blue in colors, and the leaves are fibrous and feather-like. These plants require more nitrogen, phosphorus and potassium to grow. These plants form dense mats which clog the waterways. This is also harmful for fishing as it displaces many aquatic grasses which were useful as fodder for cattle. Water hyacinth is used as fodder, manure, compost, biogas production, source of methane, alcohol and in python mediation.
And next comes the lemna, family Lemnaceae, genus Lemna, and common name common duckweed. So lemna is a free floating aquatic perennial plant, grows in high nutrient place. So leaves will be 2 or 3 each with single root. As leaves will grows hanging on the water, and plants reproduce by vegetative and form separate plant. The plants are used as compost and as fertilizer. These are food for fishes, birds and etc. Also it is used in wastewater treatment.
Ulfia, Lemnaceae, family Lemnaceae, genus Ulfia, the common name is common watermill. Ulfia are free floating aquatic plants with fronds that are nearly spherical to cylindrical in shape. They lack hair spaces or veins. They do not have roots. The flowers are rarely seen. Each flower has one stamen and one pistil. Reproduction is by vegetative and budding process. These plants are used in wastewater treatment and act as food because of its protein content.
Emergent macrophytes, typha, family Typhaceae, genus is typha and common name is cattape. Typha is an example for emergent plant are aquatic or semi-aquatic, rhizomatous and herbaceous perennial plants. The flowers are glabral, linear, alternate and stem bears flowering spikes of 0.7 to 2 inches. And male flowers make up the upper narrower half of the flower and the female the lower. Seeds are minute 0.2 mm long and are dispersed by wind. Typha is used in wastewater treatments and its leaves are used for weaving mats and roofs and are also a source of fuel. Its pollen is edible and it's also rich in carbohydrates.
Cyperus, family Cyperaceae, genus is Cyperus and the common name is nutgrass. Cyperus is an example of emergent plant and it is a perennial plant that reaches a height up to 140 cm. The leaves are radical. These weeds are commonly seen in cultivated fields, wetlands, river banks, and it is difficult to eradicate because it spread through tubers. These plants are used as food, medicine, act as a soil stabilizer, and some plants are used as ornamental purpose.
And next comes the alternator of phylloxerides. The classification of alligator weed, family Ambrataceae, genus Alternatera, common name alligator weed. Alligator weed is an emergent plant. Roots are fibrous, arising from the stems, nodes hanging freely in water. Leaves are elliptic and it reproduces by vegetative. Alligator weed is used as food, medicine, fodder and as compost.
Polygonum glabrum, family Polygonaceae, genus Polygonum, common name is marshbuck. Polygonum is emergent plants, grow up to 2 to 5 meter tall. Stems are erect, dilated and rarely branched. These plants are used as medicine for fever, jaundice, burn-ons, and its branches are used as vegetables, also used as insect repellent. Flowering will be on June to August, spiked 4 to 12 cm long like racemes.
So in Venkanada Lake we found 7 types of macrophytes, that is Iconiac recipes, Lemna, Typha, Polygonum glabrum, Alternatera, Ulfia, Cypress, and the water enters the lake through this inlet and it exits the lake through this outlet, and you can see there is a constructed wetland over here.
This is the distribution of macrophytes in Venkanada Lake. We can see large number of Alternatera 30%, Iconia 20% and Typha 20% and others are less than 20%.
And next comes the ecological importance of macrophytes. Macrophytes play a key role in structural and functional balance of an aquatic ecosystem by alternating water movement regimes, providing shelter to fish and aquatic invertebrates, serving as high protein food sources and alternating water quality by regulating oxygen balance, nutrient cycles and accumulating heavy metals. It is helpful in constructing wetlands for reducing nutrient loads and limiting algal growth. It acts as a biological indicator of nutrient levels in wetlands and it is used as a biofertilizer in rice cultivation.
In Venkanada Lake we had seen the all the surrounding buildings, sanitary wastewater and industrial wastewaters and plastics were accumulated in the lake. From this we can conclude that Vekarari lake is polluted and more plastics were accumulated. And macrophytes are a biological indicator of health and the ecological integrity of the wetland. This lake is polluted, that's why we had seen less macrophytes here. Role of constructed wetland. An artificially constructed wetland is an environmental friendly technology in which aquatic plants or a combination of plants and sediments are applied to remove nutrients and other contaminants from water. Aquatic macrophytes are more suitable for wastewater treatment because of their faster growth, greater biomass production, higher ability to absorb pollutants and a better cleaning efficiency due to direct contact with contaminated water.
Advantages of constructed wetlands. Wetlands are less expensive to build and operate than mechanical system. Wetlands are a passive system requiring little maintenance. No energy is required to operate a wetland. Results. The types of macrophytes that we have found in Vekarari Lake: emergent macrophytes, Typha, Cyperus, Alternanthera philoxerex, Polygonum, and the floating macrophytes, Iconia crespis, Lemna species, Wolffia, and we didn't found any submerged and free-floating macrophytes.
Macrophytes are bioindicators of water pollution and are used as an indicator of water quality. The lake is polluted, that's why we had seen only 2 types of macrophytes. So we raised a complaint to BBMP and we requested them to maintain a constructed wetland and they ensured us to the betterment of the lake. From this project we gained much knowledge. Conclusion. In Vekarari Lake we had seen only 2 types of macrophytes and in non-polluted lake we can see all the 4 types of macrophytes. From this we can conclude that Vekarari Lake is polluted due to the more and more plastics are accumulated in the lake. So we raised a complaint to BBMP and the BBMP corporator assured us to take an action against the betterment of the lake. Macrophytes are useful to humans. We need to make the better use of that, creating awareness among the people about macrophytes and their role in the lake ecosystem. We have to save lake and its ecosystem and also an environment.
So acknowledgement, we thank our principals and headmasters. A special thank to our guide teacher Lavanya ma'am and Chandrika ma'am. We wholeheartedly thank IIS team for giving this opportunity to work on this project. References used for this project. Thank you.
Can you please give the ecological importance of the wetland? Example.
Please can you repeat the question?
Ecological importance of our wetland. Why you wanted to conserve your wetland?
Because wetland is very important. If there is no, if the water is polluted, many aquatic species will die. And macrophytes help to clean the lake. So that's why the ecological is very important to conserve a lake.
Okay, thank you.
Thank you, ma'am.
Naintara, that is your paper. Thanks. Start.
Good evening to one and all present here. I am Navya and we are studying in 9th standard and our topic is environment literacy, need of the art. All of us know what is literacy. The ability to read and write is literacy. The literacy rate of Bangalore is 87.67%. Now, let us understand the meaning of environment literacy. The ability to understand nature, which includes water bodies, flora and fauna, is environment literacy. Knowledge, skills and understanding the problems to resolve issues to sustain suitable ecosystem is our responsibility because we are a part of the whole ecosystem. A survey conducted by Indian Institute of Science on environment literacy reveals that the literacy rate of Bangalore is only 3.5%. Sorry, there is a typing mistake. It's not 3%, it is 3.5%.
The two major problems that we are facing now is plastic menace and water crisis. To solve these problems, we have n number of innovative ideas. But why are we not able to resolve it? It's only because of environment illiteracy.
Please carry on. Don't stop. Carry on. Sunday, we have vegetable market in our place. And I found that many of the people were carrying vegetables in plastic cover. At the same time, many of the people were carrying vegetables in cloth bag also. When I saw this auntie, she was carrying vegetables in the plastic cover. When I asked this auntie, according to this auntie, not carrying a cloth bag while going for market is okay. Burning plastic is good. And she was not willing to listen to us. In the same way, when we asked about the source of potable water to 30 households, 90% of them answered as water tanker is the source of water. And they were not knowing anything about groundwater. I mean water table. Hence, our conclusion is that whatever the innovative ideas float, if people are environment illiterate, we will not be able to save our Mother Earth. Hence, the main problem is environment illiteracy. To solve this problem, the solution is environment literacy. The major problem of clogging of drains, polluted lakes, rivers, unhealthy roadsides and landfills indicates unscientific disposal of non-recyclable plastic materials. This problem can be totally resolved by using cloth bag while going for market and this discarded plastic can be used for asphalting the roads. This can be done scientifically by collecting and studying the used plastic with bitumen, and these roads would last longer and also maintenance free and also can get rid of pothole problems. In our school, we had plastic day wherein students used to store 1 week's plastic at home and bring it to school on a specific day like Monday for 5th standard, Tuesday for 6th standard and so on. We had collected 1600 kgs of used plastic and gave it to our vendor who used it for asphalting the roads. Mr. C. Srinivasan's Vellore model is the best in the country. According to him, only 10% of the waste goes to landfills.
The second problem which we took up is water crisis. Bangaloreans faced severe water crisis during last summer and Tanker Mafia played their best by charging 1000 and more for a tractor load of 4000 liters. To solve water crisis, we have ideas like borewell recharge, groundwater recharge and rainwater harvesting. For water percolation, afforestation plays a major role. Hence, we have 550 trees in our school in 4 acres of land. This is the Google map of our school. In our school, we have adopted borewell recharge, groundwater recharge and terrace water harvesting. Our school borewell is only 480 feet depth and has water year through. It is only because of the methods for recharge which we had done. This is the borewell recharging done in our school. This is the picture of terrace water harvesting. We have terrace water harvesting at 3 places. The first one is 10,000 liters, second one is 1,000 liters and third one is 2,000 liters. We have groundwater recharge pond in our school. Its bottom is not cemented, hence it allows water percolation. Our school's main motto is to prepare environment literate students because only environment literacy can save our mother nature. People like Dr. Shivakumar from Bangalore, who is well known as Waterman, is an environmentalist who never paid water bills and survived only on rainwater, are still there in our society. This itself says that we can survive only on rainwater. Python method to treat grey water is also available. Institutions and apartments should adopt this method. Dr. TVR Sir's Jakkur model, which is internationally acclaimed to treat water, is also available. Yesterday, TVR Sir in his presentation, Sir was telling about Sarangi Lake and reduction of temperature. Where is the shortage of ideas? Where does the problem lie? Problem is environment illiteracy.
Now we understood that plastic problem and water crisis have solutions, but both the problems remain as problems itself. I can only say that their severity has increased. Should we leave at this point to worsen the situation, or can we do anything? Yes, we can achieve it by conducting regular awareness programs at schools, colleges and self-help groups. Authorities as well as individuals should visit and replicate it in their place. And awareness programs should be conducted through street, place in all the villages, apartment complexes should conduct awareness in apartment complexes to reduce single-use plastic and rainwater harvesting. Hats off to Pollution Control Board because they have already started awareness program in our school and there were no POP Ganeshas were used by our school students.
Hence, we conclude that there are many innovative ideas to solve these two major problems. But whatever the innovative ideas float, if people are environment illiterate, we will not be able to save our mother nature.
Along with environment literacy, positive attitudes is also a must. So let us all be environment literate and save our mother earth because we have one and only one beautiful earth to live in. Together we can make a difference. Thank you.
Did you put plastic to asphalt the road?
Yes, we had given 1600 kgs for a vendor who used it for asphalting the Bangalore roads.
So like, how did the water percolate?
Which we showed groundwater recycling. That pond is not concretized, so that water percolation will happen.
If you put plastic on the road, how will the water go through to recharge the groundwater?
Sorry, we didn't say that we put plastic.
No. You said that you can use plastic to asphalt the road. Right. So when it rains, how will the water go underground?
It's totally different what you are asking. We said that we poured plastic for asphalting the roads, not for concretized or groundwater recharging, which we had done, not for that.
No, what the question is, we use plastic for asphaltation. How can the water pass through in that road? When through it, the water can percolate. That is our question.
You are taking the garbage from your home and you are throwing somewhere. That is the water.
No, it's not that. We collected plastic and gave it to a vendor who used it for asphalting the road.
Okay, fine. Next. Okay, fine. Thank you.
And one more. I carried a globe. No one asked why I carried that. I'll say. Because of due to many human activities, our Mother Earth is sick. So that she is sick, we carried. Sick people are carried, right? So to show that, to present it, I carried that. Thank you.
Good evening. My name is Nived. I'm a student of class 9G at BJS National Public School. Introduction. Access to clean water is a pressing global issue with many communities struggling. My project aims to address this issue by using natural and cheap methods to purify water. I will be using corn cobs and charcoal to filter out impurities. The method is eco-friendly and cost-effective. I will also use chemical filtration methods. I will compare both methods to see which works better. The goal is to provide an easy, effective and eco-friendly way to clean water. Objective: purifying lake water in an environmentally sustainable and cost-effective manner. For my project, I have chosen Ulamavi Lake. Ulamavi Lake map. This is the study area, and the purple pen shows where I collected water from. And here are the coordinates. Methods: to collect the water in BOD bottle, rinse the bottle with the sample water, then fill it completely, avoiding bubbles. Seal tightly to prevent air from entering, ensuring accurate results. Water was collected from Ulamavi Lake at 10 o'clock in the morning for accuracy. I use microscope for biological analysis. I use smartphone for clicking photos. Tests I have conducted: EC, which means electrical conductivity, biological analysis, physical analysis, temperature, TDS and pH, potential of hydrogen. Materials required for the test: I use a thermometer for the temperature, smartphone for clicking photos, BOD bottles for sampling, microscope for the biological analysis, TDS meter for the TDS, it is also present there. pH paper, beaker, gloves. Here are the tests I have conducted in the lake, specific tests I have conducted in the lake water. I have conducted tests of carbonate, calcium, sulfate, magnesium, phosphate, fluoride. All the 6 of them are present. Results for the tests showed before: biological analysis, microalgae and paramecium were found. Physical analysis, light yellowish green color. Temperature 23 degrees Celsius. EC, good conductor. TDS 329 ppm, which means part per million. pH 5. Methods of water purification. Concob method of water purification. So basically I use concobs which are very porous, which means they have a lot of tiny holes. It has a lot of tiny holes which will absorb the impurities. And then I will use charcoal, which will remove toxins like pesticides and metal. Also it will increase taste and odor of the water. Then I will boil the water, which will decrease the temporary hardness. And then sodium bicarbonate, and boiling the water will also decrease the microorganism, obviously. Then the water will be then sodium bicarbonate, also known as baking soda, will be added to increase the basically balance the pH. This 4-step process helps to make the water cleaner. Why concob? Basically, I am trying to explain the features of concob and why I selected concob for the specific project. They are very efficient, they are cost effective, they are eco-friendly, high mechanical strength and rigidity, and they are locally available. They are porous, which means they absorb a lot, and they are great absorber. So used concobs are usually thrown or burnt, and when they are thrown, animals do not eat it because they have a high cellulose content, and the same reason, so it takes a lot of time to decompose. Then it is burnt, which causes even more pollution, which is not acceptable. Used concobs can be used for a better project, purpose in my project. So in here first the water is poured through, and this is used pieces of concobs. So when the water is going through, the most of the impurities will get absorbed by the used pieces of concobs. Then it is then there are charcoal, which will absorb toxins, pesticides, metals, and there is a oil which water can come. I used tiny pieces of charcoal, which I used a mortar and pestle, and these were the pieces of concob, and this was the full entire thing. This process can be also used in a pond, lake, whatever you want. Yeah. So we can use we are going to use bamboo. Then we can cover it with some natural material. Then it will be tied to use pieces of concobs, and when the impurities are moving, it will get trapped in the porous structure of the concobs, which will make the water a little cleaner, not much though. Test I have conducted after water purification with results. Biological analysis, which I used a microscope for. Only dead cells were found in a microscope. Few charcoal fragments. Temperature 24 degrees Celsius. Physical analysis, clear water. Electrical conductivity, good conductor, TDS 249 ppm, pH 6.5. The chemical, I also conducted chemical method to see a comparison between them two. So in my chemical method purification, I used aluminum sulfate, which is also known as alum. So it is a coagulant. It is coagulant. So all the particles will become larger particles, also known as fogs. It will get settled down at the bottom. So then I'll do a decantation. Then I'll put 2 drops of chlorine per 10 liter per 1 liter. Which will kill harmful microorganisms. Then baking soda is added to neutralize minerals like calcium, magnesium, which decreases water hardness, it's a swelling. Also it increases the pH, balances the pH. This final step decreases the hardness of water and making it softer. This is the procedure. Tests we have conducted after chemical purification, biological analysis, only dead cells were found, temperature 20 degrees Celsius, easy, good electrical conductivity, physical analysis, clear water, TDS 268 ppm and pH was 8. This is the comparison study. So there is a significant decrease in both of them from the original amount, but we can see water which has been naturally purified is lesser than the water which has been chemically purified. References. These are the references. I would like to express my sincere gratitude to everyone who supported and guided me throughout this project on hydro purification. I'm especially thankful to my science teachers, mentor Chitralekha ma'am and Vijay ma'am, whose expertise helped me a lot. Also appreciate my family for the continuous support. Also thank my school management and IISC for this wonderful opportunity. Lastly, I extend my gratitude to the online resources and books that provided the necessary information to make the project successful. These are a few photos. Thank you.
So I have a few questions. I hope you'll entertain them. How effective is it for large-scale purification?
As you can see, it is not very effective, but it is definitely effective if we can do like this. It will not make the water drinkable, but it will at least make it cleaner. Yeah, this.
So you say it will not make the water drinkable. And from the readings that you displayed on the slides, I can observe very minimal changes to actually say that they've been purified. So the initial pH value for the first sample you took was around 5, very acidic, right? And then it pumped up to 6.5, but it's still acidic.
6.5 is drinkable.
I know it is within the pH standards, but it's not really close to neutral.
It is close. Neutral is 7 and it is 0.5 difference.
But the difference is not that much.
Yeah.
Then how many days it will take for the degradation? This form we will put like that.
It can be kept for I'm not exactly sure of the dates, but it can be kept for many years because it does not. It is at a very good mechanical strength, so it will not get out. And
okay, thank you.
Thank you, ma'am.
Good evening everyone, myself Dipshika from Bhakti Villa School, Bangalore. So So today I am here to present about the olive ridley turtle conservation. Olive ridley turtles are one among the smallest turtles found in India. They are usually found between December to May in the Tonka beach of the Honavar region. They are commonly found in warm waters such as Atlantic Ocean, Pacific Ocean and Indian Ocean. They are uniquely known for their mass nesting, where the female turtles arrive to the same beach as their birth and they lay their eggs and return back to the beach. The IUCN status of these turtles are vulnerable. My study area is the Tonka beach of the Honavar region. They are losing their nest because of a 4-lane road that is being constructed. They actually need a sandy shore to lay eggs, whereas the road is destroying the sandy shore. After hatching, usually the adult turtles return back to the sea, whereas the eggs have to hatch on their own and the little ones have to reach the sea by themselves. But on their way from the nest to the sea, they are being eaten by dogs, and this is also a major problem. My objective is to spread awareness about the conservation of these turtles and also to create a better and safer nesting area for them. This is the map of my study area. Here, the yellow markings mark the nesting location of the Honavar range. In this observation table, it's clearly visible that from 2014 till 2020, there is a massive decrease in the number of eggs hatched compared to the number of eggs that have been laid. Whereas from 2020 till 2022, the higher number of eggs have been hatched. We can call it the most number of eggs that have been hatched, because most probably due to the lockdown crisis, there were lack of human activities during those times. They're planning to construct a 4-lane road to transport manganese and other substances across other countries. They also claim that once the road construction is completed, around 1,500 lorries would travel every day. Frequent travel of ships and lorries can cause heavy water pollution. A company called HPPL, Hyderabad Port Private Limited, claiming to be Honavar Port Private Limited, is leading this project. They have put up a HTL line, that is the high tide line, but locals usually claim that the water reaches greater distance than the HTL line that they have put up during heavy rains. These are the picture galleries. In this, the first 3 pictures were collected from locals, where the first picture shows a dolphin lying dead on the Tonka beach, which proves the existence of dolphins in that beach, whereas the second picture shows the artificial protection constructed by the locals to protect these nests, and the third picture shows the water overflowing the houses and crossing the houses too, and the HTL that they have put up. The next 3 pictures are of the current situation of the beach. Here, in this picture, the first picture is about the footprints of those turtles, and the second picture is about the eggs laid by them. My recommendation is that they can shift this project to a better location where it won't affect any marine species, and also fencing of the nesting area would help these turtles get saved by the dogs. As per TVRAMACHANDRA's suggestion, I have also mailed to our Honorable Prime Minister about this issue, and also I have mailed our Transport Minister, that is Mr. Nitin Gadkari sir, about a similar issue facing in the Sagarmala project as well. I would like to conclude by saying most of the port construction projects are facing resistance by the locals, mainly due to threat to livelihood and threat to biodiversity. This is not just limited to Karnataka. Under Sagarmala project, there are many projects that are coming up and are facing opposition by the locals. This scenario is playing out almost in every coastal state of our country. They have tested and submitted a false report that there is no such nesting area in this beach, whereas the time of this survey was not the nesting season. And they have also informed the court that these turtles lay eggs way inside the sea, but it's a known fact that turtles don't lay eggs inside the sea. Between legal protection, dead olive ridleys were found and no action was taken. They have also implemented focus lights to avoid the arrival of these turtles. I would like to thank my mother for her continuous support for my project, and also I would like to thank our chairman for his encouragement. And I would like to thank IISC and ALVAS for this important opportunity for me. And I would like to thank R&D department for their continuous support. I would like to thank Mr. Sandeep Hegde and Dr. Prakash Mesta for their information that they have provided. I would like to thank the locals as well for their support as well. Thank you everyone. Thank you, ma'am. Thank you, sir.
A good day to one and all present here. I am Sanvi Srinivas Rao.
I am Sharika Arun.
And we are studying in 9th grade in Vidyaniketan Public School, Bengaluru. Today we are here to present a comprehensive comparative study of water quality of two different lakes, that is the Ullar Lake and the Ramchandra Lake.
A lake is a water body surrounded by land on all the sides. Studies done on Bangalore's lake says that nearly 98% of lakes are encroached and 92% of lakes are sewage fed. More than 40% of lakes are affected by eutrophication. Survey conducted by Karnataka State Pollution Control Board says that out of 167 lakes, there were samples collected from 41 lakes and more than 39 lakes are classified into Class C, which is not suitable for fishery. We can see that not even one lake falls in A or B category, which is suitable for fishing. Our objective is to compare the two lakes, which is the Ullar and the Ramchandra Lake, by analyzing the physical, chemical and the biological parameters. Now let us look at the study area of the two lakes. Ullar Lake is situated in the Ullar village of southwestern part of Bangalore city. It falls in the Vrishabhavati valley within the Bairamangala lake series. It covers an area of about 10.238 hectares and perimeter 1.57 km. Ramchandra Lake is situated in the Ramchandra locality of southwestern part of Bangalore city. It lies in the outskirts of Rajarajeshwari Nagar zone of lakes. It covers an area of about 1.96 hectares and perimeter 0.58 km. We did the mapping using the Google Earth Pro. This is Ullar Lake mapping and this is Ramchandra Lake mapping. We tried to compare the Ullar Lake's perimeter over the years. In 2008, it was about 11.03 hectares and presently it is 10.17 hectares. We can see that there is a reducing size of the Ullar Lake. This is due to the reduced rainfall, encroachment, pollution and rapid urbanization.
Now let us look at the various on-site and the off-site parameters which were tested across both the lakes. The on-site parameters include temperature, pH, TDS and color of the water. The off-site parameters include calcium hardness, magnesium hardness, total alkalinity, chloride, fluoride and dissolved oxygen content. The sampling and testing techniques followed are, we as a team went to both of the lakes and collected 3 different samples from each of the lakes. We collected samples from the inlet, outlet and the middle of the inlet and outlet in both of the lakes. We tested them for various physical, chemical and biological parameters, that is microbes, using a compound microscope. We used various testing techniques for the physical and chemical parameters. They are TDS meter probe and ready testing kits provided to us by the school, pH paper, and we also used the Brinkler's method to determine the dissolved oxygen content.
Now, looking at the on-site parameters, water quality assessment is very important to monitor the lake. We measure the TDS, pH, color of the water and water temperature. The TDS in Ulla Lake in our first observation was 163, 172 and 178 ppm respectively in inlet, center and outlet. We can see that the range of the TDS is from 500 to 2000 ppm. But in Ulla Lake, it does not exceed the upper limit. And in Ramchandra Lake, it also lies about in the same range, but it does not exceed the upper limit. The pH range for a better quality water is from 6.5 to 8.5. In both the lakes, it lies within the range. In the second set of observations, we observed that in Ulla Lake, the TDS has been increased to 206, 202 and 201 ppm. We observed this change after a period of rainfall. But in Ramchandra Lake, it was about the same range as it was before. And the pH in Ulla Lake center has been increased to 9. It has exceeded the upper limit and become more alkaline. And in Ramchandra Lake, it lies within the range.
Now, let us look at the offsite parameters of both of the lakes. The total alkalinity, total hardness, chloride and fluoride content of both of the lakes fall below the limit, which is 200 to 600 ppm for total alkalinity and total hardness, 1 to 1.5 milligrams per liter for fluoride and 250 to 1000 milligrams per liter for chloride. The calcium and the magnesium hardness of both the lakes fall within the range, which is 30 to 100 ppm for magnesium hardness and 75 to 200 ppm for calcium hardness. However, the dissolved oxygen content of Ramchandra lakes fall within the range and that of Ulla Lake is below the range. The range is 6 milligrams per liter or more. These are few images of the chemical parameters analysis. We found a variety of microbes in the Ulla Lake water sample. Chlorococcum is a genus of green algae. It is useful in the flocculation. Useful in the flocculation of lipids from wastewater. Oscillatoria is a type of cyanobacteria, and diatoms are photosynthesizing unicellular algae. We found a variety of flora and fauna around both the lakes. They are listed as follows.
We observed pollution in both the lakes, which was mainly caused by sewage and plastic. We can conclude that the Ulla Lake's perimeter has been decreased significantly over the years, and there is rapid increase in pollution, which was signified by the surroundings and microbes found, and there is no proper sewage treatment.
Few of the conservation ideas which we can follow to improve the water quality of lakes are: we can introduce bacteria which can break down plastic. Ideanella sacchinensis is a bacteria which produces hydrolyzing PET. This enzyme breaks down polymers into monomers, which is then used as energy. We can also implement the blue-grain water treatment system, which inculcates the reed bed technology. In the reed bed technology, we grow reeds, which is a type of grass over a piece of land. Wastewater is allowed to percolate through the reed bed. As the reeds try to absorb wastewater, they dissolve oxygen into the water. This increases the microbial activity and hence increases the amount of breaking down of pollutants and effluents in wastewater.
These are few references from which we collected the information.
We would like to thank the IISC team and TVRAMACHANDRA for providing us this platform. We would also like to thank our school, Vidyaniketan Public School, our teachers, our mentors and our parents for their continuous support and guidance.
Thank you.
Good morning everybody, my name is Bhavya Durga and I am from Boromira International. I will be presenting the topic, a comparative chemical analysis of Alsu Lake. Now to start off, Alsu Lake is a lake located in central Bangalore, encompassing an area of over 50 hectares. Now, my reasons for selecting the topic would be Alsu Lake's proximity to the school, which allows me to collect for seamless sample collection and testing. The drain which flows close to Alsu Lake really had me thinking, what would happen if it one day overflows? What would happen to the fish inside? Along with that, the water color of the lake also piqued my interest. My objectives for this chemical analysis were to analyze the pollution status of Alsu Lake, to find out if there are any trends in the specific chemical parameters, to discern the causes of pollution for Alsu Lake, to help people become aware and to take action if necessary. This is the terminology I have used: pH, TDS, total hardness, alkalinity and electrical conductivity. Now the causes of pollution for Alsu Lake is one of them is the overflow from the Nala or the sewer, which is closely located to Alsu Lake and which regularly floods on rainy monsoon days, with the garbage seeking into the lake. Now the breaking of the bund also is another important factor, which had released several impurities into the lake. Along with that, there are slums located close to the lake, which frequently dispose of their waste in there. Now the BBMP has taken many steps to improve the condition of Alsu Lake after the fish killing incident which happened. Now the cleaning is to be done weekly or monthly, and by machines monthly analysis is also be taken, and many cleanliness drives. This is a map of my study area. This is where I have collected the samples from. Now I had collected samples on 16th of July 2024. Sample 1 was collected near the main entrance, sample 2 in front of the fishing hut, sample 3 near the Kalyani. Kalyani is a separate point in Alsu Lake which is used for Ganesh Chaturthi idol conversion, and sample 4 was taken in the segregated area to grow saplings. Now the on-site parameters, I had noticed that in sample 3 I had taken from the Kalyani point had a strong, very pungent, fishy-like odor. The rest of the lake had none to mild odor. The average water temperature was found to be 23 degrees Celsius. The pH average was found to be 7.16. And we have observed some slight variation in the TDS, with the highest being in sample 3 collected in the Kalyani. And the electrical conductivity was found to be 396 microsieverts per centimeter. These are the off-site testing results. The off-site testing was done in Scientific and Industrial Research Center in Bangalore. These are the results for sample 1. Now I have conducted a comparative chemical analysis of the samples taken from Alsu Lake in a very specific point through the years 2015, 16, 2023 and 2024. Now this is the temperature analysis. As you can see, the temperature remains stable at around 23 to 30 degrees Celsius. It is also an indicator that the lake's health is in quite good health. Now the pH, we can see that the pH comes down from the 7.9 to the 7.1, which is great for Alsu Lake, and also shows how BBMP is working on improving the lake's condition. The TDS analysis, now we can see that in 2021 there has been some great fluctuation in the TDS. This was due to an incident that had happened in Alsul Lake, where the nearby dolphin aquatics had dumped the dirty swimming pool water inside the lake. Now this caused quite a ruckus, but then the TDS has slowly come down to 186.78 mg per liter. The calcium hardness levels are generally within the range that are found for lakes and are pretty good in 2024, which are found to be 40.04. The magnesium hardness shows some fluctuations, but then it evens out as 32.02 in 2024. Now the total hardness values fall into the moderately hard category, which is pretty good for lakes. This also indicates that the lake is in good condition to sustain its aquatic life. Now the alkalinity analysis is an area for concern. As you can see that the alkalinity levels in 2024 have dropped to over 84 mg per liter. But then the alkalinity as is dropping is an indicator that the pH levels are susceptible to many fluctuations, and these probable pH fluctuations can really stress or damage the aquatic life in Alsul Lake. Now the BOD levels we have found to be quite high near the sample 1 collection spot with 18.5 mg per liter, but then the BOD levels can vary from the spots taken in the lake, and sample 1 was collected near the shore, which can affect the BOD levels. The increase in BOD levels can also lead to fish deaths and algal blooms. Now the chloride analysis, the rapid decrease in chloride levels could potentially stress the aquatic life in Alsul Lake, but then it has stabilized out to 53.98 mg per liter, and it also means that the lake condition is improving due to efforts too. This is where I have interpreted my results from. As you can see, Alsul Lake falls within class D of lakes in fish culture and wildlife propagation. I would like to conclude from all that data that we have collected that the overall health and condition of Alsul Lake is improving rapidly. The BOD and alkalinity fluctuations do cause an area for concern, but still remain within the limits. The conductivity, chloride and calcium hardness are very stable. Along with that, the total hardness, magnesium hardness and pH levels have improved and are very good for Alsul Lake. These are the references that I have used. I would like to acknowledge my teachers, my friend Bhavna, and my elders Vijayavara for their guidance. Special thanks to the BBMP and Alsul Lake Management for seamless collection. Thank you. Any questions?
In one of your graphs I saw that there was a rapid decrease in the temperature. So why could it be like that?
Oh, the rapid decrease in temperature, maybe it's like just due to the seasons. We collected it in July, and in July there was some, in Bangalore, in Alsul Lake area, it was quite cold. So it doesn't, if the temperature drops to like maybe 30 or if it goes above, then it's a cause for concern. But then it's quite stable over here.
Okay, like in 1 year it rapidly decreased, so I asked. Thanks.
No, this could be due to like the season it was taken in. Maybe it was taken in the summer season and ours was taken in like July when it was like raining and the temperature was cold.
The conclusion can't be drawn with your one-day collection of samples, right? Conclusion cannot be drawn based on one-day collection of samples.
No, the conclusion was that if the lake's health is improving or going down based on the years. So if it's going really down, then that's a cause for concern. But we have seen that it's pretty good. So it
over the years you are telling, over the years also you have to compare it with seasonal data, right? One you cannot have it in January, one you cannot have it in June, and compare and see, draw conclusion, right? Your sampling was done one day in 4 locations you have collected. Based on that, how can you conclude? For her thesis, she is taking data of 9 years collection, seasonally and monthly data, and different timings. That's what I am
No,
saying.
but I was just doing like an overall, not very specific.
Exactly. Conclusion cannot be based on one single day collection. That's my comment. I am not saying you are wrong or right or whatever.
Any other things?
No, thank you.
Thank you.
Good evening everyone, I am Jyoti.
I'm Anushri Jiyam.
We are from Dubai Public School of 9th Standard. Today we are going to talk about the topic water analysis of Venkatadri Lake. Our topic is about the study of the lake water present in Venkatadri Lake near Centerjail, Parapanakarara, Bangalore South. The lake has a footpath of 3 to 4 meter wide that allows us to leave the lake. Its perimeter is 1.27 kilometer and its area is 13.96 acres. Our objective of this project is to analyze the water quality and the effects on aquatic creatures, analyze the pollution status of the lake and then to take action on the polluted lake. Materials we used for this project is phone parameters, chemical solutions, BOD bottle, etc. We used to go at 7 am to 8 am to collect water sample. We used to get bad odor. Then we got to know about the direct underground connection. Then and there we raised a complaint to BBMP Corporator to work on it. This is the mapping of the study area we have taken, that is Venkatadri Lake.
Water body. The water body is the area which is covered with water and it also plays a crucial role in balancing the ecosystem. Importance of water body: it acts as a source of drinking water and it also supports biodiversity for humans, aquatic ecosystems and represent a source of food, water, materials as well as commercial fishing and tourism. Another huge importance of water body comes with the aquatic plants and animals and their survival in the water. Water sample collection. Wear gloves while collecting the water sample and rinse the BOD bottle 3 times with water sample and fill the bottle within 1 to 2 inches from the top. Collect the water sample from the inlet point, outlet point and from the middle of the body. We have to take the readings of water and air temperature near the lake. Physical parameters. The collected sample had the physical parameters. Color. The water sample was slightly yellow. Odor. It had a bad odor. Turbidity. The collected water sample is turbid because it contains suspended and colloidal matters such as organic and inorganic matter and other microorganisms. Transparency. The water was not transparent because the water was turbid. Temperature. The temperature of the water was 22.7 degrees Celsius and the temperature of air was 21 degrees Celsius. On-site water quality analysis: EC, TDS, DO and pH. pH, potential of hydrogen. It measures how acidic or basic an object is. It has values ranging from 0, the pH of 7 is neutral, greater than 7 is basic and less than 7 is acidic. The pH of water increases during daytime and it decreases during the night time due to photosynthetic and respiratory activity. The pH of our water sample was 7.3 to 7.10, so that we can conclude that it is not fit for drinking.
EC, electrical conductivity. EC is the measurement of water's ability to conduct electricity, and water with high EC is unsuitable for irrigation. The electrical conductivity can indicate that the water is polluted, and the value ranges from 1350 to 1440 microsiemens per centimeter. So the value range more than 1000, so it is in saline condition and impacted by industries. TDS, total dissolved solids. It is measured in parts per million. High concentration of TDS may reduce the water clarity and decrease the photosynthesis and increase in temperature. The value range from 600 to 752 ppm. So it is more than 500 and it is not fit for drinking. Dissolved oxygen. Oxygen is essential for all the forms of aquatic life. The water with low DO is not acceptable for aquatic animals and plants because they doesn't get the sufficient oxygen to survive. The value range from 4.2002 mg per liter to 7.827 mg per liter. So it is stressful for fishes and acceptable for spawning.
Titration and calculation that we did in our school lab. We have taken the water sample in 125 ml of BOD bottle and we added 1 ml of MnSO4 and 1 ml of Ki and 1 ml of concentrated sulfuric acid to make the precipitate clear. Then we took the 25 ml of the water sample and we added stats indicator to it. The sample becomes blue, then titrated against sodium thiosulfate solution. The end point is blue to colorless. This is the calculation of DO and the value was 7.827 mg per liter. Water hardness. Water hardness is the amount of dissolved calcium and magnesium in the water. Hard water is high in dissolved minerals like calcium and magnesium. Temporary hardness can be removed by boiling, but permanent hardness cannot be removed by this method. Water is considered hard when its pH is above 8.5.
BOD, biochemical oxygen demand. BOD is the amount of oxygen consumed by microorganisms. Water pollution. Water pollution is caused by the harmful substances entering the water source such as industrial waste, agricultural waste and other waste. This substance does not change the color and it's invisible and can be harmful for their usages. The overgrowth, aging and decaying of macrophytes affecting the death of fishes in the lake occur due to the water pollution. Major effect caused by water pollution. Diseases like cholera, typhoid, hepatitis, diarrhea, etc. As water is an important element of human health, the polluted water directly affects the human health. Discharging of sewage and liquid waste of agricultural land, household, factories and into rivers and lakes can pollute the water body. We got to know about the water quality and the effects on aquatic creatures. We analyzed the lake water and studied the water quality, and then BBMP Corporator ensured us the betterment of the lake. We gained much knowledge from this project. These are the physical and chemical parameters result we have got. This is the result of Venkatadri Lake and compared with standard values of parameters such as temperature, pH, EC, TDS and DO.
Conclusion, we visited the nearby Venkatadri Lake with our teachers. By this we can conclude that the water was polluted. We can minimize the use of chemical fertilizers and pesticides with sustainable farming methods. We should recycle and use eco-friendly products. Conserving water should be our first aim. We like to thank our principal and headmistress and our teachers of our school management. I would like to express our special thanks to our teachers for their efforts for giving this opportunity. We wholeheartedly thank IISC team for this opportunity to work on this project. These are the references which we used for our project. Thank you.
Can you please tell me the importance of dissolved oxygen?
So dissolved oxygen play the crucial role for aquatic creatures. If it is less than 4, the aquatic creatures doesn't survive because of the lack of sufficient oxygen.
Okay, thank you.
Thank you. Thank you.
and 10.
Good morning to one and all present here. My name is Prabhav Nandedkar and I have done a comparative study of Shilevanta Kere in the year 2022 and in the year 2024. Before going through anything else, first we should understand the importance of conservation of lake water. Shilevanta Kere is connected to different lakes in the area through channels called Rajakalve. So if one lake is contaminated, the other lakes will also get contaminated. Groundwater is also majorly dependent on lake water. So a contaminated lake water may affect the groundwater which we consume. Originally, Shilevanta Kere was a barren lake with low number of trees and birds. But in the year 2011, many corporate employees took initiative and planted over 1,000 saplings, converting this lake into one of the most beautiful lakes in Bengaluru. Shilevanta Kere is located in Ramakondanahalli, Whitefield, Bengaluru. Its perimeter is 1.2 kilometers and its area is half a kilometer. These are its latitudes and longitudes. The objective of my project was to compare on-site as well as off-site parameters of the lake in the year 2022 and in the year 2024 to find out the causes of contamination of lake water and suggest preventive measures. I used the Jaltara water testing kit to test on-site as well as off-site parameters, and I used a sterilized bottle as a sample collection method. The on-site parameters that I had tested was turbidity, temperature, pH, odor and color. The turbidity had gone from 25 NTU to 100 NTU in the year 2024. The lake has also become more acidic, that is, the pH level had gone from pH level 8 to 6. The odor had also gone very bad. This caused many people to stop visiting the lake. The color of the lake is now algae green. The off-site parameters that I had tested were iron, phosphorus, ammonia, nitrate, chloride, residual chloride and faecal coliform. In the year 2024, we can see an increase in iron, ammonia, nitrate and chloride. These are the test results. Through the tests that I had conducted in the year 2022 and in the year 2024, these are the list of problems or the causes of contamination of lake water. Sewage from the surrounding area is being drained into the lake. New societies like Sumadura Folium have been built very close to the lake. Fishing, many instances of illegal fishing have been seen. The fishing causes a decrease in number of birds and hence affects the ecosystem of the lake. This is the sewage drain that leads into the lake. The illegal fishing caught on camera and Sumadura Folium Society. We can prevent further contamination of the lake by diverting the sewage into a sewage treatment plant. We can prevent any further encroachments or constructions in and around the lake bed. We should stop fishing as it affects the ecosystem of the lake. More encouragement of corporate social responsibility activities, more active green tribunal, more facilities in the lake, stricter laws and regulations, and deeper education and awareness drives. I acknowledge R&D department and Vagdevula school to give me this wonderful opportunity to present in this event. I thank IISC for this wonderful initiative, and I thank the people who have helped me throughout the project. Thank you.
Can you please elaborate one problem facing in your lake?
What do you mean?
Any problem. You can take anything. In here you are telling no.
So what am I supposed to do? Elaborate.
Which one is you like? This 4 points, one point you can. Fish death or sewage water.
Am I supposed to explain myself?
Yes.
Okay. So the sewage from the surrounding areas, so new apartments and all, they don't care to properly dispose the sewage. So they directly dispose sewage into the lake. And also there is a slum near the lake, and the people of the slum, because they are uneducated, they also dispose solid wastes and all other chemical waste or anything it might be into the lake.
Thank you.
Just to know, like, Vinay called and told those who have completed should go to the IISC hall because the cultural program will start. Certificate can be issued tomorrow because chairman is coming towards 6:30. Did you retain the 5 people who has to make the present? You can announce. I will check with you. Mike.
There is an announcement. Silence, please. There is an announcement. In V.S. Acharya Hall, there will be cultural program going to start right now. Those who have completed can leave this hall. Those who have not yet made the presentation, please remain here. We have to complete the presentations. Your participation certificate will be issued tomorrow. Okay? And if you are leaving by flight or any other arrangements are done, then your teachers will collect your participation certificate. So without making any noise disturbance, please leave the hall, those who have completed. 1 minute.
One minute. We will take one group photo. After that you can leave. Come. Please come fast.
They can stand here. If they can stand, they can stand. Everyone can go up. We can be here. Quickly. Come. It's okay.
Technically last. I told you.
No, you cannot. In our room, I take.
Those who are short, please stand front. Height-wise, you please go back. Or kneel down, or
We can stand here now.
Now you can come.
Yes, sir.
Please.